and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly.
Good morning. Welcome to day 4 of the 10th annual Regulatory Education for Industry Conference, affectionately known as REdI. I'm Joe Tartle, Deputy Director for the Division of Industry and Consumer Education, DICE. And again, it's a good morning, at least for me, or afternoon or evening, depending on where you all are located. This morning, we'll wrap up part 2 of the medical devices track, medical device product on the market. So far, it's been and will continue to be a fantastic program. But before we can start our program, I do have a few different items that we need to get to. So first, let's hit on our learning objectives. As you can see in that first bullet, we're going to have to discuss a couple housekeeping administrative items as it relates to the conference, especially continuing education credits. Then secondly, we'll review the part 2 devices track agenda, and then we'll meet today's speakers. And then last, I want everyone to realize your call to action. So REdI, housekeeping items. First, we're going to go through speaker and final evaluations. Then we're going to talk about the recordings, and then we're going to talk about continuing education. So first, evaluations. During each presentation, we'll have a link to a speaker evaluation under the speaker's picture or information on the right side of the screen. Please take time to complete the evaluation, as your feedback is important to us and helps us to both be able to look back and do after action reviews of this particular conference, as well as plan for future conferences. So please complete those. Also, in addition to the speaker evaluation, we'll have a final evaluation. The link on sbiaevents.com should be active now or by this afternoon, and in an email coming soon. Please note that this final evaluation is only available for 2 weeks. Also, make sure to get your certificate of attendance. And be aware that all the sessions are being recorded and will be made available to you after the conference. You can find them at www.fda.gov/REDI 2021. Okay, so please utilize those sessions. That's one of the best things about being registered, is that you can go back. It's not just, oh, did I hear that right now? You can go back and supplement your notes and update things and listen to those recordings again. I know that I've done that in the past with regards to the conference, to make sure that even with questions and answers that, yep, that's what I heard, that's what I thought we answered. So please access and utilize those recordings. They are available to you. With that, we'll move on to continuing education, or CE credits. The full 5-day conference offers a total of 30.25 continuing education, or CE credits, to attendees who self-identified themselves as healthcare professionals during registration. You need to attend in real time and through the Adobe Connect platform. Unfortunately, CE credits are not provided if you're watching on the YouTube link or after the conference ends. All faculty are expected to use generic names. If trade names are used, those of several companies should be used rather than only that of a single supporting company. CE faculty and speakers are required to disclose to the attendees when products or procedures being discussed are off-labeled or unlabeled, that is, not FDA approved, or in the case of medical devices, not cleared or not granted, basically something that is off-label or unlabeled, and any limitations on the information that is presented. There's only one disclosure for today's program participants, and that includes both the CDRH and CBER track for today. And that disclosure is as follows. It's me, Joseph Tartle. My spouse receives an hourly wage from New Horizons Diagnostic Corporation as a contractor and manufacturer. To obtain CE credit, participants must attest to their attendance and complete the final activity evaluation through the CE portal, ceportal.fda.gov, by August 7th. You'll need to complete the faculty evaluation for each day. Each track will have its own survey evaluation link. When an attendee completes the survey evaluation, they can then download a certificate of attendance, which can be used in support of RAPS, SOCRA, ACRP, and SQA credits. For any pharmacist and pharmacy tech in attendance, please be sure to provide your correct NAPB and date of birth information in the required format. Your NAPB profile number should be the 6 to 7-digit profile number assigned by the CPE monitor, and your birth date should be in the month date format. For example, 0721. Do not provide your pharmacy license number. Please click the My Account tab and then navigate to Edit Contact Information to verify that your information is correct. So please, if you're going to get this continuing education credits, follow all the directions that were provided. So with that, we're going to move on next to our agenda for part 2. So as we continue with our theme of product on the market, so yesterday we heard about the recipe, and then we talked a little bit about what happens when that dish is being made and served to you. We're going to continue with that product on the market with the following Thursday 22nd topic areas. So first off, Jake Dyer is going to talk about the Medical Device Single Audit Program. So Lieutenant Commander Dyer is going to get into MDSAP and give a great presentation with regards to that program and an overview of it. Then we're going to transition, and Fariba Maraka is going to talk about the public MAUDE database and give an overview of the public MAUDE database, which gets used by a lot of folks, not just you guys in industry, but we get calls in from consumers all the time with regards to wanting to look up medical device reports in MAUDE. And Fariba is going to talk about that. And then we're going to talk about, we'll have a short break. And then after that short break, we're going to come back and talk about additive manufacturing of medical devices, 3D printing. This is a very interesting talk. I've actually heard all of these talks, and this one in particular, I learned an awful lot. I knew a little bit about 3D printing, but not nearly as much as you're going to hear about later this morning. So Matthew DiPrima is going to talk about additive manufacturing of medical devices. Very interesting, and I'd like to say new, but it's been around for a few years now, topic. And then we're going to close out today with a representative from our Office of Regulatory Affairs, Meredith Andrews, who's going to talk about an FDA review of Class 1 and Class 2 recalls. We're going to hear about those all-important aspects of recalls as well. So please hang on for the entire device track. And then I'm going to close out and end our Center for Devices Radiological Health devices track, and then we're going to hand it over to our counterparts in the Center for Biologics Evaluation and Research and break for lunch. So that is the agenda for part 2 today. So like I said, we're first going to talk about the Medical Device Single Audit Program. Then we're going to talk about public MAUDE. Then we're going to talk about additive manufacturing. And then we're going to wrap it up with a review of medical device Class 1 and Class 2 recalls. Very great topics, very timely topics for what's going on these days. And again, I'm going to be your moderator for the question and answer sessions. And Tanya Wilbon will help me by being our online question and answer moderator. So again, a nice round of thanks to Tanya. Now, before we get into our first topic of the day, again, I do want to hit on some FDA organizational acronyms because, as Elias noted yesterday, we are with the government and we do like to use acronyms. And unfortunately, there's a lot of acronyms out there. So I want to try to keep everyone straight on the different acronyms that they hear, especially as it relates to our particular organization. So the Office of Communication and Education within the Center for Devices and Radiological Health. That is where our division is located. You also hear from a few people from the Office of Product Evaluation and Quality. This is the super office now in the center. You'll hear from folks from ORP, Office of Regulatory Programs. They are located in OPEC. You'll also have heard and will hear again this morning from a representative from the Office of Science and Engineering Laboratories. So yes, we have our own laboratories within the center, and they do a lot of great work with regards to a lot of cutting edge stuff. And then, of course, we'll wrap up today with hearing somebody from the field, which is the Office of Regulatory Affairs. So you'll get a chance to hear many different people from many different perspectives throughout the FDA agency and our organization. So with that, I'm going to end it with the call to action. So what do we want you guys to get out of this? First, we want you to enjoy this program. This program is all about you. This is the reason why we spend time putting it together. It is the reason why we bring all of these knowledgeable speakers together is for you. So we want you to enjoy. We want you to interact. We want you to ask your questions, which we'll get to as many of them as we can, and we want you to learn. I know, again, I've been coming to these for 10 years and I learn something every time. So please ask us those questions. This is your program. And take advantage of the FDA resources that are being offered here today, as well as the FDA resources that we've been giving you and we will continue to give you throughout the program. FDA, we try as much as possible to be as transparent as we can, and we always are approaching things from a scientific, science-based information. Just like everyone that's in these virtual rooms, we are scientists ourselves, and we like to approach everything from a scientific standpoint. So we love information and data, just like we know you guys love information and data. We try to post as much of that information as possible. So please take advantage of all the FDA resources that are available, whether they're guidance documents, educational tools, or data that's being provided. And of course, as we talked about before, please give us your feedback on what you need. So those speaker evaluations, the final evaluations, give us the information, because again, we'll take that data and then we'll utilize this to improve these programs, because we want to have better programs for you. Okay. So without further ado, I'm going to introduce our first speaker. So our first speaker of the day, Jake Dyer. He's a lieutenant commander in the U.S. Public Health Service. Jake currently works as a senior regulatory officer in the Regulatory Inspections and Audit Team in the Division of Regulatory Programs, DRP2, in the Office of Regulatory Programs in OPEC. Okay. He currently serves as a program analyst and assessor for Medical Device Single Audit Program, and he leads the efforts associated with MDSAP development and implementation with both us and other regulatory authorities and the industry. Jake began his career with FDA over 5 years ago as a medical device investigator, and is also a licensed professional engineer and a lean design for Six Sigma black belt, as well as a certified project manager and certified quality auditor. Lieutenant Commander Dyer earned his degree in mechanical engineering from the University of Rochester. And with that, I will turn it over to Jake.
Thank you for the introduction. Hello, my name is Lieutenant Commander Jake Dyer. I will be providing an overview of the Medical Device Single Audit Program and talking about how the FDA utilizes this data in pre-market and post-market activities. Thank you for joining us. If you have been in an office environment, then you are probably familiar with the comic Dilbert, written by Scott Adams. I thought this comic strip was relevant to the discussion of audits. Take a few seconds to read the strip. So whether you have had an MDSAP auditors, FDA investigators, or ISO auditors at your facility, you don't want this to happen. FDA investigators have seen the good, the bad, and the ugly. I should know, I once was one. The key message is that MDSAP audits are predictable in terms of approach and timeline, so you should be well prepared when the audit team visits your facility. During today's session, our learning objectives will be reviewing MDSAP updates, describing how FDA uses MDSAP, and we'll go over the MDSAP transmittal that describes extraordinary measures and remote auditing. It can't be an FDA presentation without acronyms. The first one is the Medical Device Single Audit Program, often referred to as MDSAP. Next, we have regulatory authorities, or RAs. These are the organizations like FDA and Health Canada. Then we have the auditing organizations, or AOs. These are the third-party organizations that are performing MDSAP audits. The Medical Device Single Audit Program, or MDSAP, acts as a third-party inspection program. The three major participants are the regulatory authorities, the auditing organizations, and the manufacturers. The auditing organizations conduct annual audits of participating manufacturers and certify these manufacturers. In turn, the AOs are assessed and recognized by the RAs, enabling them to conduct the audits on behalf of MDSAP. The AOs then share the audit documentation and outcomes with the RAs. The RAs receiving this information will then make requests to manufacturers for additional information. The facilities will provide this additional information to the RAs. All this gathered information is then used by the RAs to make regulatory decisions. Participation in the program starts with education. There are 10 CDRH Learn MDSAP modules on the web. You can look under post-market activities, inspections, and global harmonization. The program is recognized and maintained by the 5 regulatory authorities. They are Australia's TGA, Brazil's Anvisa, Canada's Health Canada, Japan's Ministry of Health, Labor and Welfare, and the Japanese Pharmaceuticals and Medical Devices Agency, and the U.S. Food and Drug Administration. There are 3 official observers: the European Union, the United Kingdom's MHRA, and the World Health Organization, who uses the program during pre-qualification of in vitro diagnostics. Additionally, there are 3 affiliate members: the Republic of Korea's Ministry of Food and Drug Safety, Argentina's ANMAT, and the newest affiliate member is Singapore's Health Sciences Authority. Also, currently there are 15 AOs. The affiliate membership program began in June 2019. This program allows for regulatory authorities to become members of the MDSAP program. However, the requirements of the affiliates are not included in the audit approach. The affiliate members benefit through a capacity-building relationship which includes training, information exchange, and assisting with the development of regulatory frameworks. There are obligations that the affiliate members need to meet. As part of the commitment to the MDSAP, affiliate members need to provide annual reports to RAs, which include a discussion of how the member uses the MDSAP program and the audit reports. The affiliate members are Argentina's National Administration of Drugs, Foods, and Medical Devices, or ANMAT, the Republic of Korea's Ministry of Food, Drug, and Safety, and just recently added is Singapore's Health Sciences Authority. It's important to note that affiliate members may receive audit reports and certificates only if your facility submits these documents to the affiliate members. Affiliate members do not have access to the regulatory platform maintained by the RAs. The program has largely remained the same throughout the past year. However, there are some areas to highlight. Here's a quick snapshot of the number of manufacturer locations that participate in MDSAP. We currently have approximately 5,800 manufacturer locations that participate in the program worldwide. From 2018 to 2020, you can see that there has been steady growth in the program. Due to travel restrictions caused by the pandemic, we have seen a slowdown in growth. We expect these numbers to continue to grow after restrictions throughout the world are lifted and areas begin to open back up. There are currently 15 AOs that are either authorized or recognized to conduct MDSAP audits. The 4 AOs on the left have submitted their application to the consortium, and RAs have conducted both the Stage 1 and Stage 2 audits. These AOs have met the preliminary requirements to be authorized. The first 3 MDSAP audits performed by these AOs are witnessed by the RAs. In the middle are the 11 fully recognized AOs. To be fully recognized, the AO must have had 3 witness audits and have adequately addressed all nonconformances. Once recognized, they will be subject to head office assessments and annual witness audits. Additionally, there is currently 1 application pending, which is DNV GL Presafe out of Hovik, Norway. The complete list of auditing organizations is located on the MDSAP website, and the link will be provided at the end of the presentation. This brings us to our first knowledge check question. The question is, I am marketing and distributing medical devices in an MDSAP affiliate member's jurisdiction. The affiliate member can obtain MDSAP audit reports and/or MDSAP certificates through the MDSAP database. True or false? If you answered false, that is correct. This is stated in the MDSAP Affiliate Members Roles and Responsibilities Policy Section 4.4 MDSAP Affiliate Members Document. What it says is that MDSAP affiliate members will not have access to MDSAP database on the Regulatory Exchange Platform REP's secure IT portal. In order for an affiliate member to have access to your audit report or certificate, the manufacturer itself would have to submit those documents to the affiliate member. So with all this data coming in, how does the FDA utilize MDSAP in its day-to-day operations? One thing that hasn't changed since day one, the FDA will still accept MDSAP audit reports for FDA routine inspections. What this means is that MDSAP participating manufacturers are exempt from FDA routine inspections. However, there are inspection levels and situations listed in the Compliance Program Guidance Manual, CPGM 7382.845 titled Inspection of Medical Device Manufacturers, those still apply. Here we take a look at the other FDA quality system inspections that are exempt and non-exempt if you participate in MDSAP. So for example, in the green box, routine fall under inspection levels 1 and 2. These fall into the exempt category. The other levels, 3, special, pre, and post-market, are not exempt. These are your compliance follow-up, for cause, risk-based work plan, and PMA approval inspections. These levels, 3, special, and PMA, you are not exempt. So an FDA investigator may call to pre-announce an inspection for these types of inspections. The types of FDA inspections and types of MDSAP audits are shown on this table. As noted previously, MDSAP participants are not exempted from risk-based inspections. Additionally, the MDSAP audit approach utilizes a 3-year cycle. This cycle is demonstrated on the right-hand side of the slide. The cycle starts with an initial audit followed by a certification decision. This now starts your 3-year cycle. Year 1 and 2 contain surveillance audits, with year 3 being a recertification audit followed by another certification decision. The process just continues each 3 years. MDSAP audits and FDA inspections share many similarities. In the table below, I'll compare and contrast these. For example, ISO 1345 plus additional regulatory requirements are used for MDSAP audits. For FDA inspections, they're largely governed by 21 CFR 820, 803, 806, and 807. MDSAP is a voluntary program in the U.S. However, FDA inspections are mandatory in the U.S. As mentioned earlier, FDA will accept MDSAP audit reports in lieu of FDA routine inspections. For MDSAP, a third-party auditor from one of the AOs is used, while FDA inspections are conducted by FDA investigators. MDSAP audits are conducted based on the MDSAP audit approach, while FDA inspections follow the Quality System Inspection Technique, often referred to as QSIT. As described earlier, MDSAP audits have a FDA routine inspection equivalents for their initial, surveillance, and recertification audits. For FDA inspections, additional inspection types exist, such as baseline, abbreviated, for cause, and risk-based work plan. MDSAP audits produce audit reports and NGE forms, while FDA inspections produce EIRs, FDA 483s, and exhibits. The MDSAP audit report is similar to the EIR, and the NGE form is similar to the FDA 483. A single MDSAP audit satisfies the requirements of multiple regulatory authorities, while an FDA inspection only satisfies the requirements of one, the FDA. Here's our second and last knowledge check. The statement is, I am an MDSAP participating manufacturer, therefore I am exempt from FDA inspections. True or false? If you answered false, you are correct. Based on Compliance Program Guidance Manual, CPGM 7382.845, inspection of medical device manufacturers, both risk-based work plan and for-cause inspections are considered special inspection levels. These inspection levels you are not exempt from. As we discussed earlier, you are only exempt from FDA routine inspections. The pandemic has limited travel and impacted the ability to conduct in-person audits. As a response, MDSAP released transmittals associated to extraordinary measures. MDSAP Transmittal 2020-10 was released on 12/31/2020. This transmittal supersedes MDSAP Transmittal numbers 2020-07 and focuses on the extension and expansion of temporary extraordinary measures related to MDSAP audits during quarantine orders and travel restrictions. This transmittal also describes the interim measure to address challenges. Remote audits are defined and processes identified to select the correct alternative audit type. Alternative audit arrangements can occur only when travel restrictions and social and physical distancing are in place as a result of the pandemic. A risk-based process is used to determine the type of alternative audit allowed. Evaluation of audit history, device types, and complexity of the manufacturing drives the types of alternative audit types allowed under this MDSAP transmittal. The requirements to audit are still present, and AOs are expected to suspend and withdraw certifications if they are not able to certify the manufacturer utilizing Transmittal 2020-10. For such situations, regulatory authorities would have to exercise its judgment on the device licensing and marketing authorizations based on the impact to public health. There are 4 types of alternative audits addressed in this transmittal. First, the desktop audit. This is an audit where they review documentation remotely. Second is the remote audit. This is an audit performed off-site using Information and Communication Technology, or ICT. Third is a hybrid audit. This is an audit that's partially off-site using ICT, and then having at least one MDSAP qualified auditor on ground for some portion of the audit. And finally, there's surrogate audits. And this is where the audit is conducted using ICT, and you may not be able to have an MDSAP qualified auditor on site, but another auditor is on site and is able to provide information back to the MDSAP qualified auditor through ICT methods. Throughout the year, AOs have been performing remote audits of manufacturers. Additionally, RAs have been performing remote witness audits and head office assessments of AOs, though it has not gone without any challenges. Technology, for example, has been a challenge, coordinating the different ICT systems between AOs and manufacturers, as well as RAs. There's video and audio quality issues sometimes, and the occasional connectivity issue. Being an international program, many times RAs and AOs are in different parts of the country and or world. Travel restrictions have posed problems, making it hard to plan both hybrid and surrogate audits. With facilities closed, availability of resources have caused problems. And the uncertainty of the pandemic has made it difficult to schedule and plan out audits by both the AOs and the RAs. In summary, MDSAP allows a single regulatory audit that satisfies the requirements of multiple regulatory jurisdictions. Audits are conducted by third-party auditing organizations. These auditing organizations are assessed and recognized by the RAs. Results from MDSAP audits are factored into compliance activities. In June of 2019, the affiliate membership program launched, and there are currently 3 affiliate RAs. Finally, a transmittal was issued to extend and expand extraordinary measures implemented due to travel restrictions. This slide contains a list of resources that were cited throughout the presentation. And now I'm open for questions.
Thank you, Jake, for that great presentation. And we have questions rolling in. Please continue to put the questions in the Q&A box that you see and mark them at the speaker's name. So it would be at Jake. So our first question we'll get to is, my client has scheduled an MDSAP audit with their notified body for the end of this year. Is there a way to notify FDA that they have volunteered for MDSAP and not to get a standard or routine FDA QSIT audit before then?
So that's a great question. So the notification process comes from the manufacturer to the AO. So the AO provides us a list. Provides all of the RAs a list of manufacturers that are participating in the program, and then that list is checked by the FDA prior to any inspections. So all of the communication occurs from the AO to the RAs. So that's how all the LRAs are notified based on the AOs. There's nothing for the manufacturer to do.
Okay, great answer. And I liked how you noted the AO versus a notified body. So you're looking at the AO as it relates to MDSAP. So with that, I'm going to go to our next question. Can an MDSAP certificate be used for U.S. marketing?
So the FDA does not utilize the MDSAP certificate in lieu of an FDA inspection or for marketing. However, the inspection and the associated documents, the audit report, and the NGE form, if needed, are utilized in lieu of routine FDA inspections. So it's not a direct marketing authorization, but it does play a role. But the certificate doesn't play the role. The actual MDSAP audit is what is utilized by the FDA.
Okay, and that kind of builds into this next question, but just for further clarity, does the FDA regard an MDSAP audit as an inspection?
So the MDSAP audits, we do regard them as an inspection for routine inspections. So as I covered up previously, for routine pre-market and post-market audits are still going to be conducted by an FDA representative.
Okay, great. Next question. Are there any special guidance for those companies that design their products but subcontract out everything from manufacturing to sales to logistics to customer relationship management? So it sounds like they just do the design work and then they're subcontracting out everything else. Do you have anything from a special guidance standpoint that you can offer those folks?
So for those that subcontract out much of the work, this is covered under the supplier controls. So that still holds true. So the specification developer in this case would still have the onus to ensure that they have control over the suppliers. In many cases, depending on how much of the subcontracting is out, the subcontractors will also have to undergo an MDSAP audit as part of the MDSAP certification process. So you just have to make sure that the controls are in place for your suppliers, and that includes anybody that you would subcontract out to as well.
Okay, perfect. They're still underneath of their own purchasing controls, regardless of if it's a material or a component or a service. So next question is, what is the relationship between MDSAP and MDSRP? Is the Medical Device Single Review Program, MDSRP, already active?
So I can't speak too much to the MDSRP program. However, I can talk about the interconnection between the two programs. There's not a direct connection between the two. However, the IMDRF is utilizing MDSAP, and they're leveraging lessons learned and the processes that we've put in place as for the MDSAP in developing the single review program. So they're using the work that's already been done for MDSAP to leverage into the single review program as well.
Okay, that's good to know. And for those, IMDRF is International Medical Device Regulators Program. So IMDRF, International Medical Device Regulators Forum. So when you hear that acronym, as well as many of the other acronyms, that's what that stands for. So what is the difference between the authorized versus the recognized AOs?
So for recognized and authorized AOs, both are able to conduct MDSAP audits on behalf of the program. The difference really comes with how long they've been performing those duties, and so to move from a authorized to a recognized would require that 3 successful witness audits are performed by the MDSAP consortium, and that all outstanding and unresolved issues have been resolved. So the key is they've completed all the witness audits by the consortium, and then all of the findings or any issues that were noted have to be resolved before they can be transitioned to recognized. But both authorized and recognized are able to conduct audits on behalf of MDSAP.
Good information to know when looking for AOs. And then now that we've talked about the audits themselves, this is more of a how question, how do you submit MDSAP audit reports to FDA if you want them to be used in place of the routine surveillance inspections?
So this is a good question, and the submission is different than a normal FDA inspection. So this goes back to the previous question that we discussed with notifying the FDA. All of the information comes through the AO. So there's nothing that the manufacturer needs to do in this situation. The audit reports and associated documents are submitted by the AO to the RAs, and that's how the information gets that gets provided to the FDA in this case. So there's no action that the manufacturer needs to take. All of the onus is on the AOs in this situation.
Okay, so just like the first question, the AO is going to handle those communications with the agency. Okay. Next question has to do with findings from the audit reports themselves. What kind of actions would FDA take if major observations were identified during an MDSAP audit?
Just like an FDA inspection, the list of compliance actions that are possible largely depend on the severity of the observations, as well as where they occur. Is it a domestic firm or a foreign firm? There's different actions that are able to be held, such as import alerts, as opposed to compliance meetings or follow-up inspections. So the entire gamut is available. However, it's really a case-by-case situation. So that they're very similar in how they're utilized. However, it's a case-by-case situation that's used to really factor into a much larger compliance decision for each firm by firm.
Okay, and to build further off of that, has FDA executed any administrative, any enforcement actions, such as untitled letters or warning letters, as a result of an MDSAP inspection report?
Currently, to date, that has not occurred. So we have taken follow-up actions and compliance actions to include compliance meetings and then follow-up inspections into the field. To date, there has not been warning letters issued directly as a result of a MDSAP audit by itself.
And then our next question, and this is something that we've seen come up recently in the last year and a half. What is the agency's position on remote unannounced audits? So actually we could break that down first about the remote and then about the unannounced piece. So if you don't mind answering that, Jake.
Yeah, not at all. Yeah, so I will break them up because I think they're two separate items. So for remote audits, the program has utilized remote audits as I discussed in the presentation this past year, and we've kind of refined guidance through a few transmittals that have allowed us to leverage remote audits in different ways. And there's certain things that can be audited remotely. However, not everything can, and not everything can be audited sufficiently remotely. So oftentimes, where possible, the things that need to be on site have kind of pushed off to a separate audit. But then we've leveraged both surrogate audits where sometimes due to travel restrictions this past year, we haven't been able to get maybe a MDSAP qualified auditor on site, but the AO has been able to get a qualified auditor, just not MDSAP qualified auditor on site. So they've utilized that situation where then they've used a computer technology equipment to actually be able to conduct the audit in somewhat of a remote fashion. They've also done some remote audits where the MDSAP certified auditors is away, and the entire audit is conducted remotely. We've had pretty great success, both the feedback from the AOs, the RAs, and the manufacturers. It's been a fairly successful program. So we're looking at how do we leverage that going forward as travel restrictions are lifted. And then I think for the second one, if Joe, if you could just rephrase the question just to make sure I'm answering it completely.
So they had them combined, but like you said, I think you split these two out. So what is the agency's position on unannounced audits? So I guess now we're talking about an unannounced audit.
So currently, the FDA provides a 5-day notice for pre-announcements of FDA audits and routine audits. The exceptions are for cause and compliance follow-ups can be unannounced. With MDSAP, and I'd like to kind of take it back to the actual program. All of our audits are announced, and they're all coordinated from the AO to the manufacturers. So they are predictable. They're routine audits. So all of those audits are announced and worked out between the manufacturer and the AO. So there's a little bit difference there. For FDA, routine audits are announced. The for cause and compliance follow-ups are not necessarily need to be, but for MDSAP, they are all announced audits.
Nope, and that's a great answer. So hopefully now you understand the aspect of remote auditing, and now you also understand that if it's an MDSAP audit, it's not going to be unannounced. However, from an FDA standpoint, we may have unannounced inspections due to for cause or as compliance follow-ups. So that is a great bit of information for everybody to know. So our next question that we got in. So Jake, when can we expect an update to the MDSAP companion document?
So that is a great question. The companion document has been combined and has gone away now with the audit approach. So the new audit approach documents have rolled in the audit methods in the past, as well as the companion document, into a single document that was updated, I believe, within the last year. So the new document actually takes the place of the companion document. So that companion document we would not see to be updated because it's been incorporated into a larger document now.
Great. And next question now spins it back to what MDSAP can and cannot be used for. So the question has to do with, can MDSAP count towards a pre-market approval pre-inspection? So for those who don't know, for class 3 devices that go through pre-market approval, FDA does a pre-inspection as part of the quality system module of that PMA. So the question is, we've already talked about routine surveillance inspections in MDSAP. Now the question is, can you use MDSAP for one of these PMA pre-approval inspections?
So currently, the MDSAP audits are not do not count or not used for equivalence for pre-market and post-market, so PMA inspections for FDA. However, we have due to some of the travel restrictions during this last year with the pandemic, there has been situations where we have adjusted the need for a firm to submit a PMA or post-market. So we've leveraged MDSAP remote audits in our risk assessments along with prior regulatory history to waive or delay these pre or post-market approval inspections. It's something that we're also exploring to expand the use of MDSAP audits after the public health emergency is over. So it's something that we're looking at, but currently it's not a one-for-one like the routine inspections, but it is something that we as an agency and an RA are looking at. It's something that we may use in the future.
Good to know, something to look at and keep track of as the MDSAP program continues to mature and continue on its track. The next question has to do going back to the notified body piece. So the question has to do, does an authorized auditing organization who is also a notified body, and it actually gives some names on some different ones who have both, can they audit both for their ISO 13485 certification as well as their MDSAP certification at the same time?
So this is really a great question, and I think this goes to the nice benefit of MDSAP. So yes, the idea is MDSAP is designed to be less burdensome on the manufacturers. So it's one inspection or audit that's counting for multiple things. So it's counting for 5 RA jurisdictions, plus it's starting to be leveraged more and more for affiliate members. So where they're using the results of the audit for their own regulatory frameworks, and for 1345 at the same time. So this is it's covering all these bases at one time from one single audit.
Great. And in lieu of time, we're going to have one more question. I appreciate all the questions that we received, and we got to as many as we could get to, but there are quite a few that are coming in. But this last one has to do with some of the future aspects of the program. Are there any more international regulatory authorities that will either join the MDSAP program as participants, observers, or potential affiliates?
So currently, there are no additional RAs that are expected to join at this time, and we don't intend to add any additional regulatory requirements as RA participation grows. However, as I presented earlier, just a few months ago, Singapore joined and is an affiliate member. So I think the short answer is we do expect more affiliate members and potentially RAs to join. However, there's no plans at this time.
That's great to know. And like you noted, that was something that was additionally discussed with regards to the presentation. So it's always great to be able to go back and look at the presentation as well with information that's located in there. With that, I'm going to turn it over to you for the last minute to give your final thoughts.
Yeah, thank you very much. So I think with MDSAP, it really presents the opportunity for your facility or your firm to leverage a single regulatory audit which satisfies the requirements of 5 regulatory authorities. So it's least burdensome to the facilities. The use of the program has continued to expand outside the 5 RAs as affiliate members have started to utilize MDSAP for their regulatory purposes. So it provides a one audit that satisfies many requirements. So I really appreciate the time and all the questions, and I will turn it back over to you, Joe.
Thank you, Jake. That was a great both presentation and Q&A session on the Medical Device Single Audit Program. For those that are interested, please go to fda.gov if you're not already part of the program and look it up if you're interested in it. It is a great program that is being utilized by a lot of different manufacturers out there to make things least burdensome. So there's a lot of work involved in it, but it also has a lot of benefit payoff to it. So again, thank you, Jake. So with that, we are going to transition to our next talk of the day. We're going to talk about the MAUDE Database. And to present on that is Fariba Maraka, and she is a Consumer Safety Officer for the Medical Device Reporting Team in a division of Regulatory Programs DRP3 in ORP. Again, part of that super office of OPAC. Her primary role in the position includes supporting manufacturers through EMDR, electronic medical device reporting, and everybody that's a manufacturer should have their EMDR account set up in the gateway. I know that's something that we talked about at REdI a few years ago, but at this stage in the game, you guys should have that set up. She's a primary help desk manager there and provides manufacturers with guidance and support for that account creation and report submissions and troubleshooting. Yeah, medical device reporting is a very important topic, as is that public MAUDE database. Fariba's expertise has supported many efforts to improve post-market surveillance at the center. Remember, we don't just have these reports for the heck of it. We take these reports in and utilize them to look at the post-market world of things. And she earned a master's degree in health systems management at George Mason University in 2006. And Health Service Administration in the Iran University in 1997. And Fariba comes to us with a nursing background. So with that, I will turn it over to Fariba to talk about the MAUDE database.
Hello, my name is Fariba Maramka, and I am a Consumer Safety Officer for the Division of Medical Device Reporting. In this presentation, I will provide an overview of public MAUDE database and how to search for medical device reports. You might wonder why we need to learn about MAUDE. So I'd like to highlight the value of MAUDE database, which stands for Manufacturer and User Facility Device Experience. Each year, the FDA receives several 100,000 medical device reports, MDRs, of suspected device-associated deaths, serious injuries, and malfunctions. These reports are stored in MAUDE. The FDA uses MDRs to monitor device performance, detect potential device-related safety issues, and contribute to benefit-risk assessments of these products. And consumers can be empowered to make educated decisions to improve their healthcare. And industry may use this information to improve the design and safety of their products over time. So MAUDE is a valuable database for anyone interested in exploring questions about medical device safety. After this presentation, you will learn how to use MAUDE database to retrieve MDRs. Our learning objectives are as follows. First, we define the MAUDE, which stands for Manufacturer and User Facility Device Experience. We will then identify two different choices for searching medical device reports, MDRs, in MAUDE. Afterwards, we will demonstrate how you can independently search for MDRs. Let's start with what MAUDE database is and what type of information it houses. The MAUDE database is a publicly searchable database of adverse event medical reports, MDRs. It houses reports submitted to the FDA by mandatory reporters, including manufacturers, device user facilities, and importers, and voluntary reporters such as healthcare professionals, patients, and consumers. These reports describe medical device-associated deaths, serious injuries, and malfunctions. Although MDRs are a valuable source of information, this passive surveillance system has limitations, including the potential submission of incomplete, inaccurate, untimely, unverified, or biased data. In addition, the incidence or prevalence of an event cannot be determined from this reporting system alone due to underreporting of events, inaccuracies in reports, lack of verification that the device caused the reported event, and lack of information about frequency of device use. Because of these, MDRs comprise only one of the FDA's several important post-market surveillance data sources. Here you'll find the screenshot of the database. The data publicly available through MAUDE includes reports received by FDA over the last 10 years. For reports more than 10 years old, click on Download Files. The data files are in zipped files in a table format for purposes of downloading. For these files to be useful to you, you'll first have to download them, unzip them, and then import them into a database or word processor for your further processing. MAUDE is updated at the end of each month. The search page reflects the date of the most recent update. The redacted version of the data received by FDA from the prior month is included in the database. Every Friday, existing reports are refreshed when applicable with redacted supplemental information received. Supplemental information is received from manufacturers for reports for which corrected information is provided, or for which additional information for an event is obtained that was not available at the time the initial report was submitted. When reading a report, you may notice certain words are missing. Please note that certain types of report information are protected from public disclosure under the Freedom of Information Act, FOIA. If a report contains trade secret or confidential business information, such information is replaced by B4. If a report contains personnel or medical files information, such information is replaced by B6. The designations B4 and B6 refer to the exemptions in the FOIA. For example, B4 may be found in place of the product's composition, or B6 may be found in place of a patient's age. Here you'll see two examples of redacted reports. In example number 1, the reporter's address is protected. In example number 2, the date of patient hospitalization is protected. The MAUDE database is organized primarily by reports, where report ID is the key value that identifies a specific report. If you want to view the data by manufacturer, we recommend that you download the MAUDE data files and sort by the first portion of the report number, which contains the manufacturer's registration number from the FDA registration and listing database. This method will ignore spelling mistakes in manufacturer names from the D3 field of the report. The FDA makes medical device reports available to patients and healthcare providers on the FDA's public MAUDE database as one source of information to help them make more informed medical decisions. However, there are certain cases in which such information was not included in MAUDE, like older reports received through CDRH's Legacy Device Experience Network, then the reporting system from 1984 to 1996. And reports received under the Alternative Summary Reporting Program from 1999 to 2019. They are not available in MAUDE. They are available in download data files. Okay, let's take a moment to check our knowledge by answering this question. What kind of reports does MAUDE database house? Is it MDRs or exemptions and variances or FDA guidance documents? If you chose A, you're correct. MAUDE houses medical device reports. Now let's discuss the two available searching methods. There are two choices for searching: the simple search and the advanced search. The simple search searches for terms or keywords entered in the search box in database, versus advanced search searches for a specific criteria, such as brand name, manufacturer, product code, etc. So you can build a more complex search. Keep in mind that searches only retrieve records that contain the search terms provided by the requester. And remember, reports contain as much information as is reasonably available to the individual filing the report. This means certain details, such as model number, may not always be available. However, careful compilation and review of multiple reports on a specific device type may help you better understand common issues people experience with that product. Here you'll find a screenshot of the simple search. And here you'll find a screenshot of the advanced search. When you first open the database, it's already open to the advanced search. As I mentioned earlier, an advanced search allows you to select specific criteria such as manufacturer, model number, brand name, product code, date report received by FDA, so you can build a more complex search. You can also search for a specific report number. Please note, when searching for a specific report number, clear the date report received by FDA box. Of course, you don't need to enter information in all of the fields to complete a search. Simply enter your known search criteria to obtain your result. This search engine caps the number of results per search to 500. If you need large data sets, you should use download files features. These files contain adverse event reports for each reporting year as far back as 1991. To conduct a simple search, click on Go to Simple Search. Now that you have a general understanding of the database and its two different searching methods, let's do an example search. First, we'll perform a simple search. Open MAUDE Database, enter a single word or an exact phrase in quotes. You can search a single word or multiple words separated by AND. Select the year you would like to search in the date report received by FDA drop-down menu. I have decided to search for reports related to electromechanical received in 2016. And I click on the search button. For this case scenario, the total number of reports retrieved based on our search is 33 reports for electromechanical received in 2016. Report results will be listed by date, with the most recently received reports appearing first, followed by older reports. You can print, bookmark, and share or email the result using the functions from the top right corner. You also have an option to export the data to an Excel sheet. Excel contains a subset of fields such as report number, manufacturer, PMA or 510K number, exemption number, and number of events. However, the data zip files contain more columns than the export. The Help button will take you to a list of frequently asked questions. When I click one of the results, it takes me to a page which provides more details. Here I can see the model number and review the specific device problem, event date, event type, and narratives. Please note that when an MDR has multiple versions, initial and follow-ups, there may be more than one of each narrative. The website orders them in reverse chronological order, where the newest narratives are at the top and the oldest ones are at the bottom. Now we'll perform an advanced search example. Open the MAUDE database, Select Go to Advanced Search. Here I've decided to search for reports submitted by the manufacturer, Baxter, associated with the product code FPA, which is for intravascular administrative sets. Product codes may be found by searching the product classification database. If you don't know the product code for a device, you may use a different search criteria, such as the brand name or model number. I have narrowed the search further by setting the dates to go from January 1st of 2020 to the end of December of 2020. If I don't change these dates, the database defaults to providing results from the previous month. Click on Search. Here I received a series of results spanning multiple pages. The search engine caps the number of results per search to 500. You'll notice that my search terms yielded more than 500 reports, and the database is letting me know that the results displayed are incomplete and that I may need to narrow my search further. In this case, one possible workaround is to narrow the date range. But if I must collect all the information, I can use the Download Files function instead, as I mentioned earlier, to access reports more than 10 years old. You'll also notice that the report results will be listed by date, with the most recently received reports appearing first, followed by older reports. When I click one of the results, it takes me to a page which provides more detail. Here I can see the catalog number and review the specific device problem, patient problem, event date and event type, narratives, manufacturer narrative, event description. Remember, reporters are to provide all the information that they are aware of. This means certain details such as model numbers or brand names may not always be available. However, careful compilation and review of multiple reports on a specific device type may help you better understand common issues people experience with that device. Now is time for our second knowledge check question. What is the download files used for? To get more details on reports, to locate missing data from MDRs, to access reports more than 10 years old. If your answer is C, you're correct. The download files is used to access reports more than 10 years old. In summary, public MAUDE houses medical device adverse event reports that FDA uses to monitor device performance and safety and inform its regulatory decision making. It's a searchable database available to both the agency and the public. In order to retrieve data from MAUDE Database, there are two searching choices available: Simple Search and Advanced Search. A simple search searches for all terms or keywords entered in the search box. An advanced search allows you to use a variety of criteria in combination to filter and find the right results. We hope you found this information useful. The following slides contain links to additional resources you may find useful. If you have MAUDE questions, please contact the Division of Industry and Consumer Education. We also encourage you to refer to CDRH Learn, which contain additional educational materials that are comprehensive, interactive, and easily accessible. Modules are provided in various formats, including videos, audio recordings, and slide presentations. We hope you continue to educate yourself on these MDR-related activities. Thank you for watching. Thank you for joining me for this presentation. Let's now take some of your questions.
Thank you, Fariba, for that great presentation. And we do have quite a few questions that have come in, so let's get started on them. The first question is, I have tried searching MAUDE, the MAUDE database, and it comes back with no records found.
Thank you for the question. If you are doing a simple search, shorten string you are looking for. For best results, enter only a portion of the word you are looking for example, BIO. But if you are doing an advanced search, try different combination of your search criteria to get the best result. But always keep in mind, enter only the information that you are sure applies to what you want to find. But if you are not sure about the dates or a value, we recommend do not fill it in.
Okay, great answer. And the next question actually builds off of that a little bit. When searching using a date form field, how do I get all records before or even after a particular date?
If you place the date only in the first field date and leave the second date field, then you will receive records after the given date. And similarly, if you put that particular date in the second date field and leave the first date field blank, you will only receive records before the given date.
Okay. And speaking of dates, we have a question to get some clarification. Can you please clarify what was meant when you stated that some records from 1999 to 2019 aren't available on MAUDE? Should a company search both the MAUDE database and the downloaded files to ensure that all applicable medical device report events are identified?
The current MAUDE has data only for the last 10 years. So for older than that, you must utilize only download data files.
Okay, that is good to know for anyone who's searching through MAUDE, how far it goes back and how you can get that older data. So the next question has to do with how FDA utilizes that data that's collected. So the question goes, how does data collected by MAUDE feed into risk assessments and are decisions by FDA to either begin an investigation into a device or to contact the manufacturer. Is this trended-based? How is this risk-based? So the question has to do with how do we use the information in MAUDE?
Oh, that's a great question. When you receive, for your report, you see in MAUDE or submitted to FDA, you have to file actually your MDR, your firm's report. If your investigation leads you, that report is required actually. So all the comments, all the investigation results you provided will be available to FDA analysts. And these are really important information, definitely will feed into risk assessment and help them with better making regulatory decision.
Okay. And the next question talks about that mandatory aspect in 21 CFR 803. Is medical device reporting mandatory? And should every adverse event be reported or only the ones that cause injury? So this is more of an 803 question than a MAUDE question. But if you don't mind answering this as it relates to what is a reportable event?
That's correct, Joe. It's more 803 related questions. Well, as far as I know, for any firms that have registered with FDA or any device manufacturers that have registered with FDA, they should have a compliance system in place. And definition for all MDR reports are either malfunction, deaths, or serious injury, and you can find definition in MDR regulations. But for their assistance, I believe you can contact MDR policy response line if you have any questions regarding regulation or reporting requirements.
Sure. And there's a medical device reporting team that handles the general medical device reporting questions. You can also feel free for this base information to reach out to DICE. So part of that also, as Fariba mentioned, there's a quality system regulation requirement in 820.198 on complaint handling and whether or not a complaint is MDR reportable. All MDRs are complaints. Not all complaints are MDRs. If, as Fariba noted, that you're dealing with a malfunction or a serious injury or death, or a malfunction that could cause a serious injury or death, then that becomes reportable underneath of 21 CFR 803. But feel free to reach out to DICE with this question. Also feel free to reach out to the MDR team as well. And there's also some great information on CDRH Learn that talks about the medical device reporting requirements. So let's get back to some of the MAUDE questions. So with regards to MAUDE, is there a difference between the MAUDE database and the MedSun database? So now the question has to do with the two different sets of database. So if you wouldn't mind answering, what is the difference between MAUDE database and the MedSun database?
They're completely different things. I can say that MAUDE is just a database housing all reports and medical device adverse events submitted to FDA, including MedSun reports. MedSun is a voluntary program for a set of hospitals who voluntarily reporting to FDAs, and their report is considered as a user facility report. They are also stored in MAUDE database, but under FOIA Act, the report number would be redacted. But yes, they are in MAUDE database. So completely different, I can say. MedSign is a program, a voluntary reporting program from specific hospitals, but MAUDE is a database internal, or for the public, I can say, for the public view, database of all kind of MDRs we are receiving, including MedSign reports.
Okay, good to know about the separate programs. Next question, again, has to do with MAUDE. To search product codes in the product code classification database, there are multiple product codes for the same or similar device. What are the differences among those product codes, and are they manufacturer specific within MAUDE? So you have product code classifications, and then how are those utilized in MAUDE for same or similar devices?
That's again, I believe, more 803 related questions, but each report has, each device in each report has a product code, and it depends on the device classification. If it is 510K or PMA, they may have even more than one product code, but that is one of the search criteria I can say for sure you can utilize to retrieve the reports you are looking for.
Good answer. And going off of code, so understand, this is for the audience to understand, we use this term codes for a couple different things. So make sure you know what code that you're talking about. Are you talking about product code, or the next question that came up, event codes? So the next question here is, why do some of the medical device reports have more than one event description or event code or manufacturer narrative? So now we were talking about product code, now we're talking about event codes. So why do some of the medical device reports have more than one event description or more than one event code?
That's a great question. They may have been, I mean, the more than event description, if we are talking about, when an MDR has multiple versions, initial and follow-ups, there may be more than one of each narrative. And website displays them in reverse chronological order, where the newest narratives are at the top and the oldest ones are at the bottom. But if you look at the text data in the data files, you can use the text key to tell the order in which the narratives were entered.
Okay. And then our next question has to do with the database itself. So when doing searches, sometimes when we're looking through MAUDE, we're looking at numbers that go beyond the 500 search, as some products have very high volume reports. Is there a plan to go beyond the 500 search result in the future?
Yeah, that is, the 500 is the max number of search results for from the online search. But so that message tells you that there are more records, but they are not in the results list. I am not aware of any plan to update it, to extend the number of the search results. But for the moment, for now, you can just do, one possible workaround is to narrow the date range, or you can utilize download data files because they have reports more than 10 years old.
Okay, and we have time for one last question. So if I have reported something in the past, can a report be removed from the public MAUDE database if I request it?
Well, the only way to have one of your reports removed from MAUDE is to contact the EMDR help desks at emdr@fda.hhs.gov and explain the situations. In general, we only remove reports that they are duplicate or they have bad report numbers, which prevents a supplement from correcting the error, that report. For most MDR issues, you can submit a correction follow-up to change any errors in your previous reports. But unfortunately, because the report number is used to tie initial and supplemental reports together, you cannot submit a correction follow-up to change the report number. So, like I said, try to contact the EMDR help desk and explain the situation. We will be able best assist you.
Okay, thank you. And with that, I'm going to give it over to you for the next 30 seconds or so to provide your final thoughts.
Well, thank you very much for all the great questions. Well, there are really three things I want you to remember. One, take time to learn about the MAUDE database. And two, identify two choices of searching for MDRs in MAUDE. Three, for practice, conduct a search using both simple and advanced search. I hope you have found this presentation to be helpful. Thank you.
Okay, with that, it ends our morning session on the MAUDE database. Thank you again, Fariba, for a great presentation, followed by a great question and answer session. We're going to take about a 20-minute break until 10:25, but I do want to note one thing before you jump on break. So we talked about this morning the continuing education piece. I do know and can tell you, because there are a lot of questions coming in through the chat about a single email being sent to folks. So to simplify things, I do know that emails did go out this morning from CEDAR. So they did send emails this morning with regards to all the attendees from yesterday, noting the individual days events, and the email contains information both with regards to the CE claim codes as well as the survey links. So I know we said that they were to go out at night, but it's been fairly busy. So I do know and can report that these did go out this morning. If you have any further questions on that, please let us know. Otherwise, please take a nice break and meet us back here at 10:25 to talk about additive manufacturing. Again, we're going to continue with these great topics. Thank you. Welcome back, everyone, and we will continue on with our topics this morning, or again, afternoon or evening, depending on where you're located, with an exciting talk on additive manufacturing of medical devices. Very hot topic these days. Your presenter is going to be Matthew DiPrima. Dr. DiPrima is a research material scientist in our Division of Applied Mechanics in our Office of Science and Engineering Laboratories. He's currently serving as a coordinator for the Additive Manufacturing Program area, as well as chairing the CDRH Additive Manufacturing Working Group and the agency's Advanced Manufacturing Technology Working Group. So Matt has been involved in many research efforts to help improve device testing and the understanding of material processing on device performance. He earned his PhD in material science from the Georgia Institute of Technology in 2009. And he is a good person to be talking about this very hot and very interesting topic. So without further ado, I will turn it over to Matt.
Thank you for that introduction. I'm Dr. Matthew DiPrima. I'm a research materials engineer in the Division of Applied Mechanics in the Office of Science and Engineering Laboratories. And this morning I'll be speaking about additive manufacturing of medical devices. There are 3 learning objectives for this talk. The first is to be able to describe the 7 types of additive manufacturing technologies and their applications for medical devices. The second is to be able to list the types of medical devices made using additive manufacturing. And the third is to understand the contents of the FDA guidance, technical considerations of additively manufactured medical devices. So first, I'm going to discuss some additive manufacturing technologies. So this is a time-lapse or a sped-up video that has been accelerated to 14 times, but this is an example of some of the things that additive manufacturing lets you do. So this technology works by having a pool of liquid that is then being cured with a light source. So we can see here as this platen is raising out of the essentially goop, you have this part forming. And this is incredibly powerful because we can program all sorts of shapes and sizes and just really innovative objects to be made using this technology without any tooling or additional needs for manufacturing equipment. So this allows us to make very innovative products. It lets us make products very quickly, and it allows us to design and then manufacture products very quickly because we don't have to have additional tooling or molds for these parts.
ASTM defines additive manufacturing, commonly called 3D printing, as a process of joining materials to make parts from 3D model data, usually layer upon layer, as opposed to subtractive manufacturing and formative manufacturing methodologies. So the key parts to this is really that for this to be additive manufacturing, it has to be coming from a 3D model data, and then we're generally joining this layer by layer. Now, ASTM defines 7 different AM technologies, which I'm about to talk through, but it's really important to note that there's a whole bunch of different terms, some listed here, that can refer to either a specific AM technology or a specific process that a company has developed, as a subset of that manufacturing technology. So there are lots of terms. I prefer additive manufacturing, but again, 3D printing is the more common vernacular usage. So the first of the AM technologies I'm going to talk about is called powder bed fusion. So this is a process where a layer of powder, roughly about the height of human hair, sometimes about half of that, is going to be deposited across a build platform. An energy source, either laser or electron beam, then traces and then fills in the that 3-dimensional design, which melts the layer, and melts it to the layer below. So again, we're getting our layer by layer process, and this continues until the part or the set of parts is complete. At the end of the process, the part is completely encased in powder, which then has to be removed. This can be used for metals as well as polymers, and in the medical space, we've seen implants as well as cutting guides made using this technology. The second technology is called vat photopolymerization. The two sort of sub-methods of this are called stereolithography, SLA, or digital light projection, DLP. So in this process, a liquid monomer or monomer mix flows into a tank, and then light or a laser traces over that two-dimensional sort of stencil that then causes the material to cure. So it cures in that layer and then cures to the layer below. We've seen this commonly used in a lot of dental applications, and there's been a lot of promise in drug printing as well. The third technology is called material extrusion. This is one of the most common types out there, and this is what most people are familiar with when you think 3D printing. So I like to tell people this is essentially a hot glue gun on an XYZ gantry, but here we're taking a feedstock. It can either be a liquid or it could be solid that is then melted, and we're extruding it through a nozzle to precisely lay down a bead of material. This technology is nice because it's one of the few where you can use multi-materials, so you can print different materials at different colors all at once. It does have a little bit of a worse resolution than some of the other technologies, but again, based on the application, it can be perfectly good. So this is probably the most common bioprinting technology, where you can have media and cells intermixed being injected through a syringe. One of lots of promise in pill printing, where you can take a feedstock and dope it with your active pharmaceutical ingredient. And then in the medical device space, we see lots of prosthetics as well as anatomic models made using this technology. The fourth technology is called binder jetting. So this is a sort of combination of powder bed fusion with sort of kind of material extrusion. So here we put down a layer of powder. We then trace over that, and instead of hitting it with energy, we're spraying essentially a glue. So we're going to be gluing that powder together. In some instances, that glue can be doped with a dye, so you can print with different colors. Or in other cases, this is just going to hold the part together until you can stick it into a furnace or other post-processing system. Because you can do cool things with colors, anatomic models are often made using these technologies, and the first FDA-approved 3D printed pill was made using this technology, where it took an existing powder that was already used in other pills and an approved glue. So we're just sort of mixing two different approved systems or materials together into this printer. The fifth technology is called material jetting. So this works just like your inkjet printer. So we're going to be taking a liquid, running it through essentially an inkjet printer head, and we're going to be spraying drop by drop. These drops are very small, so we can get very high resolution. And much like a printer, an inkjet printer head, it can easily print with multiple materials, again giving us different colors or different properties of the material that you're printing. There's been a lot of work in this space in bioprinting, where you can actually set it up so you are printing a single cell at a time, which can give you amazing spatial resolution as you're putting down your cells. And again, because of the colors as well as the different stiffnesses of materials you can print, it's been used for anatomic models in the medical device space. The sixth technology is called direct energy deposition. So in this technology, we're taking a feedstock, be it a metal a metal wire or metal powder, and we're blowing it in the path of our electron beam or laser. So unlike powder bed fusion, where we have this bed that the beam is tracing over, here the beam and the feedstock move together. This is commonly used in aerospace applications because you can make very large parts. parts. In medical devices, we've seen some limited use of it in orthopedic devices. The seventh and final AM technology is called sheet lamination. So there are a couple of different ways of using this technology, but generally you take a sheet of feedstock, you put it over your build tray, and you either melt or cut the layer to the part you want. You then put another sheet on. This schematic is showing a roller. So in medical devices, we haven't seen this used yet. However, there's a really cool bioprinting approach where you have a layer of cells and you use a laser to actually shoot it off of the sheet. So again, this is something where we see the technology moving, and there are some bioprinting applications for it. Hey, knowledge check 1. Which AM technology has not been used to manufacture medical devices to date? Powder bed fusion, sheet lamination, vat photopolymerization, or material jetting? Take a second to consider which answer you think is correct.
The correct answer is sheet lamination. From those slides, sheet lamination is the AM technology that hasn't been used yet to make medical devices, although it is showing some promise in the bioprinting field. Having overviewed the types of AM technology, let's talk about how they're used in medicine and medical products. So the first application of this process can be seen with instruments and guides, followed by medical implants, and lastly, a subset of implants or other products where we say that they've been personalized or patient matched. So AM brings a couple of different things to medical devices. So the first is complex structures. So within the resolution and sort of residual material removal and support structure limitations, additive manufacturing is generally not affected by part complexity. So you can make really complex parts that would be difficult or impossible in other manufacturing techniques, which allows for interesting things for tissue ingrowth, reduced weight or stress shielding, or also to make it very easy to match medical devices to a patient's body. So I've already mentioned a few of these, but a lot on complex structures and the ability to integrate sort of a complex structure to a solid component, so we no longer necessarily have a distinct layer between like a porous coating and the substrate. You can do some fascinating gradient components. And this will come up later, but roughly 2/3 of the cleared AM devices to date utilize some sort of complex or porous structure. So this is a conceptual demo of this process. So this is an acetabular cup. So this would go into your hip on the pelvis side. And you can see what looks like this random structure, and it's computationally generated to be random. But the manufacturing process is actually very controlled, and it's in making each of these struts. So every hip cup that's made using additive manufacturing is going to have the exact same random architecture. So the next part is personalized medicine. So when we go back to the definition of additive manufacturing, we're talking about taking 3D model data, and usually you can generate this in a CAD or engineering software. But if you pair this process with a 3D imaging technique like CT, MRI, optical scanning, we've even seen some ultrasound, you can now take patient data and then build parts or devices personalized based on that specific anatomy. And we've even seen this go a step further where virtual surgical software is incorporated. So you have the patient's data, a surgeon can go in and say, this is where I'd want to make the cuts on the bone, and then you can create personalized cutting guides, tools, along with an implant that's not only matched to that patient's anatomy, but to the surgery that the surgeon or clinician wants to perform. These are just some examples of patient-matched devices. We have a craniofacial implant on the far left. In the middle, we have what we'll call a skull plate or a cranioplasty plate, and on the right we have a patient-matched knee. Now, the articulating surface, or where these two parts are meeting, that's going to be standard for this device. But instead, what's been matched is the backside of the implant. And the idea here is the surgeons can remove less bone, and the implant will be printed so it's going to perfectly fit the cuts that they were going to make. And again, this is an example where they would also have patient-matched cutting guides to ensure that the device is going to fit perfectly to the patient's knee. So having said that, a very common question we get are what kinds of devices has the FDA cleared? So some of my colleagues and I published a paper back in 2018 that looked at the kinds of devices that were cleared from 2010 to 2016. Real quick, the review methodology went into the FDA database for clearing devices. We looked at a number of terms. We then manually verified the findings from the database call. Then we de-identified and aggregated the data so we could talk about it without releasing any proprietary information. So this is a little bit of an eye chart, but let me explain what we're looking at. So on the outer ring, we are looking at the kind or the type of additive manufacturing used in that cleared submission. So 83% of the submissions were made using powder bed fusion. So we broke that down. About 25% of the types that we saw were made using electron beam melting. 66% of the powder bed fusion was made using a laser, and then there was 9 where we couldn't quite tell if it was laser or e-beam based. So even though vast majority of what we've seen is powder bed fusion, we've also seen products made using stereolithography, again, the subset of that photopolymerization, as well as ink jetting, which is a type of material jetting, and then extrusion is in there as well. So while we've seen a lot of powder bed fusion, we have seen a range of other products. The next ring in is going to be the type of application. So 53% of what we cleared was in the orthopedic space. And I should say these are orthopedic implants because you can see the next category over being surgical guides, making up 34%. Quite a few of those are for orthopedic applications. In terms of the implants, we can see that we cleared the most knees at 35% of the overall orthopedic implants, followed by hips, spine, shoulder, and then there was a range of others, and we weren't going to sort of create bins for every single orthopedic implant. The next ring in is going to be the material. Very kind of surprising for us, but almost half of the cleared products we saw were made out of polymer. Of the metals, a vast majority were made out of either titanium, and most of the titanium ones were the titanium alloy Ti6. We have seen some cobalt chrome, and in the others, there's some stainless steels and other materials as well. And then when we look at the devices for porosity, we can see that roughly 2/3 of the implants, so this doesn't include cutting guides, but just the implants, had some sort of porous structure to them. Knowledge check 2. Between 2010 and 2016, what statement about FDA 510K cleared AM devices is false? Option 1, powder bed fusion was the most commonly used technology. Option 2, knees were the most cleared AM product. Option 3, relatively few AM products were made from polymers. Or 4, 2/3 of implants had a porous structure. Take a second or two to consider which answer you think is correct. The answer is 3. Again, almost 50% of the AM products cleared were made from polymers. Okay. So moving along to the FDA guidance, technical considerations for additive manufactured medical devices. This was issued in final version in December of 2017. So the guidance was roughly broken into 2 parts. We have the design and manufacturing considerations, which addresses the technical considerations needed to address the quality system's requirements. It's very important to note that the quality system requirements for a medical device is determined by existing regulatory classifications or regulations, generally not affected by the parts made using additive manufacturing. And then the second half of the guidance was focused on device testing considerations, which describes what AM-specific information should be included in a pre-market submission. And what's more, the type of pre-market submission is determined by the regulatory classification of the product and generally isn't affected by the manufacturing technique. This is a list of what's in each of the sections. We're going to go into a little bit more detail on a few of them, but we're looking for the device design, especially if product's going to be patient matched, ensuring that the software workflow has been properly validated. Lots of different softwares involved in this process. We want to make sure that everything's being properly controlled. Material controls is critical. The starting material that's being fed into an AM system is not the same material as the final material in the device. So it's very important to understand that process. Lots of post-processing steps. I already touched on the need to make sure that residual powder has been removed from powder bed fusion, or dealing with the uncured material on sterile lithography, lots of heat treatments. Some AM technologies, you have to physically cut the parts off of the build platform. So lots of additional steps that need to be taken care of. And then, of course, you have your process validation and acceptance. On the pre-market side, a lot of what sort of information is needed in terms of the performance testing. I'm going to discuss this a little bit more, but the key thing that we're looking for in a 510K involving additive manufacturing is that we have assessed what the worst case product is going to be. So we want to make sure that we have the worst case factors being considered based on the manufacturing process. Again, lots on material characterization if the material is changing through the course of the process. I touched on this a few times already, but we want to make sure that residual manufacturing materials have been removed. Biocompatibility, which turns out to be pretty comparable to every other medical device out there. And there's some additional labeling considerations for patient-matched products. So let's get into some of the specifics. So device design. So this is the motivation for this is AM technologies have many different design considerations. Some AM technologies require you to have extensive support structures, some don't, some have challenges in removing excess material, some don't. So it's important to understand what the AM technology is to make sure that all the appropriate factors are being considered and mitigated, as well as if it's being patient matched. So what we generally look for is a description of the AM technology as well as the process flow chart, so we can understand what the process is and what the specific mitigations for the device are. And for patient matching, we are looking to understand what the patient matched features are, as well as what the design envelope is for all the products that could be made under that clearance. In terms of material controls, what we're really looking for is understanding how the material reuse and storage and just generally how the AM system is handling that material and if it's going to change product performance. There are certainly ways where reusing powder and powder bed fusion can change the properties of the final product. There are also ways that it might not. So we're just looking for what the sort of mitigation is for material changes as a function of AM use. Then we move on to performance testing. So here we're really trying to understand how the AM process is going to change the performance of that final product. So we generally are looking at orientation in that build platform. Again, we're building things layer by layer, and that layer effect can change the mechanical or dimensional performance. Build location, certainly different AM systems are more sensitive to where that part is being built, and we just want to make sure that's being assessed. And then there's going to be a host of other parameters based on the specific AM technology that could affect product performance. So once more, we're looking for what is the worst case factor in terms of final device performance. We want to make sure that we're properly assessing it. This is often done through sort of looking at the validation testing and using coupons to demonstrate how the different parameters change final performance. And again, we're not looking to sort of clear or approve a manufacturing process. We're just looking to understand and ensure that the worst case device is being assessed in that submission. So I touched on patient matching a few times, so it's very important to note that patient matched devices generally don't fall under the custom device exemption. There's a custom device exemption guidance, and in Section 5E, it specifically discusses patient matched devices and why they generally don't meet the custom device exemption requirements. So we're treating these patient-matched devices sort of to a specified design envelope. Now that will require validation, and in a submission, you're going to have to show substantial equivalence for the worst case or cases of the design based on what the requirements are. But essentially, this is letting us go from a series of discrete sizes in a submission to, again, a design envelope, and will let us assess, ideally, everything inside that design envelope. That being said, the guidance is only addressing patient matching in conjunction with the AM process. There are certainly other patient match considerations not covered by the guidance. Knowledge check 3. What does the 2017 AM technical guidance not cover? Option 1, pre-market considerations for AM devices. Option 2, post-market considerations for AM devices. Option 3, policy on using AM to manufacture devices at hospitals or other points of care. Take a second to consider which answer you think is correct. All right. So, as previously mentioned, the AM guidance was covering the pre-market and post-market considerations, and therefore it's not addressing any technical or policy concerns related to using AM at the point of care.
In summary, we have reviewed the 7 types of additive manufacturing technologies and their applications for medical devices. We've also discussed the many medical devices that use additive manufacturing, and it's, again, worth noting that most of these have been in the orthopedic product space, along with cutting guides. And we've also touched on the FDA guidance that was published in December of 2017 that outlines the design, manufacturing, and device testing considerations for devices made using this technology.
So with that, I'd like to thank you, and I am now ready to respond to any of the submitted questions.
Thank you, Matthew, and we'll get to as many of the questions that we can. We got quite a few of them. The first one to start off is, are 3D printers themselves regulated as medical devices?
Thank you, Joe. 3D printers, as all manufacturing equipment, generally aren't regulated as medical devices. However, there are certainly submissions where the additive manufacturing equipment or 3D printer is going to be treated as an accessory to that submission.
Great. And the next question, I'm actually bundling quite a few of these that came in together because it's talking about the raw materials being used. So do the resins or materials for building these medical devices have any special regulations to follow or comply with themselves? So we talked about the actual printing, but this question has to do with the materials being used.
So first off, materials aren't cleared or approved by the FDA for general medical use. There are certain specific applications like bone void fillers or in the dental space where the material is the medical device, but it's only cleared or approved for its specific intended use. So stepping back to that, the material controls would generally fall under the sponsor's quality systems. So it would be really up to the sponsor to ensure they have the correct material and protocols in place for the material handling. Certainly, there are materials designed for each printing technology as well as each printer type. So you do have to make sure that you're using the right material and the right printer and you're handling it and reusing it appropriately. But that would all fall under the general quality systems regulations.
Okay. And to continue on this question about materials, you noted that they have to be controlled under 820. Does the material used to print devices need to be defined in the 510K? Or is there wiggle room to change a printing material after a 510K clearance, say through a letter to file, as long as the appropriate testing is being conducted as part of the quality system? So now you noted it with regards to 820. What about with regards to 510K and changes to a 510K?
So we have a very good guidance on when you need to submit a new 510K, and there's a number of flowcharts in that guidance. And I would say, for a general response, you'd really need to follow that guidance. There are certainly instances where changing the material would require a new 510K, but really that decision would be made off of existing FDA guidance.
Good to know. And now to follow up on that, we have a couple of different specific material questions. So in your presentation, you noted TI6 and that particular titanium alloy. So the question is, why are so many of the additive manufacturing devices made from TI6A I4V? So the question has to do with, I guess people are seeing this one particular alloy used a lot.
Certainly. It's worth noting that most of the 510Ks we've seen have been for orthopedic applications, and that alloy was already commonly used in non-AM applications. Additionally, that's an alloy that's readily available for AM systems, probably because it's also used in the aerospace and automotive industry. So it's a material that we've already seen a lot and is readily available for AM systems.
Okay, and then we have another one with regards to a different type of material. Do you know if the FDA is considering the approval of additive manufactured stainless steel, specifically UNS 31603 for medical devices?
So, again, we don't clear or approve materials, and we review the material of the device for the intended use of the device. We've certainly cleared AM stainless steel devices. We've seen, the implant grade materials as well as instrument grade materials being used. It's all in the context of what is the product that's going to be using that specific alloy, and does it meet the performance requirements for that product?
Correct. And to build off of that, somebody's asking what kind of biocompatibility concerns should be considered when 3D printing a patient component?
This is a good question, and that was something we were very worried about early on when we were looking at additive manufacturing. But it turns out the standard biocompatibility testing requirements per ISO 10993 have been sufficient to assess the biocompatibility risk. It's always worth noting, though, that you want to perform biocompatibility testing on your final finished product. There are lots of post-processing steps involved in the additive manufacturing process. So it's important to capture all of the sort of additional materials and processes contacting the AM part before you perform biocompatibility testing.
Okay, thank you for that. And I know you mentioned this one during your presentation, but I think it's worth being repeated since we received a couple similar questions on this. So how does custom-made device regulations intersect with additive manufacturing, personalized medicine, and patient-matched devices? Are they the same thing? Are they different? And can you point us in the right direction on this? And I know you did mention the guidance, but I want you to note a little bit more.
So it's really worth noting that the custom device exemption is its very own separate process with lots of requirements and restrictions. And the custom device exemption guidance, we didn't even talk to them, but they put a great section in. It's 5E of that guidance, and it really sort of explains the difference between what custom device is and a patient-matched product that would cover sort of a design envelope. So they're very different, and there's certainly AM devices that fall under the custom device exemption, but they fall under that based on the existing rules for that exemption. And most of what we see that's patient matched would not qualify for the custom device exemption and falls under the sort of patient matched design envelope assessment.
Perfect. And I also will give an additional plug on CDRH Learn. There's a great learning module with regards to custom device exemption that's given by Nathan Ivey, who helped put that guidance together. So if on top of reading the guidance, please take a look at that module. It's a great module that talks about and provides a lot of clarity, because I know there's a lot of confusion, not just with regards to additive manufacturing and custom devices, but custom devices in general. So please take a look at both of those. So now I'm going to switch it up to some process pieces. So one of the questions that came up has to do with regards to changing the actual additive manufacturing process. So for one component, our cleared 510K device was switched from molding to laser 3D printing. Should we inform FDA about this change of how we're doing the manufacturing process? And considering the fact that the manufacturing processes were not included in the clear device, should we provide the 3D printer installation qualification, operational qualification, and performance qualification validations underneath of 820 as part of a new 510K? So quite a few things to unpackage there.
All right, so I'm going to go back to my previous response and say you should really look at the guidance on when to submit a new 510K to make that final determination. There's a lot of complexity in there, and it's never safe for me to answer, the question of should I submit a new 510K. So if you do need to submit a new 510K, certainly the IQ, OQ, PQ data can be useful for us to understand the worst case build parameters. As I mentioned in the discussion, a lot of the technical guidance is really focused on making sure that we understand that you are testing the worst case device, and if not, what the manufacturing variability is going to be and that you've mitigated it. And certainly IQ, OQ, PQ data is one way to do that.
Okay, and that actually builds into this next question. So how do you go about ensuring a worst case device is properly assessed?
All right, so we do that through looking at the sort of build parameters and the sort of process from start to finish. So certainly for a patient matched device, we look at what that design envelope is and determine what the worst case design would be for each of the required performance tests. We then go and look at the AM process, and we look at build variability, orientation variability. We look at what the material reuse protocol is and make sure that that sort of worst case condition is being assessed. And then once we've done all of that, we can make sure that the worst case build and design parameters are going to be the ones undergoing the performance testing for that product.
Okay, perfect. We have about a minute left, so I'm going to turn it over to you, Matthew, to give your last thoughts on this really interesting and great topic.
Thanks so much. So I think it's really worth emphasizing that the adoption of AM into manufacturing medical devices has been highly successful, and I really hope that the industry continues to be responsible in the adoption of AM as well as any new manufacturing technologies as they're developed. And secondly, as horrible as the COVID pandemic was, we saw a tremendous response from people with additive manufacturing capabilities. We've seen the utility of both the distributed manufacturing setup, as well as the really agile way to transition what you're manufacturing using this equipment. And I'm really hoping that we can build on this success and lessons learned from COVID into non-pandemic times.
Thank you. That is a great presentation followed by a great question and answer session. Lots of valuable information. I actually learned quite a few new things just by working with Matthew, seeing the presentation, and even hearing more stuff today. So I know that this is, again, a very hot topic. I know it's been around a few years, but more and more of us are seeing this everywhere. I can even tell you that my son, who's in high school in an engineering program, they're learning how to use these type of manufacturing devices early, and you're seeing younger and younger folks get into this. So definitely a great topic as it relates in general and also specifically to medical devices. So with that, we are now going to go into the home stretch here for the medical devices track and move to our last session of the day on Class 1 and Class 2 recall analysis. So doing this presentation for us is Meredith Andrus. She comes from the Office of Regulatory Affairs, where she serves as a recall coordinator for medical device recalls underneath the compliance branch of OMDRHO. That's the Medical Device Commodities Group in ORA. Currently, she is one of 9 recall coordinators in the medical device program, and the responsibilities include reviewing and recommending new recalls for classification by CDRH. She became a recall coordinator in March of 2015 and has been a frequent speaker of recall-related topics to the industry. She's been with FDA for 11 years, and prior to coming to FDA, served as a couple different public health roles. And she received her master's degree in public health with a specialization in veterinary public health from Ohio State University. So please welcome Meredith and her talk on recalls.
Thank you for the introduction. Today I will be speaking and giving a presentation regarding an analysis that was performed on Class 2 recalls. This is my disclaimer slide, and I will give you a couple seconds to read over this. This slide is just a snapshot to give you an idea of the volume of recalls the Medical Devices Program and ORA receives and processes. Over the past 3 fiscal years, we have averaged between 900 and 1,000 recalls annually, with a total of about 3,000 different products. This is a very large volume compared to other programs. In fact, in fiscal year 20, medical device recalls represented 40% of all reported recalls for FDA. During this presentation, I will provide a basic review of recall classifications, briefly review a previous analysis of Class 1 recalls, discuss our current project in which Class 2 recalls were analyzed, and then we will look at key takeaways and best practices that can be applied from this analysis. In addition, we will discuss some things that you could expect during an FDA follow-up inspection. So here we will begin with a recall review. Before we get started, I wanted to quickly explain recall classifications and then compare them with medical device classifications. Recall classifications listed on the left start with the highest risk being Class 1 and decrease in relation to risk as the classification gets higher, Class 3. Each of these classifications and their definitions are listed on the slide. So looking at Class 1, there's a reasonable probability that the use of or exposure to a violative product will cause serious adverse health consequences or death, in comparison to a Class 3 where the use of or exposure to a violative product is not likely to cause adverse health consequences. In contrast, medical device classifications go in the reverse order, with the highest risk devices listed as Class 3. I just wanted everyone to see this and to keep it in mind so there's no confusion. For more information on basic recalls, you can visit CDRH Learn on FDA's website. Now we will move to looking at the previous Class 1 recall analysis that was conducted. For this project, a work group was created in 2020 to review Class 1 recalls from the time frame of January 2016 through December 2018 across all 3 Office of Medical Devices and Radiological Health divisions, which we also refer to as OMDRO, and in this included international recalling firms. For those that are not familiar with OMDRO's structure, it's broken up into 3 geographic divisions. There's Division 1 in the east, Division 2 and centrally located, and Division 3 in the west. The work group's review of the 118 Class 1 recalls included the root cause analysis, corrective actions, and follow-up inspection data. Over these next few slides, we will go over some of the information that was gathered during this review. This next slide just illustrates how the Class 1 recalls were broken down across all 3 divisions. As you can see, Division 3 had the highest number of Class 1 recalls in this analysis. Of the 118 Class 1 recalls reviewed, there were 74 inspections initiated and completed after the recall. These inspections were not necessarily initiated because of the recall, they just occurred after the recall was initiated. So of those 74 inspections, 44, or 59%, resulted in a 43 being issued citing quality system deficiencies, while 30 inspections did not identify any quality issues and no 43 was issued. Now, for those of you that are not familiar with ORA's term 483, it's just a shortened title for the FDA Form 483 Inspectional Observations. This is specifically the form number and how investigators document their observations of citations during inspections. So going back to those 74 inspections that were conducted after the recall, 52 of those were actual follow-up inspections that covered the Class 1 recall. Of those 52 inspections, 25, or 48%, resulted in a 483 citing deficiencies related to the actual Class 1 recall. So what does this mean? Approximately half of the inspections, which covered the Class 1 recall, resulted in 483 items specifically related to that Class 1 recall. This next slide lists the top 5 product codes for the Class 1 recalls reviewed with their associated root cause. The top 2 and bottom 2 product codes are cardiovascular devices, and the middle one, CBK, is anesthesiology. It is important to acknowledge, though, that cardiovascular devices are more high risk and complex by their nature. So there are a lot more Class 1 recalls associated with these devices. Not surprisingly, device design was present in all 5 of these product codes, with it being the top root cause in 4 of the 5. Process controls are also listed in 4 of the 5 product codes. So overall, in looking at these 118 Class 1 recalls, device design and process control were the most common root causes for these Class 1 recalls. Now we will turn to the most recent study, which analyzed Class 2 recalls. Similarly to the Class 1 recall analysis, this work group was created to conduct an in-depth root cause analysis of 437 fiscal year 2020 Class 2 recalls and included firms from across all 3 OMDRO divisions, including international recalling firms. The goal here was to provide industry with feedback on their corrective actions and to identify best practices both internally and for industry. Before we get into the details of the data, here's just a snapshot of the 437 recalls by division. The largest number of Class 2 recalls were in Division 1, which is really not surprising because that is our largest division by volume. This data, however, contrasts with the data from the Class 1 recall analysis, where the largest number of Class 1 recalls were in Division 3. There does not seem to be an obvious answer as to why this flips, but it could just be the location of these firms that manufacture higher-risk devices in the data set that we reviewed. Looking first at industry types associated with these Class 2 recalls, you can see the top areas of industry included orthopedic, cardiovascular, radiological, chemistry, and surgical devices. They made up approximately 71% of these Class 2 recalls. These top industry types will be looked at individually later in this presentation. This next slide compares and overlays the root cause versus the industry type. If you notice, each of the top industry codes we looked at earlier, orthopedic, cardiovascular, radiological, chemistry, and surgical, all contain process control and device design root causes. Next, we will see the breakdown of these top root causes for each of the 5 industry types. Before we move on to these next 5 slides, I wanted to point out that we are only displaying the top root causes that are over 5%, and this is in order to focus on the main root causes and not distract with the smaller percentages. The top root cause that we observed in orthopedic devices was process control followed by device design. These 2 areas represented 57% of the total root causes for industry type. Some examples of recall issues seen in orthopedic devices involving process controls were line clearance issues, product or labeling mix-up, and inadequate record keeping and documentation. This chart represents the top root causes seen for cardiovascular devices. Process control was again the top root cause for these Class 2 recalls, followed by device design. This represented 53% of the root causes for these products. Examples of process control issues from the recalls reviewed for cardiovascular devices were inadequate process procedures, soldering failures, and component failures. For radiological devices, the top root causes observed were software design followed by device design, representing about 63%. Examples of software design issues from the recalls reviewed include software bugs, unvalidated software changes, and lack of testing. Similarly, for chemistry devices, the top root cause was software design, followed by device design and process controls, representing about 54% combined. Some examples of these issues with software design seen in chemistry devices were software bugs, supplier issues, and design validation issues. Lastly, this chart represents the top root causes for surgical devices. The top root cause was process control at about 50%, followed by device design at 11%. Some examples of recall issues seen in surgical devices involving process controls include supplier issues, specifically component failures, inadequate production and process work instructions, and packaging issues. Another way we looked at the data was the number of recalls per recalling firm. This slide shows that breakdown. A takeaway from this slide is that FDA is starting to look more closely and more in depth at these Class 2 recalls. When we see multiple recalls for one firm, it signals concern for FDA. This may generate certain questions that we ask that manufacturer, such as, does it involve the same root cause? Is it the same device? Is there a systemic problem going on? When a firm issues a recall and implements corrective actions, are they looking at other similar products or systems to ensure the issue is resolved? Some of these questions guide follow-up coverage during follow-up inspections of these firms. Last, we looked at next inspections that identified quality issues. This chart depicts the percentages of quality issues observed during inspections. So of the statistically significant subset of Class 2 recalls we reviewed, we identified firms who had inspections after the initiation of the recall. Of those inspections, 55% found quality issues. I do, however, want to add a statement of clarification. The time frame for this data set we reviewed was fiscal year 20, and because that is so recent, with so many recalls still in progress, along with the travel restrictions encountered, the number of inspections is lower than what we would normally expect. So within those quality issues observed during the next inspection, 33% of those were related to the actual Class 2 recall. This is significant and raises questions of whether the firm's corrective actions are appropriate or adequate, and if the root cause identified is even accurate. The chart also indicates that 67% of those quality issues observed were not directly related to the recall. And I don't want people to think that this is something to be comfortable with or to ignore. This leads FDA to question what were they related to. For example, they could be indicative of a systemic quality issue, unreported recalls, quality issues that caused expansions of recalls, or even could lead to new recalls down the road. So now this leads us to our first knowledge check. True or false? The top root cause identified in the Class 2 analysis was process control. And the answer is true. Next, we will present our key takeaways of the analysis of these Class 1 and Class 2 recalls. First, we observed that the top root causes identified across various medical device specialty areas or product codes were design controls for Class 1 recalls and production and process controls for Class 2 recalls. When FDA does follow up on these recalls, investigators may focus in these areas. Second, we wanted to emphasize the importance of conducting a thorough root cause investigation. These investigations should be systemic to help ensure the root cause is identified and ultimately appropriate corrective actions are implemented to prevent the recurrence of non-conforming product, i.e. a recall for the same or similar issue. Third, systemic issues that are not addressed appropriately through corrective actions can result in firms having multiple recalls for similar processes, procedures, validations, etc. A firm's ability to verify the effectiveness of their corrective actions is critical to ensure root cause issues have been addressed appropriately. Next, we will look at best practices. Looking specifically at production and process controls, we observed the following examples of root causes in the Class 2 recalls we analyzed. I don't have time to review each example, but we're going to offer some insight to a few of them. For packaging and labeling issues, one example is packaging integrity regarding the sterile barrier system. Physical characteristics of the device, movement during shipping, and its effect on the sterile barrier system should be considered as part of the design input. This will ensure proper controls are in place and help prevent expensive corrections such as retooling, new packaging materials, repeated qualifications, and repeated validation testing. In many instances, these issues can generally be discovered during the process validation and addressed before moving to performance validation of the sterile barrier system. Inadequate equipment and/or process validation. This really speaks to equipment that may not have been properly maintained or new equipment that was not properly validated. Inadequate process procedures. For instance, they could be inadequate to ensure the product meets specifications and may lack clarity and detail. Lastly, we have supplier issues. Manufacturers must work with suppliers to establish critical controls, limits, and an ongoing review of process control data to ensure that the validated processes operate within the state of control. Remember, the supplier may own the process, but the manufacturer owns the product. Where required by the QSR, process controls must be implemented and demonstrated to the customers by both classic verification and validation. The initial cost of validating a process can be significant, but doing so inevitably adds value by ensuring a stable process that consistently achieves desired results with a high degree of assurance. Once a validated state is achieved, process controls must be established to ensure processes remain in that state of control. Continuing in production and process controls, manufacturers must establish and maintain process control procedures that describe any process controls necessary to ensure conformance to specifications. In other words, manufacturers are responsible for the evaluation of manufacturing processes to determine whether the lack of process controls could affect the reliability and repeatability of the manufacturing process. If it is determined that the process controls are required, those controls must be established, verified, implemented, and monitored to the degree necessary to prevent product non-conformance. The characteristics of the device define process and required process controls. For example, manufacturers typically begin with design controls to develop the manufacturing process based on the product design, materials, and components along with necessary quality requirements. manufacturers may validate production processes through a series of qualifications, which can include equipment installation qualification, operational qualification, and performance qualification. Based on these activities, documented instructions, for example, procedures, process specifications, process routers, are developed for routine production controls. A big takeaway from this analysis is how vital it is to identify the true root cause or root causes. This can be achieved by performing a thorough and broad investigation, resulting in objective evidence to support a root cause determination. This should also include an evaluation of the impact on other lots, processes, products, and facilities. An effective investigation will help prevent a potential problem from occurring in the first place or an existing problem from reoccurring by identifying the issues or root causes that need attention. Common pitfalls we observed include not asking the why enough times, therefore not getting to the true root cause. Associated lots not identified or evaluated, basically an incomplete scope for your recall. Inadequate justification for the absence of a root cause. Conclusions not being supported by data, in other words, using common sense or common knowledge. Not having the right people involved in your investigation. And the absence of documentation for justification. Corrective actions. These actions are taken to address the root cause and must be commensurate with the magnitude of the problem and associated risks. For instance, many manufacturers tend to overuse training as a corrective action and are not adequately addressing the systemic problem. Corrective actions must include any additional actions if necessary. These may include component suppliers, acceptance activities, field actions, etc. Firms must also verify and/or validate corrective actions to ensure that the actions are effective and do not adversely affect the finished device. What this does not mean is just verification that the actions were taken. What this means is that criteria has been established to determine if the corrective actions taken are effective. For example, a firm might review data from manufacturing step XYZ after, let's say, 3 months or 50 manufacturing lots, and the CAPA would then be considered effective if less than X nonconformances occur. This might involve an audit of the affected process and review the data that demonstrates the system changes will prevent the system from occurring again. Review the effectiveness of the corrective actions by reviewing data to determine if similar quality problems exist after implementation. Last, the quality system must provide for control for finished devices, both distributed and not yet distributed. Continuous monitoring of the quality system is essential in preventing any reoccurrences. This includes trending product complaints and any nonconformances in production. So overall, manufacturers should be ensuring that their corrective action documentation demonstrates to FDA that the manufacturer's quality system is effective and enables them to identify problems quickly and implement effective corrective actions. This is our second knowledge check. So true or false? Root cause investigations do not need to include the quality system. And the answer is false. Follow-up inspections. During FDA inspections, we just want to emphasize that Class 2 recalls are being covered and reviewed. As a whole, FDA wants to see that the firm's quality system is effective and enables that manufacturer to identify problems quickly and implement their effective corrective actions. Thus, this would prevent the distribution of violative product that can pose a public health risk. When we're following up on these recalls, our goal is to ensure that the firm has implemented these adequate corrective actions and focus on the quality system so that similar recalls are prevented in the future. Example areas of coverage include coverage of the CAPA subsystem to verify issues are detected and resolved, general review of key elements of the recall, review of the depth of the investigation to ensure that it is sufficient, ensure the decision process is based on risk, and ensure the action plan is developed for corrective actions. We also want to verify that a solution was effective and did not cause other potential nonconformances. Last, I just want to mention that every firm is subject of these follow-up inspections, even if they participate in the MDSAP program. So in summary, the top root cause identified in Class 1 analysis was device design, and the top root cause identified in the Class 2 recall analysis was process control. Additionally, during our review of these recalls and the resulting inspections, we noticed that a lot are resulting in related 483 items to those actual recalls. Last, we just wanted to emphasize how important it is to identify your true root cause or root causes in order to take appropriate corrective actions and prevent these recalls in the future. This concludes our presentation, and I wanted to provide some resources and contact information for you. We've included the email address for each division along with each state that each division covers. We've also included the link to the industry guidance page on the FDA website. Thank you so much for your time and attention. We really appreciate this opportunity to share this recall review and analysis. And now I believe we will take questions.
Thank you, Meredith, for that fantastic presentation. We already got quite a few questions in the queue, but please keep them coming in the question and answer chat box. Please put @ and then Meredith, and then we'll get that question in. But to start off with the first question, who is responsible for identifying the cause of the recall? Is it the firm or is it FDA?
Hi, great question. We would place that on the firm since they're the most familiar with their devices, and two, they should be conducting that investigation to discover, what caused this issue to occur. There could be multiple causes as well. So we would put that on the firm, but that is something we do have dialogue about. And that when they've concluded their investigation and they talk to us, sometimes we'll discuss that with them, but that is the firm's responsibility.
Okay, so the cause is the firm's responsibility. Another question, who is responsible for the classification of the recall, the firm or FDA?
So that responsibility resides with FDA. So after we receive a recall and we review it and process it and input it in our system, CDRH is actually who ultimately, we as a recall coordinator will make a recommendation based on precedent and experience. But ultimately the reviewers up in CDRH are who classify the recall.
Okay. And do recalls come specifically from FDA, and are they a result of FDA investigations from an inspection?
So recalls can come in a couple different ways. We have them where the firm discovered the issue on their own, could be through complaints. If they're trending their complaints, it could just be from something they noticed internally when they do internal investigations. But it also could be an issue that is discovered during an FDA inspection. So it can come either ways, but it would not specifically the language coming from the FDA. We may discover the issue and discuss that with the firm, but it usually comes all the way from the firm unless there's a mandatory recall involved, which is not. It's very rare.
Okay. And to build off of that with regards to the actual recalls themselves, are Class 1 recalls mostly associated with Class 3 medical devices in your experience? And can you provide any examples to this?
I would say most often when you have a Class 3 device involved, it would most likely be a Class 1 recall, just because, like for instance, like cardiovascular devices, just based on the type of device and the nature of that device, they would normally, because they would potentially have the biggest harm if there was a malfunction, would be a class 1 recall. I don't know that I could say like a blanket statement that they always are, because we have some, I mean, there are some class 2 devices that, depending on what the malfunction is and maybe what the population that it affects, it can be a class 1 recall.
And further building off the class 1s, when would a class 1 recall be reopened?
So after termination, it is very uncommon to reopen a recall. In fact, I want to say I may have only heard of one in my career as a recall coordinator, and it wasn't my recall, but it could be after it's terminated. We received some additional information, maybe from complaints or from the firm, that something changed, or maybe they found out something they were not aware of beforehand. For instance, when a recall is terminated, if there's some process that maybe was long-term that really wasn't necessary for the termination, it could be like if they're looking for a new approval on something, or if maybe they found out when they went to fully close the CAPA, it was not effective, they found a new piece of evidence, then we could reopen it. In fact, I think the one that I'm aware of, I believe it had something to do with a CAPA at the end, but it's very, very rare. We try to have everything organized and ready to go and completed when we do terminate, so we don't ever have to deal with that.
Okay. And this next question I've actually seen quite a bit with regards to the classification, asking the question of why are the device classifications versus the recall classifications flipped? In that class 1s being the highest risk with regards to recalls, whereas class 1 devices are the lowest risk devices.
That's a good question. I don't fully know the answer to that, but I do believe the recall classifications, which cover all commodities, foods, drugs, animal drugs, human drugs, biologics, those were in place prior to, I think, the device classification. So I'm not really sure why they did them opposite, but I know they weren't created at the same time.
Nope, and you're correct. So as you noted, the drugs food recalls predate the 1976 medical device amendments. When they put the device classifications in place during the late 1976, 1970s, I can't answer as to why they went with the class 1, class 2, class 3 devices, but you're absolutely correct. The recall classifications predate the device classifications. So with that, we have a question with regards to timing. What or are there requirements requiring the timing and closure of a recall?
So we do not have any specific time frames for termination. We really want the firm to do a complete and effective recall for really within reasonable timeframes, but every recall is different, and depending on the corrective action or how many customers are involved, how much follow-up is needed, and even like long-term, if there are certain corrective actions that may take place, further down the road, if it's a multi-step process, there is no time frame for termination, but there are elements that need to be present in that termination, and that it needs to be effective, and the firm needs to provide a request for termination and demonstrate the effectiveness of that recall and that corrective actions were implemented. And I believe it's in 21 CFR Part 7, where it discusses when to request a termination.
Okay, thank you for that. When FDA initiates a recall and not the manufacturer, so now we're talking about a mandatory recall, which I know there's not a whole lot of them, who terminates the recall, FDA or the manufacturer?
I have actually not personally handled a mandated one, but I can do more research into that. My suspicion would be that it would be the same termination process that would follow for any recall, and that once the firm believes they are ready for termination and they request termination, we would review it and then FDA would terminate it. There could be additional review involved in that termination, maybe outside of just the recall coordinators. I'm sure there would, I'm positive that there would be maybe somebody in CDRH that would be involved in that review. So we would be the one terminating it, though.
Okay, next question. I noted that for Class 1 analysis, FDA used a broader time frame, and for the Class 2 analysis, only looked at 1 year. What is the reason for the difference in the report analysis that you talked about?
So in general, the number of Class 2 recalls is the largest category that we have. So there are nowhere near as many Class 1 recalls. So when that analysis was performed in 2020, they were able to take a larger time span. And I think it was only for, it was like, I think 3 years. They had 100 recalls that they could look at for Class 1 recalls, whereas on average, we receive, let's see, 900 to 1,000 recalls total per year. And I think for fiscal year 2020, there were 850 Class 2 recalls. So we just didn't have the time to review all 850. So we picked a statistically, we analyzed it based on a statistically significant subset that we chose. And it ended up being those firms with 3 or more recalls. So it ended up being half. So we did 437 out of 850, but that's why we couldn't do the same time frame. Maybe in the future, if we had more time to do the analysis, we could maybe look at a full year or maybe pick a different subset.
Okay, great. Going further into the analysis, and this is kind of an opinion type question. So you noted that there's a large percentage of process control findings. Do you believe that this large percentage of process control findings are the result of outsourcing to third parties or to contract manufacturers?
Ooh, I don't know that I can give just an opinion on that, because we see it more than just with contractors and outsourcing. So we see it within the actual, on-site manufacturing too. So I don't know that I could specifically, I've never looked at like a trend like that, so I don't know that I could specifically speak to that.
Understood. The next questions have to do with agreement on calls. So there's a couple questions. I'll try to loop them together the best I can. Does FDA need to confirm or agree with the root cause of the recall identified by the manufacturer? And if so, what is the best way to get this confirmation from FDA?
Good question. So when we receive the recall and we are reviewing all of the information and the strategy and any investigations that are provided, we look at the root cause. And if we have a question or a concern with that, we try to discuss that immediately with the firm. And sometimes, most of the time, that's resolved. It could just be we think they should maybe dig a little bit deeper in their investigation, or maybe we'll ask them provide the evidence of why they really think that that's the root cause. I would say generally it's usually resolved. I don't know that I'm aware of any instances where we could not come to an agreement. If there was serious concern, we would work very closely with the center and the experts up there to maybe come to some kind of solution or meet with the firm to discuss further. But most of the time it's resolved.
Okay, and that helps answer the next question was, what if we as a manufacturer identify a cause and FDA disagrees? So it sounds like you guys would work through that to come to some agreement.
Correct. And also knowing that some of the information for each recall is present on FDA's website in the enforcement report. I can't remember if root cause is posted, but we don't want our system necessarily to contain something as important as root cause that is in disagreement with what the firm has, because at the end of the day, if we want to make sure a recall is effective at eliminating these root causes, we need to agree. So, and we want to hopefully prevent that from happening again. So we don't want to just be at odds and have something different.
Okay, perfect. And with that, that leaves us about a minute. So I'm going to turn it back over to you to give us your final thoughts.
Okay, great. So just a couple items I just kind of wanted to restate and remind is to remember to conduct an in-depth root cause investigation to determine your root cause or root causes contributing factors to what caused the issue. So just a reminder, don't stop too early in the investigation. Take bias out and follow your data and your questions and really have the right people involved on your team. That's really important. Also, just to ensure that corrective actions are adequate and appropriate, make sure they're appropriate for the magnitude of the issue and adequate to resolve the issue, and do more than just verify that they're complete. So verify that they're effective in preventing the situation from occurring again. Review your effectiveness of the corrective actions by reviewing data to determine if quality problems are existing after the implementation. And I know this was stated in the presentation, but I thought it was important. Just overall, manufacturers need to ensure that their corrective action documentation demonstrates to FDA that their quality system is effective and it enables the manufacturer to identify problems quickly and implement effective corrective actions. And lastly, just kind of reiterate that we are reviewing, following up on Class 2 recalls, so just be aware of that. And thank you for this opportunity, and that's all I have.
Okay, thank you for that call to action, and thank you, Meredith, for this whole presentation and Q&A session. I think it was a very engaging discussion on recalls and the recall analysis that you all did. So fantastic. So with that, we're at the end of the REdI 2021 medical devices track. So this closes out part 2, products on the market portion. So whether you are with us for the past day and a half or the entire 4 days of the conference so far since Monday, I'd like to thank you for making this program such a success. As Brenda and Elias mentioned earlier, without everyone here, this event would not be possible. Our mission at CDRH and at the Division of Industry and Consumer Education in particular is to serve you and provide you with understandable information about medical devices. So we hope that we met your expectations with this medical device track as part of the REdI conference. Additionally, I'd like to thank Elias Malice for moderating the first part of the device track, and also to thank Donna Headley and Tanya Wilbon for facilitating the questions that were coming from you. And also a special thanks to all of our CDRH speakers who presented for you over the past few days. I think the presentations were phenomenal with regards to providing great content and information on some very important topics. And I thank them for putting in the hard work, for putting these together and presenting them and speaking to you on them the last day and a half. Also, a thank you to Brenda Stoddard for leading the coordination of the fine program that is put together annually with regards to REdI. So, and we also want to thank the expert crew working behind the scenes, Jeff and Julie and their team, that make this online experience so smooth. And then last but not least, I want to thank all of you. So thank you. We know that your time is valuable and important, and we appreciate that you've invested this time, this important time, to have with us. We hope that you took away something that's valuable. I know that I have, listening in the last day and a half, picking up even more information than I've already had known for the last 26 years in this industry. I always pick up new things from this conference, but we definitely appreciate you taking that value time to help do the hard work to advance the availability of safe and effective medical devices. So as much as we can help you, we are going to help you to basically design and develop and manufacture and get those devices safe and effectively onto the US market. Because as I noted yesterday, these are medical devices we all use and that we all depend upon. So I understand the importance of the work that you do, as you understand the importance of the work that we do by attending conferences like this and learning and getting more information. So last, thank you again for joining us. Please be safe, and we'll see you hopefully again at next year's annual REdI conference. We're going to break for lunch in just a minute here, but please come back in an hour from now at 12:40 Eastern Standard Time, US Eastern Standard Time, for the start of the Biologics track that will be given by the Center for Biologics Evaluation and Research. They are going to end out the REdI conference this afternoon and tomorrow. So please stay tuned and come back for that Biologics track. It will be great, just like the device and previously the drug tracks that you've heard. So thank you again, and have a nice lunch, dinner, or breakfast, depending on where you are. So thank you.
Good day, good night, everyone, and welcome back from break. Hopefully you were able to use it productively and also grab something to eat if that worked for you. Once again, this is Brenda Stoddard with CEDA Small Business and Industry Assistance, and I hope that everyone is thoroughly enjoying the event so far and learning a lot. Continue to invite your friends to join us. They can join us via Adobe Connect through the registration process or through YouTube. And everything is on a one-stop shop webpage if you were here the first day, and that's the page where you signed in. Well, we had the CEDA track on Monday and Tuesday. The CDRH devices track began Wednesday morning and finished up today, just before lunch. And now we begin our third track, which will take us out to tomorrow, just around 3:55 p.m. Eastern Time when we close. I don't know how many of you were with us at the opening of the event on Monday morning, but it is my utmost pleasure to welcome the Center for Biologics and Evaluation, CBER, as the host of our third track and most recent addition to the REdI agenda. This has been a long-standing desire of mine and of the SBIA program, and today we accomplished that goal. Thus, without further ado, I welcome my CBER colleague, Dr. Larissa Lapteva. Dr. Lapteva is a physician with a long-standing experience in clinical research with novel drugs and biological products, including cell and gene therapies. Since joining FDA in 2006, Dr. Lapteva has held review and supervisory positions in CBER and provided scientific and regulatory advice for clinical development programs with investigational products across different therapeutic areas. Dr. Lapteva, I hope you are as thrilled as I am to be the first CBER office involved in this three-track agenda. So please, audience, help me to join me in welcoming Dr. Larissa Lapteva. Thank you.
Good afternoon. It's a great pleasure to welcome everyone to CBER track of the Regulatory Education for Industry Conference. My name is Larissa Lapteva, and I'm the Associate Director in the Division of Clinical Evaluation, Pharmacology and Toxicology in the Office of Tissues and Advanced Therapies in the Center for Biologics Evaluation and Research. With a team of moderators, I will be hosting CBER track, welcoming our speakers and faculty, and moderating CBER sessions. As many of you know, the Center for Biologics oversees a wide variety of biological and related products, including blood products, vaccines, allergenics, tissue-based and cellular and gene therapies, as well as devices used with biological products. We regulate products throughout their life cycle, from the time of preclinical development and first-in-human studies through the time when products are already on the market and used in clinical practice for healthcare delivery. As you have already heard, this is the first time when CBER participates in the Regulatory Education for Industry annual conference organized by the Small Business Assistance Program. On the first day of the conference, you've heard the remarkable story of the development of vaccines and treatments for COVID-19 and the emergency use authorizations issued by FDA to combat the coronavirus pandemic. The deep understanding of science and knowledge of the available technologies, coupled with the tremendous work and dedication of the teams from the three centers and the rest of the FDA, but for CBER particularly from the Office of Vaccines Research and Review and Blood Research and Review, have helped make these much-needed products available for vaccination and treatment. But besides the unprecedented situation with the pandemic, it really is about time for the Center for Biologics to join this annual event together with the Center for Drugs and Center for Devices, because in recent years, we have seen a remarkable growth in the development of novel products, regenerative medicine therapies, including cell therapies, gene therapies, and tissue-based and tissue-engineered products. And with this booming development in the past 5 years, CBER has approved technologically advanced novel cellular and gene therapies for the treatment of serious diseases. All of our presenters today structured their presentations based on numerous applications they have reviewed, and we will be sharing our collective lessons learned from the many product development programs we see, and based on which we maintain and continue developing the current policies that CBER has in place for the diverse products that we regulate. But this time we have only a day and a half to share and discuss the wealth of our experiences. Therefore, CBER presentations today and tomorrow will be focused on the biological products which are regulated by one of CBER's offices, Office of Tissues and Advanced Therapies, or we call it OTAT for short. In OTAT, we've been receiving increasing numbers of applications with new product development programs, the largest bulk of which over the years have been cellular therapies and gene therapies. Those are the areas of an increased interest and uptick in clinical studies, with now observable trends of transitioning from academic research toward industry-sponsored product programs. So today we're glad to be part of this educational effort and discuss a variety of topics, from the explanation of tissue role and the regulatory framework for human cells, tissues, and cellular and tissue-based products, to the different aspects of the design, manufacturing, control specifications, and preclinical and clinical development of cellular and gene therapies. We will also highlight the available regulatory programs established to facilitate development of these novel treatments, discuss applicable consensus standards, highlight the unique issues with protein therapeutics, illustrate the development of medical devices used with biological products, and share CBER's approach to manufacturing facility inspections with examples of inspectional observations. Please prepare for a fun ride with us during these 2 days. Our agenda wouldn't be complete without everything we managed to include in it. And without further ado, our first presenter for the CBER track is Dr. Wilson Bryan, who is the Director of the Office of Tissues and Advanced Therapies, and he will give you an overview of the office's multiple and exciting activities.
Good afternoon. My name is Wilson Bryan, and I'm Director of the Office of Tissues and Advanced Therapies, or OTAT, and it's my pleasure to provide this introduction to OTAT. Now, this graph shows the number of investigational new drug applications, or INDs, submitted to OTAT or its predecessor offices over the last several decades. The part of the graph that is in orange represents expanded access INDs, and these INDs are primarily for treatment of patients. They're not for doing research, they're for making investigational products available for treatment. The part of the graph that is in blue represents INDs with product development programs. That is, they're doing research to gather evidence of safety and effectiveness to bring a product to market. OTAT came into existence in 2016. At that time, we received 223 total INDs, that is expanded access plus research INDs. By 2020, 4 years later, that number had almost tripled to 666 INDs. So as you can see, there's a lot of excitement about the products that we are regulating and a lot of interest in developing new products that are regulated by OTAT. Now, part of the excitement is that the great majority, over 90% of our applications, are for severe and life-threatening diseases, bad diseases, and approximately 50% of the applications are for rare diseases. So the great majority of our work is for serious and life-threatening diseases, and about half of it is rare diseases. And so we're very interested in moving these products forward. A few fundamentals about OTAT. First of all, as many of you know, the FDA has seven centers, which are depicted on this slide. And the three centers on the left of the slide are the ones that regulate medical products for humans. And these include the Center for Drug Evaluation and Research, or CDER, the Center for Devices and Radiological Health, CDRH, and then highlighted here in yellow, the Center for Biologics Evaluation and Research, or CBER. And in CBER, particularly in OTAT, we work very closely with the Oncology Center of Excellence, which coordinates the clinical review of oncology applications across CDER, CDRH, and CBER. This diagram shows the structure of CBER. The center director is Dr. Peter Marks. The deputy director is Dr. Celia Witten. And the three boxes across the bottom of this diagram show the three offices that are specific for specific types of products. On the left, the Office of Vaccines, which regulates the pneumonia vaccine, the COVID-19 vaccine. In the middle, the Office of Blood, which regulates blood products. And then highlighted here in yellow, the Office of Tissues and Advanced Therapies, or OTAT. I do want to mention that for the purposes of this session focused on CBER products, we worked closely with the Office of Communication, Outreach, and Development, or OCOD, and the Office of Compliance and Biologics Quality, or OCBQ. OTAT's mission is to promote the public health through collaborative, science-based regulation of medical products. And this includes facilitating drug development and ensuring safety of individuals. And our regulatory decisions must be data-driven, impartial, and compassionate. When we say science-based, I note that OTAT has 22 research laboratories, which put out 31 publications in 2020, and also in 2020 participated in over 40 external conference presentations. So our lab scientists are very research oriented and focused on keeping us grounded in solid research. OTAT products, we regulate a wide variety of products. These include purified and recombinant proteins for hematology, such as coagulation factors, thrombin, fibrin sealants. We regulate antivenoms for snake bite, antivenoms for spider bites. We regulate stem cell and stem cell-derived products, all kinds of stem cells, hematopoietic stem cells, mesenchymal stem cells, cord blood-derived stem cells, embryonic stem cells, induced pluripotent stem cells, any type of stem cell we regulate it. Terminally differentiated cells, such as pancreatic islets, myoblasts, hepatocytes, and we also regulate therapeutic vaccines and other antigen-specific active immunotherapies. These include cancer vaccines and immunotherapies, such as dendritic cells, lymphocyte-based therapies, and cancer cell-based therapies. We also regulate non-infectious disease therapeutic vaccines. These can be peptides, proteins, or small molecules. For example, a therapeutic vaccine to treat Alzheimer's disease would be regulated in OTAT. A therapeutic vaccine to treat opioid addiction would be regulated in OTAT. So therapeutic vaccines to treat non-infectious diseases are regulated in OTAT. And of course, we regulate gene therapies. Lots of excitement about gene therapies these days. And sometimes these gene therapies are genetically modified cells, or they can be gene therapies that are delivered by plasmids or viral vectors or bacterial vectors. We also regulate xenotransplantation products. Somebody decides that they want to grow a kidney in a pig and transplant that into a human. Then that's a xenotransplantation product, and we would regulate that. Tissues and tissue-based products. Some devices and combination products, particularly devices that have a cellular component. Now, as examples of recent approval in OTAT, an in vivo gene therapy, Luxturna. This product is directly administered adeno-associated viral AAV vector-based gene therapy that targets a rare disease caused by mutations in a specific gene. And it's indicated for the treatment of patients with a rare mutation that causes retinal dystrophy. These patients have progressive visual loss and eventually go blind. Now, I'm going to show two videos, and they are of a young boy who has this rare retinal dystrophy, and his vision is not good, and he receives the Luxturna treatment in one eye, and the other eye is untreated. And these videos are taken 90 days after the treatment. And the task that he has is to navigate a maze. And the maze has arrows on the floor that he's supposed to look at and figure out which way to go in the maze. And the maze also has obstacles, things that he's supposed to step over. It has a stop sign. And as you'll see in this first video, which shows the untreated eye, and he's struggling. He says he sees the black squares, but he really doesn't see the arrows. And so he's not sure which way he's supposed to go. And he says this is hard. He's struggling to know which way to go. He says, I can't even see anything. This is a bad disease. It really very frustrating for him as you see him struggling to try to complete the maze using his untreated eye. Now, let's go forward to using the treated eye. So again, this is 3 months after receiving the treatment in one eye.
And you can start whenever.
And you can see the arrows on the floor in this video. And he is moving. He's got no trouble seeing the arrows, following the maze. He steps over an obstacle. It's almost too easy. So this product, Luxturna, helps blind people see. This changes lives. And this is typical of what we've seen with so many of our gene therapies. Let's talk about ex vivo gene therapy. And we have now 5 products approved that are examples of ex vivo gene therapy. They are chimeric antigen receptor T-cell products. So what they do is they take a patient with a hematologic malignancy, and these are patients who had run out of treatment options. They had tried all the available treatments and either had not responded or had relapsed. And they take the blood cells from these patients, they isolate the T cells, which are part of the immune system, and then genetically engineer those T cells to express a protein on the surface that will attach to the cancer cells. So they then take these genetically modified T cells, put them back into the patient, and it latches onto the cancer and attracts other T cells to kill the cancer, and it activates this T cell, and it's like science fiction. They take your immune system cells, genetically manipulate them, and put them back in to attack your cancer. It's just, these are life-saving products. We also have some exciting cell therapies. As an example of an allogeneic cell therapy, we recently approved Stratigraft. And this is a product that is produced from 2 kinds of human skin cells, keratinocytes and dermal fibroblasts, grown together to make a bilayered construct. And it's used to treat adults with deep partial thickness burns. This shows 2 rows of photographs. The row on the top, the 5 images across the top, show the leg treated with Stratigraft. The 5 images across the bottom show the leg treated with autograft. And autograft is the standard of care. If you look at the images on the far right, which show the results at 1 year, Stratigraft seems to do pretty much just as well as autograft. And autograft is a painful process. So this really can save patients from a painful autograft procedure and give good healing results. So this has been a real boon to patients. Purified plasma protein. Last month we approved Riplasm, which is plasminogen. It's a purified plasma protein. And it's used to treat a very rare disease, plasminogen deficiency type 1. And this disorder can impair normal tissue and organ function and can lead to blindness. These patients have what are called ligneous lesions that can grow in different locations, can grow in the eyes, can grow in the trachea and block breathing. The image on the left shows one of these ligneous lesions on the inner surface of the eyelid before treatment. The patient is treated with Riplasm for 4 weeks, and 4 weeks later, the lesion is gone away. A dramatic result. A very rare disease. I think the evidence of effectiveness came from just 15 patients, because when you have these sorts of dramatic results, it doesn't take a large number of patients to know that you have a product that's beneficial and needs to be on the market. Now, our job at FDA and at OTAT is to facilitate development. And let me mention what's going on, particularly in gene therapy. This graph shows the number of research INDs. So this does not include expanded access INDs. These are research INDs submitted to OTAT each year, last several decades. And again, I'm going to highlight 2016, which was the year that OTAT came into existence. And there were 67 research INDs received in 2016. And 4 years later, in 2020, we've got 160, more than twice as many research INDs 4 years later. So there's a lot of excitement about gene therapy, and we're excited about gene therapy, of course. And we're also excited about cell therapy. This graph shows you what's been happening with cell therapy INDs the last few years. And again, we'll start with 2016, when we had 63 INDs. And partially as a result of the pandemic, there's been a huge increase in the last year, so that in 2020, we had 151 INDs submitted. Now, this is over twice as many, again, as we had in 2016. There's a huge jump, you see, that occurred from 2019 to 2020, and that's because there's a lot of interest in cell products and cellular-derived products as treatment for COVID-19. So we saw a huge increase this past year in the number of research INDs and cell therapies. Now, our responsibility in OTAT, of course, is to facilitate the development, and we have a variety of expedited programs. And some of these are accelerated approval, priority review, and fast track designation. I'm going to focus here in this graph on two expedited programs: breakthrough designation and regenerative medicine advanced therapy, or RMAT designation. Now, breakthrough designation came into existence in 2012. So it's been around for just about 9 years. And we have received a total of 120. This slide is cumulative, by the way. It's not each year. It's 120 breakthrough designation requests received, and we've granted 37 of those. So that's about a 31% rate. So we have 37 products that have breakthrough designation. In contrast, RMAT designation came into existence at the very end of 2016. So it's been in existence for only 4.5 years, but we've already received more RMAT designation requests, 173, than we received a breakthrough designation requests. And we have granted 62 RMAT designations. So we've granted more RMAT designations than we have breakthrough. So between RMAT and breakthrough, we have a total of 99 products that have either RMAT or breakthrough designation. So these products get extra attention from the FDA, and that's what they deserve, because they provided clinical evidence that what they can do for patients. In summary, the scientific advances, which have been very dramatic over the past couple of decades, they've spurred recent growth in research and development of advanced therapies, particularly cell therapies and gene therapies. And OTAT is dedicated to working with our sponsors, with academic sponsors and with pharmaceutical companies, to facilitate the development of novel products to treat serious and life-threatening diseases. This is what we are here for. I want to acknowledge all the folks that you're going to hear from over this afternoon and tomorrow from my Division of Cellular and Gene Therapies. I'm not going to name all these folks, but I thought I'd provide their photos along with their names. Also representatives from my Division of Clinical Evaluation and Pharmacology Toxicology, both pharmacology toxicology reviewers and some from our clinical team as well. We have representatives from my Division of Human Tissues, from my Division of Plasma Protein Therapeutics, Division of Regulatory Project Management, and I also particularly want to mention that Katarina Allen from our Office of Compliance and Biologics Quality, Division of Manufacturing and Product Quality. So she will also be presenting, I believe, tomorrow. And then the planning committee who put this session together, the folks who served on that planning committee, and then the folks who helped me particularly with this presentations, Drs. Anatole Rousey and Wang, who helped me with this presentation. The contact information for the OTAT, and I'll leave that up for a second, and then my contact information. If there are questions that aren't answered over the next half day or day and a half, and you want to ask somebody, if you can't reach the speaker, then you can send me a question and I'll get it to the right person. I usually don't know the answers myself, but I'm happy to help in any way I can. So here's my contact information, and I thank you very much for your attention, and I hope you enjoy the presentations the rest of this afternoon and tomorrow. Thank you.
Thank you, Dr. Bryan, for introducing us into the topics related to the development of tissue-based products and other advanced therapies. I love the last 3 slides with everyone's faces on them. When we can't see each other in person, here we have all CBER's faculty nicely put on the slides. But speaking of the faculty, I would like to repeat again that all CBER speakers who are presenting today have nothing to disclose. You have heard this morning the information about how to receive the continuing education credit. I will not repeat what was said this morning, but I will go over this information again tomorrow. And with that, now we can proceed to our first session in CBER track, which is going to be about the regulations described in Part 1271 of Section 21 of the Code of Federal Regulations and the respective policy considerations for what is known as tissue rule for human cells, tissues, and cellular and tissue-based products, or we also call them HCTPs. The session will include 2 presentations. The first one will describe the essence of the tissue rule and its applicability to product development. This presentation will be delivered by Mr. Scott Brubaker, who serves as the Director of the Division of Human Tissues in the Office of Tissues and Advanced Therapies. In this position, he leads the division in the maintenance of existing and development of new federal policies to support the regulations and the associated guidance documents governing the oversight of human cells, tissues, and cellular and tissue-based products. He also serves as the chairperson of the Tissue Reference Group and the Tissue Safety Team. Prior to working at FDA, Mr. Brubaker was the Senior Vice President of Policy at the American Association of Tissue Banks for 12 years. And before that, he worked for 18 years in the field of tissue banking and organ donation and procurement. So that would be the first talk of the session. In the second presentation in the same session, you will hear the outline of the regulations and policies related to donor eligibility determination, including exemptions and alternatives. The second presentation will be delivered by Ms. Safa Karandish, who is a consumer safety officer in the same division of human tissues. The primary focus of her current work is on tissue regulations, policies, stakeholder outreach, and review of cellular and tissue-based products. Prior to joining FDA in 2010, she had had over 24 years of experience in cellular therapy product manufacturing and cord blood banking in academic and industry settings. Now I will give the floor to Mr. Brubaker and Ms. Karandish, and we will have the Q&A part of our session after their presentations. While listening, please send us your questions through the Q&A chat box, and we will try to respond to as many as we can. Scott, take it away.
Greetings. I am Scott Brubaker, the Director of the Division of Human Tissues in the Office of Tissues and Advanced Therapies in CBER. This presentation covers Part 1 of the regulatory framework for human cells, tissues, and cellular and tissue-based products, referred to as HCTPs, found in Title 21 of the Code of Federal Regulations in Part 1271. The Part 1271 regulations are often referred to as the tissue rules. Specific events play an important role in the development of regulations, and the history surrounding the promulgation of the tissue rules is not an exception. You'll see that the safety of public health became a concern as the use of medical products of human origin became commonplace. Through federal regulatory processes, steps were taken to require safe practices. Our learning objectives are structured so you may understand events that led to promulgation of regulations designed to prevent the introduction, transmission, or spread of communicable diseases by HCTPs. You will also be able to list general requirements for manufacturers to meet the scope and purpose of the regulations in 21 CFR Part 1271, and you'll have knowledge to correlate criteria and definitions in these regulations to understand how an HCTP should be appropriately regulated. Let's review some pre-regulation history starting in the 1970s and 80s. Adverse events were reported that occurred both outside and within the United States where disease transmissions from the use of tissues or cells were recognized. They included diseases that caused mortality and morbidity such as Creutzfeldt-Jakob disease, HIV, and HBV, and were sourced from donors who were living that provided gametes for artificial insemination, or from deceased donors of cornea and human dura mater. Although transmission was donor-derived, risk was also identified related to processing steps such as pooling products from many donors, and there were also limitations of donor testing that were experienced. This included availability of tests and the potential for hemodilution of the donor specimen that can affect the accuracy of test results. Technical advancements continued through the 1990s, whereby human tissue could be preserved and stored for years by cell and tissue banks. And it was experienced that transmission of viral disease may not be recognized for years after the product was used. Such was the case surrounding HIV transmission from a donor whose organs and tissues were recovered in 1985, but communication and recognition of the donor-derived transmissions were realized only 6 years later. Limitations of communicable disease testing continued to play a role during this time, as did contamination by microorganisms during handling of tissues and cells. A turning point occurred when, in 1993, brokers were intercepted attempting to sell unprocessed human tissue to U.S. tissue banks. The donor tissues originated from Russia, Eastern Europe, and Central and South America. There were no donor records, and a donor specimen tested positive for hepatitis B. The regulation of banked human tissues from cadavers began in 1993 with an interim final rule, and a guidance document followed in 1995. At the request of Congress, the Government Accounting Office published a report in 1997 outlining gaps in federal regulatory oversight, and that same year, an infection occurred after use of a cryopreserved allograft heart valve was contaminated with fungus. A final rule was published and codified in 21 CFR Part 1270, and final guidance was issued to provide recommendations for donor screening and testing. There was a plan to further develop these regulations, and a tiered risk-based approach was described. In general, the goal was to prevent use of contaminated tissues and to expect clinical safety and effectiveness for tissues that are highly processed, used for non-natural purposes, are combined with non-tissue components, or that have systemic effects on the body. A series of proposed rules that formed the 21 CFR Part 1271 regulations were published over the next few years. Establishment registration was first, followed by donor suitability determination and current good tissue practice requirements. Another federal report, this one from the Office of the Inspector General in the Department of Health and Human Services, provided a profile of the external oversight system for tissue banking and identified limitations of the system. The same year, in 2001, a Clostridium sardellii infection from a cryopreserved femoral condyle allograft resulted in the recipient's death. The driving forces and challenges to finalizing the tissue rules are listed here. They include that a large number of tissue allografts can be processed from one donor, which can affect an equally large number of recipients. It became understood that as technological advances are made, the degree of tissue and cell manipulation increases. With the development of new tissue engineered products and cells that may be expanded in culture, hope for new therapies evolved, but public expectations remain that the product should be safe and effective. Although industry's self-imposed standards promoted safety, they are voluntary and not enforceable. Through experience over the last 3 decades leading up to the millennium, there were increasing concerns for public health. The regulatory framework for human cell and tissue products is organized into the 6 subparts shown here. To fully cover each of these subparts would require, at a minimum, a 20-minute presentation for each, so this presentation is limited to focus mostly on subpart A that includes definitions and the criteria for determining the regulatory pathway for a cell or tissue product. The tissue rules additionally include subpart B covering establishment, registration, and listing requirements, subpart C, donor eligibility, which my colleague Safa Carandish will expertly cover in part 2 of our session, subpart D, current good tissue practice requirements, plus subpart E that provides deviation and adverse reaction reporting requirements, and subpart F that includes inspection, importation, and enforcement actions. Take note that subparts C and D also apply to HCTPs regulated as drugs, devices, and/or biologics. HCTPs are articles containing or consisting of human cells or tissues intended for implantation, transplantation, infusion, or transfer to a human recipient. And they can be grouped further according to their basic function in the body as structural or non-structural tissues. Examples of both types are listed here in the column on the left. The column on the right provides examples of what an HCTP is not, such as vascularized human organs for transplant, blood vessels recovered with an organ transplant that are intended for use in organ transplantation, whole blood or blood components, and secreted or extracted human products such as milk, collagen, and cell factors. The table shown offers more examples. For an HCTP to be regulated solely under Section 361 of the PHS Act, the HCTP must meet all the following criteria, which are codified in 21 CFR Part 1271-10A. They must be minimally manipulated, intended for homologous use only as indicated by the manufacturer's objective intent. They cannot be combined with another article with some exceptions, and either the HCTP does not have a systemic effect and is not dependent upon the metabolic activity of living cells for its primary function, or the HCTP has a systemic effect or is dependent upon the metabolic activity of living cells for its primary function, and it's for autologous first or secondary blood relative or reproductive use. A product's level of risk determines the level of applicable regulations in the tiered risk-based approach. The agency wrote the tissue rules broadly to allow comprehensive regulatory oversight of the diverse HCTP industry. If all the criteria in 1271-10A are met, the HCTP is appropriately regulated solely under the authority of Section 361 of the PHS Act. The regulations in 21 CFR Part 1271 are required, and pre-market review and approval is not a requirement. If any of the criteria in 1271-10A are not met, the HCTP is regulated as a drug, device, or biological product under the authority of Section 361 and Section 351 of the PHS Act and or the FD&C Act and applicable regulations. In this case, pre-market review and approval is required. If an HCTP is appropriately regulated as a drug, device, or biological product, we recommend reviewing the CGTP final guidance with specific attention to Section 3 at Listing E. It provides a detailed answer to the question whether CGTP requirements must be followed for such a product and outlines where current good manufacturing practice and quality system regulation supplement the CGTP requirements. It also provides which CGTP requirements would not be partly or completely covered by a corresponding CGMP or QS regulation requiring the same practice. There are limited situations in which certain entities are not required to comply with the regulations in Part 1271. For example, 1271-15B describes an exception for an establishment that removes HCTPs from an individual and implants such HCTPs into the same individual during the same surgical procedure. More on the meaning of such HCTPs on a slide to follow. To clarify the same surgical procedure exception, draft guidance was issued in 2014 and finalized in 2017. It not only describes the relationship between the exception in 1271-15B and the four criteria in 1271-10A, but also the types of procedures generally considered to be the same surgical procedure, and what processing steps meet the exception. For the exception to apply, an establishment must remove and implant the HCTP into the same individual from whom they were removed. The HCTP must be implanted within the same surgical procedure, and it must remain such HCTP, which means that, generally, the only processing steps are rinsing, cleansing, sizing, and shaping. This exception is intended to be narrow. Let's review two definitions in 1271.3. The regulations define minimal manipulation for structural tissue and for cells or non-structural tissue, and this relates to how the HCTP functioned in the donor. For structural tissue, MM is processing that does not alter the original relevant characteristics of the tissue relating to the tissue's utility for reconstruction, repair, or replacement. And for cells or non-structural tissues, processing that does not alter the relevant biological characteristics of the cells or tissues. The regulations define that homologous use means the repair, reconstruction, replacement, or supplementation of a recipient's cells or tissues with an HCTP that performs the same basic function or functions in the recipient as in the donor. To clarify for industry how to apply the MM and HU criteria to cell and tissue products, final guidance was published in late 2017 and updated in mid-2020. It was issued jointly by CBER with CDRH as well as the Office of Combination Products. It includes many examples that can be used to determine if an HCTP is minimally manipulated and intended for homologous use, and it provided information on FDA's compliance and enforcement policy. A flowchart illustrates how to apply the same surgical procedure exception and how to apply the four criteria at 1271-10A. The first step on the flowchart is to determine if your product meets the definition of an HCTP. If it does, determine if the same surgical procedure exception applies. If that exception does not apply, then assess whether the four criteria at 1271-10A are all met. The answer to that assessment determines the appropriate regulatory pathway for your HCTP. The final guidance also explains that to give manufacturers time to determine if they need to submit an investigational new drug application or marketing application, and if such an application is needed, to prepare the IND or marketing application, the guidance describes a period of enforcement discretion for products based on a determination of the risk to public health. The last day of the period of enforcement discretion was May 31st, 2021. A product that requires but lacks pre-market approval may not be lawfully marketed, including when a sponsor has an IND or is pursuing a biological license application or BLA. These are the options for obtaining regulatory classification for an HCTP. An informal recommendation can be obtained by submitting to the tissue reference group, or a pre-RFD process is available. To obtain a formal decision on the regulatory assessment of an HCTP, a request for designation, or RFD, is the option to use. FDA provided another option for industry, the TRG Rapid Inquiry Program, or TRIP, that ran a course of 21 months and 19 days. It was a temporary program that provided a preliminary, informal, non-binding assessment from FDA's TRG as to how an HCTP appears to be appropriately regulated. Resources permitting, FDA responded as soon as possible to submissions. The TRIP began in mid-June 2019 and was extended 3 times, then ended on March 31, 2021. There was a total of 242 inquiries. The first quarter of 2021's total was 70, making it much busier than all of 2020, and at that pace would have surpassed 2019's numbers, covering 6 months. The results of these informal assessments were that more than half of the products submitted appeared to be appropriately regulated as a drug, device, or biological product. Okay, time for a knowledge check. This is the first one. True or false? History has shown us that communicable disease agents and diseases transmitted through use of HCTPs have been limited to viruses such as HIV, HBV, or HCV that were derived from the donor. Okay, the answer is false. History has shown that transmission of disease from use of HCTPs has also been attributed to prion-associated disease from the donor, as well as bacterial contamination that was unrecognized or that occurred during manufacturing steps such as during processing. Knowledge check 2. Which options below are available to manufacturers to obtain from FDA a regulatory classification for their HCTP? A request for designation, or RFD, a pre-RFD, or an option is the tissue reference group, the TRG. All 3, of course, are available options. Resources are available to you at these links to access the regulations. The Tissue and Tissue Products homepage is also listed here, where the tissue-related Federal Register notices, proposed, and final rules can be found along with guidance documents. A link is provided here to a website for the biologics industry hosted by the Office of Communications, Outreach, and Development. And the second listing is to the Tissue Establishment Registration webpage aimed at establishments that manufacture HCTPs regulated solely under Section 361 of the PHS Act that are required to register and list their HCTPs with FDA pursuant to 21 CFR Part 1271. The language in the blue box is taken directly from the regulations at 1271.27b. Product designation as a 361 HCTP is a self-assessment, so it should be clearly understood that FDA acceptance of an establishment registration and HCTP listing does not constitute a determination that an establishment is in compliance with applicable rules and regulations, or that the HCTP listed is licensed or approved by FDA. The last resource I'll provide is a link to where you can subscribe to podcasts and news feeds from FDA. I cannot stress enough that it's wise to subscribe so you are informed when new information becomes available. In summary, use of HCTPs has led to risks to public health due to transmissions of disease and FDA used a tiered risk-based approach when promulgating regulations to prevent the introduction, transmission, or spread of communicable diseases by HCTPs. To understand how an HCTP should be appropriately regulated, refer to criteria and definitions in the 21 CFR Part 1271 regulations that pertain to how the product is manufactured and how it is intended to be used. Thank you for your kind attention. Be sure to attend Part 2 of this session.
Good afternoon. My name is Safa Karandish. I'm a Consumer Safety Officer in the Division of Human Tissues in OTAT. I appreciate this opportunity to provide you with some additional information about the 1271 regulations. To enhance product safety, it is important to ensure donors of cells and tissues don't have infectious diseases that can be transmitted to recipients. The regulation for donor eligibility specifies the necessary measures for achieving this goal. At the completion of this presentation, you should be able to describe the purpose and applicability of HCTP donor eligibility or DE rule in 21 CFR Part 1271 Subpart C, describe the requirements for donor screening, donor testing, and making a DE determination, and also identify tips for submitting a request for an exemption or alternative if these requirements are not met. So let's begin with the purpose and applicability of the DE rule. In the previous presentation, Scott Brubaker described the regulatory framework for all HCTPs. The regulations in Part 1271, also known as the Tissue Rule, mainly focus on prevention of communicable diseases. Subpart C, which includes the donor eligibility requirements, is a major part of the Tissue Rule. The purpose of the DE rule is to increase the safety of HCTPs and public confidence in their safety by preventing the introduction, transmission, and spread of communicable diseases. The DE rule applies to all HCTPs recovered on or after May 25, 2005. This means these requirements apply to HCTPs regulated solely under Section 361 of the Public Health Service, or PHS Act, and subject to 1271 regulations. These are the so-called 361 HCTPs that are not subject to pre-market review requirements. The DE requirements also apply to HCTPs regulated as drugs, devices, and/or biological products under Section 351 of the PHS Act and/or the Food, Drug, and Cosmetic Act, and subject to regulations in Part 1271 and pre-market review requirements. Now I will describe the requirements for making a donor eligibility or DE determination. What is DE determination? A DE determination is based on screening and testing of HCTP donors for relevant communicable disease agents and diseases, or RCDADs for short. A DE determination is required for all donors of HCTPs, but the regulation provides some exceptions. An HCTP must not be implanted, transplanted, infused, or transferred until the donor has been determined to be eligible. Again, some limited exceptions are provided in the regulation. What are relevant communicable disease agents or diseases, or RCDADs? There are two groups of RCDADs. Some are specifically listed in the regulation, and others that meet the definition of RCDAD as described in the regulation are notified through guidance documents. On this slide, you see the current list of RCDADs for which donor screening and/or testing are required. For all cells and tissues, RCDADs include HIV types 1 and 2, hepatitis B and hepatitis C virus, human transmissible spongiform encephalopathy, or TSE, including CJD and variant CJD, Treponema pallidum, vaccinia, sepsis, West Nile virus, and Zika virus. For viable leukocyte-rich cells and tissues, RCDADs also include HDLV1 and 2. Additionally, for reproductive cells and tissues, Chlamydia trachomatis and Neisseria gonorrhea are additional RCDADs. This list may get expanded as new infectious diseases are identified and determined to meet the definition of RCDAD for donors of HCTPs. When is a donor eligible? In order for a donor to be considered eligible, donor screening must indicate that the donor is free from risk factors for and clinical evidence of infection due to relevant communicable disease agents and diseases, and the donor is free from communicable disease risks associated with xenotransplantation. Additionally, donor testing results for relevant communicable disease agents must be negative or non-reactive. However, an exception is provided for tests for syphilis. Once a DE determination has been made, certain information and records must accompany the HCTP at all times. A distinct identification code must be affixed to the HCTP container, and this code must relate the HCTP to the donor and to all records pertaining to the HCTP. The accompanying records must include a statement whether the donor has been determined eligible or ineligible, and a summary of the records used to make the DE determination. The information that must be included in the summary of records is specified in the regulation. Earlier, I mentioned the regulation provides some exceptions for DE determination. Specifically, donor screening, donor testing, and making a DE determination are not required for cells and tissues that are for autologous use or for certain reproductive cells and tissues. Even though DE determination is not required for these HCTPs, there are special labeling requirements. The 1271 regulation also addresses some limited situations when products from ineligible donors may be used. Specifically, an HCTP from an ineligible donor may be used if the HCTP is for allogeneic use in a first or second degree blood relative, reproductive cells and tissues from a directed reproductive donor, or if there is a documented urgent medical need as defined in the regulation. There are special labeling and physician notification requirements when products from ineligible donors are made available for use. I explained that DE determination is based on donor screening and donor testing, so I will now go over the donor screening requirements. To screen an HCTP donor, you must review donor's relevant medical records for risk factors for and clinical evidence of infection due to RCDADs and communicable disease risks associated with xenotransplantation. The regulation specifies that a donor must be determined ineligible if a risk factor or evidence of any RCDAD or risks associated with xenotransplantation is identified. What are relevant medical records? Relevant medical records, as defined in the regulation, is a collection of documents. It includes a current donor medical history interview, a current report of the physical assessment or examination, and other available records such as additional laboratory test results, medical records, autopsy reports in the case of a cadaveric donor, and relevant information from other sources. To conduct a donor medical history interview, you should ask questions about the donor's medical history and relevant social behavior considered to increase the donor's relevant communicable disease risks. You must also review the relevant medical records for clinical evidence of RCDADs and the records of the physical assessment or examination, looking for any sign that may indicate high-risk behavior or infection with the RCDAD. The 2007 Donor Eligibility Guidance document includes a list of conditions and high-risk behaviors, and also examples of clinical and physical evidence of RCDADs that should be considered for screening a donor. As shown on this table, all donors must be screened for HIV, HBV, HCV, TSE, T. pallidum, West Nile virus, Zika virus, sepsis, and vaccinia. Donors of viable leukocyte-rich cells or tissues must also be screened for HDLV. Additionally, donors of reproductive cells or tissue must be screened for chlamydia and gonorrhea. So what are the donor testing requirements? The specimens for donor testing must be collected at the time of recovery or within 7 days before or after recovery of HCTPs. An exception is provided for donors of oocytes, peripheral blood stem cells, and certain bone marrow donors. In these cases, the donor's specimen for testing may be collected up to 30 days before recovery. Donor testing must be performed using appropriate FDA-licensed, approved, or cleared donor screening tests following the manufacturer's instructions for use. The laboratory that performs the donor testing must be CLIA certified or meet the equivalent requirements as determined by the CMS, which manages the CLIA certification program. This table lists the infectious diseases for which donor testing is required. All donors must be tested for HIV types 1 and 2, HBV, HCV, and T. pallidum. Living donors of HCTPs must also be tested for West Nile virus during the time period described in the guidance for West Nile virus testing. Donors of viable leukocyte-rich HCTPs must also be tested for HDLV1 and HDLV2, as well as CMV. It's important to point out that CMV is not considered the RCDAD. Therefore, the CMV test results don't affect the donor eligibility determination. However, in some settings, the CMV status of the donor may be clinically relevant. Testing for chlamydia and gonorrhea are additional requirements for donors of reproductive cells and tissues. For the required testing, more than one test may be necessary to adequately and appropriately test for a single disease agent or disease. On this slide, you see the current list of tests that FDA considers to be appropriate and adequate. You can find this information in the donor eligibility guidance and other related guidance documents, but this summary table may be helpful as a quick reference. But please keep in mind the list of recommended tests may change over time, so it is always important to refer to the latest relevant guidance documents. Earlier, I explained the donor testing must be performed using FDA-licensed, approved, or cleared tests. You can find the list of these tests on the 2 FDA websites listed on this slide. These lists get updated periodically, so it would be good to check these websites for any updated information. In the remaining time, I will review the process for requesting an exemption or alternative. The regulation in Part 1271 provides a mechanism for requesting an exemption from or an alternative to any donor eligibility or current good tissue practice requirements. The request must be submitted to the OTAT office director, and it must include information justifying the requested exemption or a proposed alternative method for meeting the requirement. An establishment or sponsor must not begin operating under the terms of a requested exemption or alternative until it has been granted. FDA would consider granting a request for an exemption or alternative if the action is consistent with the goals of protecting the public health and the submitted information justifies the request. More information about these requests is available on the FDA's Exemptions and Alternatives website. To submit a request for an exemption or alternative for HCTPs regulated as drugs, devices, and/or biological products, we encourage sponsors to consider several points. It is helpful to discuss the request during early communications with the FDA, for example, during the Interact or pre-IND meeting. It is also helpful if the request is submitted prior to submitting the application to ensure there is adequate time for FDA to review the information and request any additional information that may be needed. Please note an application may be placed on hold if regulations are not met and the request for an exemption or alternative has not yet been granted. In the request, include supporting documentation and relevant valid scientific data regarding how risk of communicable disease transmission is mitigated. It is helpful to provide a description of the manufacturing process, a description of the process for donor screening, donor testing, and donor eligibility determination, as well as information on any in-process and final product testing that is performed during manufacturing to mitigate the risk of communicable disease transmission. Now that you learned about the donor eligibility requirements, we will go over a couple of knowledge check questions. Donor eligibility requirements are applicable to option 1, HCTPs regulated solely under Section 361 of the PHS Act or 361 HCTPs. Option 2, HCTPs regulated as drugs, devices, and/or biological products under Section 351 of the PHS Act and/or the FD&C Act. Or option 3, which includes HCTPs described in options 1 and 2. I will pause for a few seconds so you can think about your answer. Option 3 is the correct choice because the DE requirements apply to all HCTPs regardless of how they are regulated. An HCTP donor eligibility determination is based on the results of option 1, donor screening only, option 2, donor testing only, or option 3, donor screening and donor testing. Again, I will pause for a few seconds before revealing the answer. Hopefully, all of you think option 3 is correct. The DE determination is made based on the results of donor screening and donor testing. I was not able to review all the requirements in this presentation, so I encourage you to refer to the 1271 regulation and related guidance documents for more information. And here are the links to those relevant documents. So to summarize, donor eligibility requirements are applicable to all HCTPs, regardless of how they're regulated. Donor screening and testing for RCDADs are required for making a DE determination. You may consider submitting a request for an exemption or alternative if the DE or CGTP requirements are not met. Here is my contact information. I've also included contact information for submitting questions to the FDA. Thank you for your time, and I hope you found this information helpful. I will now turn it over to our moderator for the question and answer session.
Thank you, Scott and Safa, for these two very informative talks. Really excellent overview of the history as well as the contents of the tissue rules and the approach to regulation of HCTPs. I find that this is one of the areas of biological product development and regulation that is not known as well as, for example, a regulation of drugs and other biological products. But I see a number of questions here, and we will proceed with the Q&A part. Let's start with Scott. So here's a question for Scott. If a particular product has a Centers for Medicare and Medicaid code, or CMS code, does this mean I can use the product? Does this mean that the product is legally marketed?
Yeah, thank you for the question. Please be aware that the availability of a CMS code for a particular HCTP or other biologically derived product, such as amniotic fluid, does not mean that product is appropriately regulated solely under Section 361 of the Public Health Service Act and the FDA regulations in 21 CFR Part 1271. The availability of a CMS code also does not mean that a particular HCTP is lawfully marketed.
Thank you, Scott. The next question goes to Safa. Is an exemption request necessary if we want to pool cells from multiple donors during manufacture of our cellular therapy products?
Thank you for the question. This question refers to one of the current good tissue practice or CGTP requirements that we didn't cover during our presentations, but the answer to the question is yes. According to the CGTPs in the 1271 regulations, pooling cells or tissues from 2 or more donors during manufacture is prohibited due to concerns that pooling increases risk of communicable disease transmission such as TSE. You may submit a request for exemption from the pooling regulation specified under 1271.220b. As I explained during the presentation, the request must include justification, supporting documentation, and relevant scientific data regarding how risk of communicable disease transmission will be mitigated. For investigational products, if you plan to pool cells or tissues from multiple donors during manufacture, discuss it with FDA as early as possible, for example, during Interact or pre-IND meetings. Again, thank you for the question.
Thank you. Now, another question for Scott. What could happen if I continue to market or offer for sale an HCTP that requires but lacks pre-market approval as a drug or biological product?
No, as described in our compliance and enforcement policy regarding certain regulatory requirements, FDA no longer intends to exercise enforcement discretion with respect to the IND and the pre-market approval requirements for certain HCTPs. So if you market or offer for sale an HCTP that requires pre-market review as a drug and biological product, you do so at your own risk.
Thank you. And as a follow-up question, I guess from the same channel, if I submit an IND for my HCTP, may I continue to market the product or offer it for sale? And IND is the investigational new drug application, as I understand.
Okay, yeah, a product that requires but lacks pre-market approval may not be lawfully marketed or offered for sale, including when a sponsor has an IND or is pursuing an IND or BLA.
Thank you. So the next question goes to Safa. Where can we find a list of CLIA certified testing laboratories? And CLIA stands for Clinical Laboratory Improvement Amendments. So where can we find a list of CLIA certified testing laboratories outside of the United States?
This is a common question we get from manufacturers outside the U.S. The Centers for Medicare and Medicaid Services, or CMS, is responsible for the CLIA certification program. For information on CLIA certified laboratories outside the U.S., you may contact CMS. The contact email address is clia-iointake@cms.hhs.gov. You can find this email address and additional information on the CMS CLIA website. I just also want to point out that donor testing laboratories outside the U.S. generally do not have the appropriate CLIA certification or access to FDA licensed, approved, or cleared donor screening tests. Establishments outside the U.S. that manufacture and import HCTPs into the U.S. may wish to consider sending donor specimens to an appropriate laboratory in the U.S. for testing. Thank you for the question.
Thanks, Safa. And another question for you here. The agency recently withdrew the guidance on Zika virus for donors of blood and blood components. Does the FDA plan to reevaluate Zika virus screening for donors of HCTPs?
For the purpose of screening donors of HCTPs, you should continue to use the May 2018 guidance titled Donor Screening Recommendations to reduce the risk of transmission of Zika virus by human cells, tissues, and cellular and tissue-based products, and also refer to the February 28, 2019 communication titled Important Information for HCTP Establishments Regarding Zika Virus Transmission Risk in the World, posted on FDA's Biologics Safety and Availability website. FDA is committed to reevaluation and reconsideration of its policies regarding donor eligibility determination for donors of HCTPs. Should there be any changes to HCTP donor screening or testing recommendations, they will be communicated to industry through guidance. Thank you for that question.
Thank you, Safa. Another question for Scott here. I administered an HCTP to a patient with a good response prior to May 31st, 2021. However, the product requires but lacks pre-marketing approval. Can I continue to administer this product to the same patient?
Good question. The answer is no. A product, again, that requires but lacks pre-market approval may not be lawfully marketed. So you may not administer an unapproved product without an approval or an IND being in effect.
Okay, then as a follow-up question, if an IND is in effect, can I contact FDA to confirm that this is true and that the product has an IND in effect?
Yeah, I can't, I wasn't ready for that one. Can you move to another one? Sorry.
Well, let me see. So here I have a question for Safa. What are the donor eligibility requirements for a cellular therapy product manufactured using a cell line that was generated before 2005?
That's a good question. During the presentation, I mentioned that the 1271 regulations became effective on May 25, 2005. If the cellular starting materials were recovered before May 25, 2005, they're not subject to the regulations in 21 CFR Part 1271. Therefore, the donor eligibility requirements in the 1271 regulations are not applicable. However, in the IND application, the sponsor will provide sufficient information to demonstrate that the cell line is safe and acceptable to use as a starting material to manufacture the cell therapy product. This information should include sufficient information about the donor, including pertinent donor screening and donor testing that has been performed to mitigate infectious disease risks, as well as any in-process and final product testing for adventitious agents. Sponsors are encouraged to discuss these types of questions during the Interact or pre-IND meetings with FDA. Thank you for the question.
Thank you, Safa. Scott, I'm back to you with that question, and maybe I'll rephrase it. So the question is about somebody administering an HCDP, which requires but lacks pre-market approval to patients continuously, and the manufacturer of that HCDP informed them that there is an IND in effect for the product. And so the question here is whether they could contact FDA to confirm that the IND is in effect for the product in order to continue giving it to people.
Right. Okay. Yes, I understand. Under FDA's regulations, the agency cannot disclose the existence of an IND. You can confirm whether a product has an IND in effect by obtaining from the sponsor the FDA-issued IND number, as well as a copy of the official FDA communication that acknowledged the IND. But please, though, keep in mind, however, that an IND is not marketing authorization for a product. Rather, when an IND goes into effect, the product sponsor can ship an investigational new drug and unlicensed biological product to clinical investigators without an approved marketing application, like a BLA. And so if you're receiving a product that has an IND in effect, then you would be considered a clinical investigator associated with the IND.
Thank you, Scott. I hope this was helpful to those who asked. So I think we're at the point where we will be now concluding our session, and we will go on a short break, after which Leish Mini, who is our next moderator, will introduce our next speaker and will proceed to the next session. Thanks, everyone, and thanks, Scott and Safa.
Good afternoon, everyone. Welcome back from break. My name is Leash Manet. I'm the acting branch chief for Division of Regulatory Project Manager within the Office of Tissues and Advanced Therapies. It's with my great pleasure I introduced Jennifer Albert, who is also a project manager. Jennifer had been with FDA since February of 2020. In her capacity as a regular manager, she manages investigational new drug application, device submission, and formal meeting with industry. Ms. Albert holds a Bachelor of Science in Nursing from Cellular University. She's a registered nurse and has experience in critical care and oncology. Prior to working at FDA, she worked at Clinical Center of National Institute of Health for 11 years. She has certification in oncology nursing and bone marrow transplantation. In 2012, she joined the National Center Institute, NCI, as a clinical nurse specialist, where she was responsible for managing phase 1 and 2 clinical trials involving stem cell transplantation, immunotherapies for hematology malignancies, multiple myeloma, and metastatic solid tumors. She was also involved in the design, implementation, and evaluation of clinical research protocols and managed the implementation of multiple trials in the patient population studied. She also directed and performed quality assurance and quality control activities as they relate to protocol adherence, monitoring data collection and abstraction, and data analysis. Today, she'll be presenting on developmental of 351 biological products. Her presentation will cover the basics of biological submission, review the life cycle of biological products from investigational new drug IND to biological licensing application BLA, and familiarize the audience with FDA and sponsor meeting procedures for development of biologics. At the end of her presentation, we will have a Q&A session. With that, I'll pass on the floor to Jennifer.
Hello, everyone. I'm Jen Albert. I am a regulatory project manager with the Office of Tissues and Advanced Therapies, OTAT, which is within FDA CBER. Today, I will be presenting the basic overview of product development for 351 biological products. A short disclaimer. All my comments are an informal communication and represent my own best judgment. These comments do not bind or obligate the FDA. Our learning objectives are to recognize the role of FDA CBER OTAT, describe what is a 351 biological product, identify the structure of an IND and BLA submission, select the appropriate PDUFA meeting types for milestones in development, and identify resources available for industry. The overall goal is to give you basic regulatory guidance that could save you time and resources when working with CBER OTAT. Let's start with the who and the where in which FDA comes into play. As you are all aware, FDA is a centralized federal agency within the U.S. Department of Health and Human Services. It is tasked to oversee the entire life cycle of a medical product, from investigational product development to post-marketing surveillance and studies. The FDA's ultimate mission is to protect public health. The Center of Biologics Evaluation and Research, or CBER, is FDA's primary biological product review center. The Office of Tissues and Advanced Therapies, OTAT, is one of 3 product review offices within CBER. And this is where you can find me. CBER regulates many complex and diverse products, including vaccines, allergenic extracts, blood and blood products, tissues, cellular and gene therapies, as well as selected devices. An important exception to note is that CDER regulates most peptides, proteins that are less than 40 amino acid chains, and therapeutic toxins. On this very busy slide shows more specifically the products my office, OTAT, regulates. It is an understatement to say this covers a lot. onwards we move to why all this regulation is in place. These requirements are set by FDA under statutory authority from Congress. These policies and guidelines are published in their Federal Registrar. Here is a slide showing a quick summary of the U.S. paradigm for medical product regulation. Brief reminder that statutes are laws passed by Congress and signed by the President. Regulations give the details of the law, which can be written by FDA and approved by the executive branch. And guidance are FDA's interpretation of the regulations. These are often written and approved within FDA and are advice that is non-binding. Now let's talk about what is a 351 biological product. I hope my previous slides gave you a hint. 351 biological products are referring to the FDA regulatory requirements as defined in Section 351 of the Public Health Service Act of 1944 for the distribution of biological drug products in the United States, aka PHS Act, thus giving the FDA authority to regulate and approve new biological products. By definition, a biologic or biological products can be made of sugars, proteins, or nucleic acids, or complex combinations of these substances. These products may be produced through biotechnology in a living system, such as a microorganism, plant cell, animal cell, or tissues, and are often more difficult to characterize than small molecule drugs. This brings us back to the dreaded regulations. Biological products are dually regulated under the PHS Act and Federal Food, Drug, and Cosmetic Act, FD&C Act. This means that most biological products are approved for marketing under the PHS Act, but here's the kicker. They must also meet the definition of drug and therefore must also comply with Title 21 of the Code of Federal Regulations, CFR. The regulations for biologics can be found under 21 CFR 600. On a side note, as stated earlier, CBER does regulate some devices, and of course, these are not drugs. Device regulations can be found under 21 CFR 800. CBER regulates medical devices and tests used to safeguard blood, blood components, and cellular products from HIV, hepatitis, syphilis, and other infectious agents. In addition to reagents used to type blood, this covers machines and related software used to collect blood and blood components. There are two common regulatory pathways for biologics. The first is by a biologics license application, BLA, and the second is if an investigational new drug, IND, is in effect. Now, I only have a few moments with you, so I'll focus on these two, IND and BLA pathways. Unfortunately, I will not be able to cover other pathways such as the biosimilars license application, in addition to IDEs, 510Ks, de novo, pre-market approval applications, and humanitarian device exceptions. Okay, the life cycle approach. We've heard it and seen it many times. Now let's apply this to 351 biological products. Here is a colorful picture of the regulatory and product life cycle. As you can see, the biological product and the regulatory life cycle is very similar to that of CDER's drug side, including the following regulatory stages: discovery preclinical or pre-IND phase, clinical development or IND phase, and then the final marketing phase, which at CBER includes the Biologics License Application, BLA 351As, and Biosimilars Biologics License Application, abbreviated as BLA 351Ks. As stated earlier, due to our limited time together, we will focus on the clinical development, or IND phase, and the ultimate stamp of approval, the BLA process. Please note that most biological submissions fall under specific regulatory mandated timelines of the Prescription Drug User Fee Act, PDUFA, And at the time of this talk, we are in PDUFA version 6. For all those newbies, PDUFA is a commitment between the FDA and sponsors to maintain a review timeline. The fees collected under PDUFA support the program. The goal of the program is to improve the efficiency and effectiveness of the first cycle review process. Through improved communication and even greater transparency, the hope is to decrease the number of review cycles necessary for a biologic to be approved. PDUFA will apply to almost everything I am covering today. Now let's review the IND process in the context of biologics. Most of you are familiar with CDER's INDs, and in general, the same applies for biologics. An IND is a formal living document with a defined structure and content. Its purpose is to request an exemption from pre-marketing requirements and to allow lawful shipment of a biologic across state lines, specifically for clinical investigation. It is during this IND phase where clinical investigations of an unapproved biologic may be conducted. As a reminder, the FDA regulates U.S.-based clinical investigations to ensure subjects' safety and rights throughout the clinical trial. As a sponsor, you will typically have your Phase 1 studies that assesses the product's safety, as well as aspects such as tolerability and maximum tolerated dose. The data collected then guides dosing and monitoring for the next Phase 2 trial. When you initiate Phase 2, these studies involve more patients and primarily assess the magnitude of effect using defined endpoints. This data in turn guides the design of the subsequent confirmatory Phase 3 trials. Finally, Phase 3 trials, which typically involve many more patients, collect more data, thus gather more evidence of safety and effectiveness on a much larger scale to support a marketing application. The FDA's role in all of this is focused on safety, and during Phases 2 and 3, there's an additional concern to ensure that the scientific evaluation's quality is adequate to permit the evaluation of the biological product's safety and effectiveness. Again, just like CDER's INDs for drugs, the IND requirements for biologics are found in 21 CFR 312. You will notice the requirements are the same when submitting an IND to CBER OTAT. Here on this slide is a helpful link to the CBER-specific webpage for INDs. An ideal CBER OTAT IND submission process can be broken down into 3 parts. Preclinical, where interact or pre-IND meetings are highly recommended for new products, new dosage forms, new routes of administration, and new indications. At CBER OTAT, you could get 1 interact and 1 pre-IND meeting at the preclinical stage. If granted, that's a possible 2 extra preclinical meetings in which you can obtain preliminary consultations for your biological product. To keep you listening, I'll cover interacts later, as the scope of each of these preclinical meetings are different. Say, next, you're a pro, and you don't need an interact or a pre-IND meeting, and you are ready to submit a complete IND package for review at CBER OTAT. Please note that the Electronic Common Technical Document, ECTD, format is required for commercial INDs. Paper or PDF submissions are allowed for research INDs. Note, due to the current public health crisis, normal document processes have been changed, and we are not receiving any mailed-in versions, but allowing PDF packages sent to CBER's DCC email linked here. Once received, the initial 30-day review period begins. These are calendar days. During this time, a multidisciplinary review team completes a review for safety, with the goal to ensure that research subjects will not be subjected to unreasonable risk. Regulatory actions and the status of INDs after this initial period can be active, clinical hold, or partial hold, where active means that the IND may proceed, and an OTAT, you will be notified by email. In the event of a clinical hold, the IND may not proceed, and a formal letter is issued within 30 calendar days of the telephone or email notification. If your IND is placed on partial hold, this is a delay or suspension of part of the clinical study under an IND. An example of this would be where the IND has 2 protocols and only one can proceed and the other one is on hold. Just as with the clinical hold letter, a formal partial hold letter is issued within 30 calendar days of the telephone and/or email notification. What happens during your clinical development has huge impact downstream, so I want to encourage certain interactions and meetings with you. While information requests IRs will be sent via email during the initial and for future reviews of the IND amendments, your requests for meetings can frequently be useful in resolving questions and issues raised during your clinical investigation that can impact the content of your BLA. Possible CBER OTAT meetings during the IND phase are listed here, and these are requested via formal amendments to the IND file. Let's say you breezed through the phases of clinical development, and now you are ready to submit a BLA. A BLA is a request for permission to introduce or deliver for introduction a biological product into interstate commerce. A BLA is submitted by an applicant and must contain data derived from non-clinical laboratory and clinical studies, which demonstrate that the manufactured product meets the prescribed requirements of safety, purity, and potency. BLAs are regulated under 21 CFR 600 to 680 and under Section 351 of the PHS Act. A BLA is submitted by a legal person or entity who is engaged in manufacturing or takes responsibility for compliance with the product and the establishment's standards. Applicants are required to submit their BLA application and subsequent information through the FDA's central transmission point called the Electronic Submissions Gateway, often abbreviated ESG or Gateway. And ECTD is the standard format for submitting applications, amendments, supplements, and reports to CBER. Please note, the reference triangle here is a basic breakdown of the ECTD modules. RPM negative advice. Please do not forget your Form 356H, because not only is it necessary, it will help guide you on the requirements needed for the review of your application. An incomplete BLA submission, including failure to provide agreed upon information within 30 days of receipt of the BLA, will be subjected to a refuse to file decision, also known as RTF. Here is a simple graphic showing the typical regulatory review lifecycle of an original BLA. An original BLA at CBER OTAT typically follows PDUFA and is highly recommended to begin with a pre-BLA meeting. This pre-BLA meeting is helpful for filing determination and designations and sets some expectations regarding review planning. The actual review of a BLA begins upon acceptance by CBER, not receipt date. This decision occurs no later than 60 calendar days from data receipt and only after receipt of all components of your BLA. Your filing letter will be a formal letter, either informing you of the acceptance of the BLA file, titled Filing Notification Letter, or Notification of Refuse to File, RTF. Side note, if a rolling review has been granted, all portions of the application should be submitted no more than 1 year after the initial portion. Any review of the portions already submitted may be suspended because our ability to conduct a meaningful review may be inhibited without the missing information. Now back to the BLA lifecycle. Once your original BLA is accepted, the review occurs either in standard review time, which is 10 months after the 60-day filing date, or if your BLA has priority review, that is 6 months after the 60-day filing date. After doing some math, that's about 12 or 8 months total time for each review timeline, respectively. Okay, so I mentioned priority review, and your ears perked up. Who doesn't want to be labeled priority? Priority review is where your application gets to have a reduced review schedule, thus potentially allowing the product to reach the market faster. As described in the Guidance for Industry for Expedited Programs posted here, marketing applications for Fast Track, Breakthrough Therapy, and/or RMAT designated products may be eligible for priority, accelerated approval, or rolling review. Fear not. A colleague of mine will be presenting in detail on this subject in a separate session. Actions. Your BLA will have a multidisciplinary review team who will assess the application and then provide a recommendation to or oppose the biological product's approval. As mentioned, a filing action will be achieved by 60 days and has the possibility of an RTF, which is issued when the submission has been deemed insufficient for a meaningful review. But hopefully after this talk, you will get the filing notification letter that will include initial dates, preliminary plans, and any IRs, or may even say no deficiencies identified. Depending on if there are deficiencies, either of these formal filing action letters will be issued by 75 calendar days from receipt of your BLA. A complete response action, CR, is a letter issued after a complete review indicates that there are deficiencies remaining that preclude the approval of the application. A CR letter stops the review clock and within it itemizes all the deficiencies remaining and describes the actions necessary that must be corrected prior to approval. Once a CR letter is sent, a resubmission will need to be submitted. Resubmissions are classified into Class 1 or 2, where Class 1 resubmissions will be reviewed within 2 months' time and are for deficiencies that are identified as minor reanalysis of data, draft or final printed labeling issues, final release testing, stability updates to support provisional or final dating periods, commitments to perform Phase 4 studies, and reports on compliance status. A Class 2 resubmission has a 6-month review clock, and examples of Class 2s are major reanalysis or if significant additional clinical data is needed, if there is a need for re-inspections, and for any items that require presentation to an advisory committee, and even assay validations. I have included CBER's public SOPP for resubmissions so you can reference these classifications. Your resubmission is your reply to the CR letter, and if you have addressed all items, then the clock restarts. At any time, you can withdraw a BLA. Note that if you withdraw before the filing action date, 75% of your user fee will be refunded. Ultimately, you all want that green light approval letter. This is issued by the action goal date and marks when your biologic is approved for marketing. Hooray! To get to approval as an RPM with CBER OTAT, I expect many interactions with you and anticipate a few BLA-specific meetings. Interactions can start as early as the optional application orientation meeting, which is usually requested at the pre-BLA stage and held within 60 days of an anticipated receipt date of the BLA. Your BLA will have a mid-cycle communication from me to you, especially if there are additional negotiations needed. Mid-cycle communication telecons are held to discuss the status of the review. Any significant issues or deficiencies identified by the review team, any major safety concerns, any pending IRs, and other projected milestones for the remainder of the review cycle. Then, a late cycle meeting will have discussion of the progress of the review. Here, FDA will identify issues and recommend plans on how to address those issues. These could include possible dates for further deliverables and interactions. For this late cycle meeting, we will need to have the meeting materials 10 to 20 days prior, and depending on if an advisory committee meeting is planned. A meeting summary letter will be issued and will itemize what was discussed. At any time throughout the review, CBER OTAT will send information requests by email. Under this last section of Other are a few extra meetings that could also occur with your BLA. Advisory Committee meeting or AC meeting is product dependent and is typically determined near the BLA filing action date. Its purpose is to obtain independent expert non-binding advice and recommendations on scientific, technical, and policy matters. The public vetting for an AC meeting occurs so that non-FDA experts provide additional advice to the agency regarding the product's safety and efficacy for the proposed indication. At CBER OTAT, the default is to convene an AC meeting, since we interpret for all new molecular entities to include all new biologics. However, exceptions are made for biologics, such as when the product is not first in class or the application did not raise substantial public health questions regarding the role of the biologic in the diagnosis, cure, mitigation, treatment, or prevention of a disease. If scheduled, there is a lot of meeting preparation that goes into an AC meeting. For PDUFA products, depending on review timeline, they should be held no later than 2 months or 6 weeks prior to the user fee goal date. If scheduled, the FDA will provide final AC questions to you 2 days prior to the meeting. I highly recommend referencing this linked AC guide for industry because I know that was a lot of information to cover in a few minutes. We urge you to have a complete BLA submission from the get-go. However, during the review period, you can send in certain amendments. These can cover responses to IRs, proprietary name review requests, and pre-agreed additions to the BLA. If you submit one that is unsolicited by the FDA, it may not be reviewed. Short note on major amendments. An applicant does not choose to submit a major amendment, but it is at FDA's discretion to convert an amendment that is supplying significant data or major reanalysis to this classification. A major amendment may extend the BLA review time by 3 months and is limited to 1 extension per review cycle. Let's move to the types of interactions and meetings held for PDUFA biologics. The bottom line is that CBER is here to help, and we do this by facilitating meetings with you. Meetings between a sponsor and the FDA are frequently useful in resolving questions and issues raised during a product's development. We encourage meetings to the extent that they aid in the evaluation of the biologic and in the solution of scientific problems concerning the product and, of course, as resources permit. We encourage meetings even before starting a clinical investigation because, in some situations, unnecessary animal studies or additional testing may not have to be done, thus possibly saving sponsors time and valuable resources. Here is an easy-to-follow picture of the many opportunities to meet with CBER OTAT. I will highlight our specifics and give examples in the following slides. Interact is a CBER-only meeting that stands for Initial Targeted Engagement for regulatory advice on CBER products. It is a one-time informal consultation with us for when you are in the earliest stages of product development and need to address issues in advance of a future pre-IND meeting. Interacts were previously called pre-IND meeting. Interact is a benefit to sponsors to obtain initial non-binding advice from CBER. This meeting is helpful to assist sponsors in conducting early product characterization and preclinical proof of concept studies. It can initiate discussion for novel devices, provide early advice about early phase clinical trial design elements, and possibly identify critical issues or deficiencies. Before requesting an Interact meeting, you will need to have selected a specific investigational product or product derivation strategy to evaluate in a clinical study. As mentioned before, the scope of Interact is different than that of a pre-IND. We're asking for clinical recommendations regarding future first-in-human studies in a targeted population are within the scope of Interact, whereas new dosage forms may be out of scope. Per the CBER SOPP 8214, found in the link here, an additional scope difference is that routine questions regarding specific aspects of clinical study protocol design, such as inclusion and exclusion criteria, are within the scope of Interacts, while the review of clinical study designs or protocols occur in the context of a pre-IND meeting, thus are outside the scope of an Interact. Interact does have its own unique process, so please refer to this link for additional details regarding Interact, as it is not covered under PDUFA. Listed here are PDUFA meeting types you can request at CBER OTAT. Type A meetings are reserved for stalled product development due to an FDA action or when there has been a no agreement or non-concurrence of a special protocol assessment. CBER OTAT does not consider sponsored end stalled development such as a failed interim analysis as a Type A. In my experience, I have seen this granted in response to clinical hold, where the development has been stalled and a new path forward needs to be discussed. Type A meetings are held or responded to as written response in 30 days. If this type of meeting is granted, preliminary responses will be sent at least 2 days prior to the scheduled meeting date. The message here is that Type A meetings are unique and are reserved for critical issues. Please contact us prior to requesting a Type A meeting to make sure you fit this category. Common CBER OTAT Type B meetings are the ones listed here. These meetings are held or written responses sent by 60 days. If granted, preliminary responses will be sent at least 2 days prior to the meeting date. A pre-IND meeting is a non-binding, formal scientific discussion between you and our reviewers. The goal is to achieve a successful IND submission, and its purpose is to allow early communication about your product and or clinical development plan. I have seen pre-IND meetings requested for advice on product characterization issues, questions regarding a preclinical testing program, and questions regarding the scope and design of a planned clinical trial. Pre-BLA meetings are strongly encouraged to discuss the planned content of your BLA application with the appropriate review divisions. I suggest that you request your pre-BLA meeting 4 months before you plan to submit, so that your pre-BLA meeting can be held at least 2 months before the official receipt of your BLA. A special note regarding Breakthrough Therapy and RMAT designations. These allow you to request additional Type B meetings, but again, a colleague of mine will cover these in more detail. Additional special Type B meetings specific to end of phases are listed here. These will be held or responded by written response at 70 days from receipt date. I highlight end of phase 2 or pre-phase 3 meetings because it is important to know that your pediatric study plan, PSP, should be presented by this milestone meeting. An agreed PSP is an essential item that needs to be addressed with your BLA submission. All BLA submissions must include an agreed pediatric study plan or granted a waiver or deferral for which this applies to many biologics with orphan designation. This is to ensure that the data is being collected for biological products for which children ages 0 to 17 might benefit. Lastly, a Type C meeting with OTAT is any meeting other than what we just covered. They generally are discipline-specific and held or written responses sent by 75 days of meeting receipt. CBER OTAT has granted Type C meetings for questions related to CMC issues, facility meetings with DMPQ, or even clinical in nature, such as early consultation on a new surrogate endpoint. It is a CBER OTAT policy, not PDUFA, that Type C meetings regarding new surrogate endpoints will not be responded in written response. I know I swung a lot of numbered timelines, so to make it easy, I've provided the next slide. Here is a quick reference and summary table of the current PDUFA timeline goals. I always keep this pretty handy, so I can easily reference the number of calendar days from date of receipt when each decision, action, or communication is needed for each meeting type. Needless to say, but important to mention, are the meeting formats. You can request a meeting in any format. However, pending the initial request and final review of your meeting package, FDA may change the meeting format. When we aren't in a public health crisis, face-to-face meetings are typically granted for Type A into Phase 2 and pre-BLA meetings. Teleconferences are most often granted for Interact and pre-INDs, and written responses can be given for any meeting type if requested. Now, are you ready to submit your IND, BLA, or meeting request to CBER? If yes, here's how. Prior to the pandemic, requests came either postal mail or electronic. Now, no mailed submissions are being accepted, and electronic submissions are a must. ESG and the Gateway is the preferred CBER transmission site, but PDFs may be accepted. In some cases, especially when rapid response is needed, you may send a reply email to the IO requester directly. Note, this all should be sent with secure encrypted end-to-end email to protect your proprietary information. You will be asked to establish your email with FDA by sending a request to secureemail@fda.hhs.gov. I have linked a past webinar to help you with becoming a secure email partner. Just like CDER, at CBER, you can request a pre-assigned submission tracking number, STN, by emailing CBER RIMS. The email link and necessary information that you will need to provide are listed here. This concludes the content part of my presentation. And now, on to our challenge. Challenge question number 1. FDA regulates and approves biological products under the authority of A. 21 CFR 312, B. Section 351 of PHS Act, C. I don't know. The answer is B. The keyword here is approved. Section 351 of the PHS Act gives FDA the authority to regulate and approve new biological products. Challenge question number 2. Which regulatory pathway allows a new biological product to be exempt from restrictions on interstate shipment and administration? A. IND, B. BLA, C. I don't know. Answer is A. Only under an IND can a biologic be exempt from restrictions on interstate shipment and administration. These can be any new biological product that is not the subject of an approved BLA and allows an unapproved new biologic to be used in clinical investigations. A biological product is approved for marketing through the BLA process. Congratulations! We certainly covered a lot. I hope I have helped you understand the basics of CBER Biologics. I understand that this was a lot, so here are a few consolidated resources to refer to. And by a few, I do mean the next 6 slides. Here is slide 1. Here is slide 2. Here is slide 3. Here is slide 4. Here is slide 5. And here is slide 6. If you have any questions, please reach out. Email is fastest. I may not know all the answers, but I'm sure I can find someone who does. Thank you for your attention. I look forward to your future questions, applications, and meeting requests.
Thank you, Jennifer. That was an excellent presentation. You've provided a great overview of the life cycle of biological products, starting from IND, going to marketing application to BLA, including FDA sponsor meetings. With that, we'll start our Q&A sessions, and I'm going to read the questions, and then if you could help us answer and respond to those questions. The first question is, do you have to submit a CBER application through the Gateway? If not, to where and how do I submit my research application to CBER?
Thank you so much for that question. So no, you don't have to go through the gateway, especially if you are a research IND. You will need to go through the gateway if you are a commercial IND, a BLA, or phase 3 INDs that are essentially usually commercial. I will, and so yes, please submit electronically in a PDF format to CBER DCC email sub at fda.hhs.gov. And please submit using...
Could you please put your volume up? It's a bit softer.
How about now?
Oh, perfect. Thank you so much.
So sorry about that. So to go back, apologize. You do not, if you are a research IND, the ECTD and the gateway is really for all the commercial INDs, the BLA and the phase 3 INDs that are generally commercialized. But we do want to say it is preferred as it is our central location for our submissions. Now, if you are a research IND and do not want to go through the gateway, you can submit in a PDF format and email it to cberdcc _email sub at fda.hhs.gov. Please do use the ECDT format if possible, and we do note that we will be accepting submissions through this email option only because we are in a public health crisis on this emergency. Thank you so much. Sorry about the audio.
No problem at all. Thank you again. Question number 2. What do I need to do to include a My Meeting request?
Great. The meeting request, please refer to slide 30 of my, my presentation. And I refer you to the formal meetings between FDA and sponsors or applicants of PDUFA products, the guidance for industry, and also the best practices for communication between IND sponsors and FDA during drug development. Again, this is another guidance for industry. There is one more, CBER 8101.1 SOPP. This is a regulatory meetings with sponsors and applicants for biological products. These are available to the public. To be a little bit more elaborate with OTAT, we would like a number of things, and we will send this when we grant your meeting. The number of questions need to be clear, targeted, and numbered. No more than 12 questions is a good roundabout number. The size of your meeting package should have relevant background and information in relation to the questions in your package. Voluminous meeting packages are discouraged, so ideally packages should probably be no more than 250, 300 pages. If your meeting is granted, the agency or us, OTAT, will provide you with preliminary responses. We, as the RPM, will need the agenda, or if you would like to cancel the meeting, at least 24 hours prior to your meeting that is scheduled. And I do have that table as a reference in my slides to kind of simplify this. You are able to cancel the meeting or specify which specific items you'd like to have for discussion after you get the preliminary responses. And one big thing I want to make sure we mention is please do not submit new information or alternate approaches in response to the preliminary responses. The FDA team, specifically our reviewers, will not be able to review and provide adequate feedback on your new information ahead or even during the meeting. So I stress that clearly. Thank you so much.
Excellent point. Our next question is a combination question. I have a combination product. How do I know I'm sending it to the correct center?
We are large and mighty here in the FDA, and there are many centers, and this is a hard one to answer, but we can simplify this by using, there is a CBER product jurisdiction office, and their email is cberproductjurisdiction@fda.hhs.gov. And this is so that you can ask for a formal assignment request, and you can even use the request for designation document that is found on the FDA's Office of Combination Products. Generally, when you are looking at a combination product, this will be based on primary mode of action and indication, so it can get quite muddy, but always ask before if you can, if you have time. Thank you, Leish.
Our next question is about a meeting. When will I hear back if my meeting was granted or denied?
Yes, the when and where you'll hear back from us. So right now for Interacts, we have, you will get an FDA response at 21 days. Again, this is not a PDUFA mandated response, but Interacts will be 21 days. You will get a notification from your regulatory project manager. And it will also, if you are denied, it will include the rationale for that. Now for the PDUFA meeting types, so for type A's, you will get a response within 14 days of your request receipt date, okay? These are calendar days. Type B meetings, this will be a basic B meeting. This will be 21 days. And then end of phase B, type B meetings, those special ones, will be 14 days. Type C meetings are 21 days. And if at any point in time we don't encourage it, but you are able to reach out to the regulatory project manager to ask for an update. Thank you, Leish.
We have a question about COVID. How has COVID impacted request times for meeting dates, referring specifically to Interact Type B and Type C meetings?
Thank you, Leish. COVID has definitely impacted our lives very much. However, we still are looking at PDUFA goal dates. So these dates are still what we are essentially aiming to please. If there is a delay, your regulatory project manager will notify you as soon as possible, and we will work in negotiating a acceptable timeline. As I mentioned before, these meeting PDUFA timeline dates goals will remain the same, and we will strive to meet them. And we actually have been pretty good about them. Thank you so much.
Next question. Could you clarify how FDA defines a research IND that's exempt from ECTD submission requirements? Is this correct? It's the nature of the IND sponsor. For example, if the sponsor is an investigator or academic sponsor, and not the objective of the clinical research study.
Great, Leish. That's an interesting question. The exemption from ECTD is based on if you are research IND. Commercial INDs, if you are aiming for a BLA, definitely will be ECTD format. We encourage to be in ECTD format for review processes, but if we go back to the 1571 form, there's a section in which you will identify your own research or commercial IND, and that check mark is your first indicator. If there needs to be a change or you move into a phase 3, you should be notified, or we will notify you that you will need to go into ECTD. Thank you, Leish.
So next question. If a sponsor submits an IND to CDER or an IDE to CDRH for clinical investigation of ED 351 HCT-P, will FDA redirect it to the appropriate CBER office, or must the sponsor resubmit the application directly to the appropriate CBER office? What effect does this have on the 30-day review period, if any?
Hi, thank you so much. That is, again, another jurisdiction question that happens frequently. This does affect the 30-day review period, and so identifying and making sure you're submitting to the correct office. If you are sending to CDER and you try to go through, or I'm sorry, I apologize, if you're trying to send it to us at CBER and you're using NextGen, which is a CDER product, it makes it very difficult. The redirection could be will be identified by your regulatory project manager. You will either be told to resubmit through the gateway or through the submission email, and it will affect either restart, or you may on our end may eat into that 30 calendar day review period. So if it is identified, we will re-notify you immediately, and the next steps will be identified. However, to avoid this, please use our jurisdiction office, CBER jurisdiction office, before, if there is any question or concern. Thank you.
Okay, we have about 2 minutes left. Hopefully we can do one more question. And this question is it okay to provide an update during a rolling review before the last piece received? As an example, after 5 months, there may be an update to the CMC ahead of the last clinical piece. Is it okay to provide this update ahead of the official clock, before the official clock starts?
Hi, so rolling review being granted is very special. However, all portions of the application should be submitted no more than 1 year after the initial portion. Any review of portions already submitted may be suspended due to the ability to conduct a meaningful review, may be inhibited without the information. So again, that one year after the initial portion should be your goal to get all the information in. Thank you.
Let's see if we could squeeze in one more question. When we submit a BLA after receiving a refuse to file, does the sponsor need to pay a new filing fee?
Oh, a money question. So if you withdraw your BLA before the filing action, you will get 75% back. Now, if you are RTF'd, as long as you're resubmitting, you should not have to pay another fee. Now, if you withdraw and have to resubmit a brand new BLA, yes, you might be subjected to a new fee. Thank you.
Thank you so much, Jennifer, and thank you to the audience for attending our presentation. This concludes our session. With that, I'll pass on the floor to our next moderator, Irina Tipper.
Hi everyone, my name is Irina Tipper and I'm a biologist in the Cell Therapies branch. I'm here to introduce our two following speakers, Dr. Thomas Finn and Dr. Malcolm Moose. Dr. Thomas Finn received his PhD in cell biology from Oregon Health and Sciences University, did postdoctoral work in neuroimmunology at the Portland VA Medical Center, and was research assistant professor in the Department of Neuroscience at Georgetown University. He then joined the FDA in 2006, where he reviews regulatory submissions at all stages of product development. Dr. Finn reviews a wide range of cellular therapies such as cancer immunotherapies, regenerative medicine product, including neural and cardiovascular stem cell-based product, and immunomodulatory therapies. Dr. Finn has regulatory expertise in bioassays, product comparability, in product stability and process validation. The next speaker, Dr. Moose, majored in biological sciences at Stanford University and obtained his MD and PhD at the University of Minnesota. He completed his residency training in laboratory medicine and pathology with a fellowship in clinical oncology at the same institution. He then came to the FDA CBER to continue his work in the area of cyclic nucleotide-mediated signal transduction and became a recognized expert in protein microsequencing, separation techniques, and protein analytical biochemistry. His work has resulted in 7 patents issued, 1 allowed, and 2 pending. While at CBER, he has been a CMC reviewer on IND submissions and BLAs, primary contributing author on various FDA International Council of Harmonization guidance documents, and he is a recipient of many awards, including the Harvey W. Wiley Medal, the FDA Award of Merit, 2 Center Directors Target Research Award, among many, many others.
Thank you for that introduction. Today, I will talk about some of the challenges for cell therapy product development that are either unique to these products or similar to other biologics, but of greater concern. I'll highlight some of the major regulatory concerns and point out some of the control strategies that can be used. And I'll discuss the importance of product monitoring quality as it relates to manufacturing consistency and considerations for evaluating comparability after a manufacturing change. One of the most common questions our division in CBER gets asked is, what are the major regulatory concerns for cell therapy products? But it's important to understand that cell therapies represent a huge range of products with very different levels of manufacturing complexity, product types, and underlying risks. So there is no one size that fits all. You need to consider what it is, how it's made, and what it's supposed to do. So in thinking about products, and we don't have time to go over all the different types, but we can think of kind of a spectrum of products. And at one end of the spectrum, you have kind of the individualized products, say an autologous product that's going to treat a single patient, and an off-the-shelf product at the other end that might be based on a multi-tiered cell bank that's going to be used to treat hundreds, possibly thousands of patients. So there are common concerns with all of these products, and they're similar to other biologics as well. But there's also specific concerns about each, or there are concerns that are more of importance for one type of product than the other. So for patient-specific individualized therapies, we're very concerned about product tracking and segregation because it's very important to make sure that the patient gets the right product and it's not cross-contaminated. There can be high product variability, issues with shelf life, and manufacturing logistics. On the off-the-shelf, variety, you have lots of concerns about qualification of the cell banks and how such a bank would be replaced if it was exhausted. Concerns about the stability of the intermediates and the cell banks and how that might be scaled up. So an example of a concern that is common to all these products is reagents. And there's kind of 2 sources of confusion surrounding reagents. So FDA always recommends using the highest grade or quality version that's available. So if that's a licensed product, all the better. But in some cases, only research-grade reagents are available. And it'll say right on the package, not for clinical use, not for manufacturing, for research purposes only. So that means it can't be used, right? Well, it possibly could if the risks are assessed and mitigated, if it's properly qualified, and if justification is provided. I mean, in some cases, only research-grade is available. There's also confusion around GMP grade. So, FDA likes things being done under GMP. So if I buy a GMP version of a material, that means all the regulatory concerns go away. Not necessarily. For one thing, GMP is not a grade. It's not a universal standard. It is meant to maximize manufacturing consistency and minimize cross-contamination or contamination. The fact that something is made in a GMP facility does not necessarily guarantee that all regulatory concerns would be addressed or that appropriate controls are in place. And very often it's not even disclosed what GMP controls are used by any particular supplier. So you need to think about that. When thinking about cell therapies, you have to realize that it's kind of a double-edged sword. So you have a lot of advantages. So, these products can have multiple mechanisms of action, so that gives them multiple ways that they could work. They might be used as a platform technology, and you could treat a variety of diseases. And it's not perhaps just a treatment, but a possible cure. And the cells can be manipulated in culture and drive to a particular phenotype or specificity or activation state. So that's very powerful. But for some of the same reasons, there are also a lot of challenges. If there's multiple mechanisms of action, well, how do you know what to measure? If you have something that is very sensitive to, culture conditions, that means that's also something that you need to pay a lot of attention to control, because little differences could really add up. And there can be lots of issues with product variability and manufacturing consistency. So a good example of a product where, to kind of illustrate some of the complexities and considerations around control strategies is tissue engineered products. So say you want to grow cells on a 3D scaffold, and that's going to be transplanted into the patient as kind of a regenerative medicine approach. You're going to replace a tissue. So you have, first off, all the normal concerns about the manufacturing of the cells, the reagents, the facilities, the quality of the materials. But then also, the level of testing that you're going to do, how you're going to aseptically manufacture it, the viability of those cells and the stability and the potency. But now you also have concerns about the scaffold itself. So now you have some material issues to consider. So like the physical strength and integrity, how quickly this might break down in vivo, or how long it might persist if you want it to persist. Again, the materials that are used and any residuals that are left over in the scaffold, and biocompatibility of the scaffold with the tissue, you're going to transplant it into. And so now you're going to, grow cells on that scaffold. And because cells are sensitive about their environment, the scaffold could change the properties of the cells. Likewise, the cells could change the property of the scaffolds. They could secrete enzymes. They could, result in the scaffold breaking down faster. They might change the 3D properties. Then one has to also consider, how do you sample something like that? How uniform is it? How reproducible is it to make a construct like that? And how would you ship this to a clinical site, and how would they handle it? And what's the stability of it while you're waiting to transplant it? And would it remodel in vivo? So lots of things to consider when you're dealing with cell therapies. And when I talk about manufacturing, I don't want to just limit that to a consideration of what goes on within the walls of the GMP facility. So you want to think very carefully about how the product is going to be delivered to the clinical site, how they're going to handle it. Do they have to reformulate it? Or is it going to be in a pre-filled syringe? How stable is the cells? How reproducible, is the injection volume? Are you going to deliver the correct target dose? Is the route of administration appropriate? And, what is the suitability of both the injection device and, the final container and so forth for that particular product? So there's biocompatibility of both the injection device with the patient, but also biocompatibility of the product with the injection device itself. And then there's concerns about logistics. So for some cell therapies, the logistics can be quite complicated, especially for some patient-specific products, because now you have to coordinate a donor with the manufacturing schedule and the testing schedule, you might need to precondition the patient, and that has to be coordinated with the timing of the manufacturing and when you're going to do a final harvest and formulation and release. And ultimately, it has to be delivered to a clinical site that works for the patient, that works for the healthcare professionals, and all this working within the shelf life of the product. And so that could be a very delicate dance. Establishing specifications is an important control strategy for any product. And it's a good reminder that, specifications don't determine whether a product is of the quality of the product. The process will determine, how good an individual product lot is. But the testing can be used to confirm the quality of the product and to make sure that it meets minimum quality standards. With regards to cell therapies, it can be tough to decide what to measure and at what level. And very often what we see is that there's too little that's being measured. And we understand it's challenging to decide what to measure, but you really should be measuring things that are important to assure the quality of the product and is consistent with the mechanism of action. And also to think about, measuring not only what you want to have in the product, but what you don't want to have. So some other common issues with choosing release specifications is that, the manufacturing and clinical data are inconsistent with what the specifications are. That is, maybe the specifications are set much wider or what much lower thresholds than what your actual experience is. There could be lack of supported data or rationale. Or the criteria can be set for a very wide range, and that could add variability to your clinical trial and then make it more difficult to assess the quality of the product and manufacturing consistency. Perhaps one of the most challenging assays to develop is the potency assay, and you'll hear more about that in a couple of gene therapy talks tomorrow. But what we generally recommend is that for the cell therapy, in addition to concerns about, the usual, precision and accuracy of an assay, is to measure something meaningful. And often that's where, a potency assay for cell therapies falls short. And what we recommend is that you try to gain as much product knowledge as you can and try to build a potency assay based on the mechanism of action in your proof of concept studies, so that hopefully you end up with something that is predictive of efficacy in the patient. And we strongly recommend you start early, because this is not easy to do. And it may not just rely on one assay. You may need to use a matrix approach to measure multiple things, or you might need to try out different assays to decide what will work best for you. When thinking about specifications, it's not a one and done. You should continually think about the suitability of your specifications for assessing product quality. So you should use the cell phone model, where you always want the latest and greatest. And it's understood that during product development, during the life cycle, those specifications might be pruned or expanded or tweaked or optimized and so forth. So they can and perhaps should change. So that brings us to our first challenge question. Which of the following is not true? That is, which is false about cell therapy manufacturing controls? Is it A, meaningful sensitive potency assays are particularly challenging to develop? B, as long as the manufacturer uses GMP grade reagents, all regulatory concerns are addressed? C, manufacturing and testing logistics can be very challenging? And D, cells are very sensitive to manufacturing conditions, which offers advantages but some challenges. So think about which of those is not true. So the correct answer here is B. We recommend that the highest grade version of a material be used, and in some cases that would be a GMP version. It's not a grade, but that is not necessarily a guarantee that it's of appropriate quality. So let's talk more about variability of patient-specific products. And by variability, well, for some products that may not be so much, but for some products that could be many-fold. So, this could be a really wide range of final product attributes. And so you might think, well, hey, these are individualized patients' products, and so, you expect every patient to be different. It's expected that these will be very individualized and unique, and like snowflakes, no two will be alike, and therefore, variability is fine. Well, you should try to control for what you can. So there's variability of the source material, yes. And there can be variability that's introduced by the manufacturing process. You should try to the extent that you can, control both. For your acceptance criteria for your source material, you can decide, what upstream you need to generate a consistent product downstream. You could also introduce in-process flexibility in how you're manufacturing in order to try to drive a more uniform product in the end, in a quality by design like approach. But in some cases, you may have to set really wide specifications. So what's the consequence of that? Well, if you have, say, a well-characterized biologic, and you have really good process and product understanding, and you have a lot of controls in place, you can typically generate a final product made to tight tolerances. And if something meets those tolerances, you can have, good assurance that that's a quality product and that your process must have worked well. That's not necessarily the case if your specifications are really wide, because it's a very big target, and something could go wrong and still meet your minimum threshold. So what we've kind of recommend is that, as you gain process and product knowledge, you should try to make predictions about what you start with and where you ended up with, and always try to, target within that range, more specifically, rather than just treat the whole range as the target. As long as something lands in there, it's fine. And an example of this is in evaluating deviations. Let's say you have a situation where you have something go wrong in your manufacturing process and you lose a bunch of active ingredient. You're trying to decide, well, how big of an impact that was. If your only criterion is to say, well, hey, did it meet lot release? Okay, well, that's an important measure. You certainly should look at that. But that doesn't necessarily determine the full impact. You really should, because that's a pretty low hurdle. You should also consider what is, the manufacturing history. What's an average product look like? For this particular source material, what would you have expected to target, and how close were you of that? That's probably a better predictor of the impact to manufacturing quality than to just to say, did it meet lot release? So that brings us to questions about demonstrating consistency and comparability. So comparability is particularly challenging and can be really limited by things like the consistency of your manufacturing process before the change. So if your manufacturing process is highly variable, it's going to be challenging to demonstrate comparability, because you may have to use statistically a large number of new lots to compare before and after with your new manufacturing change. And that may be unfeasible. You also have to consider the variability of your analytical methods. So we've certainly seen situations before where somebody wants to demonstrate comparability between facility A and B, and they want it to be within 25%. But some of their assays are more variable than that. Well, you could be setting yourself up for failure. So it's in your best interest to try to develop assays that are highly reliable and reproducible, so that, and that will pay dividends down the road when you're trying to do comparability studies. It's also very helpful to have a good level of product characterization so that you have a good idea of what to measure for your assessment. And ultimately, if you have assays that correlate with clinical outcome, that can make it a lot easier to decide, well, what level of difference is acceptable between process A and B, or facility A and facility B, because you'll know from clinical data what does or doesn't have an impact. But that doesn't always occur. For some cell therapies, there is no correlation. That doesn't mean a comparability study can't be done, but it's more challenging. So one approach that can be used, to kind of deal with this high variability lot to lot, is to do split manufacturing, where you will take a particular product lot, you will split the source material or an intermediate, and then you'll look at a change of a downstream process, and you'll do a direct side-by-side comparison. And so you're taking the variability of the source material out of the equation and just looking at the variability of the process and the impact of the manufacturing change. And that can make interpretation of comparability data a lot easier. So a popular situation where there's a manufacturing change is when there's a scale up or a scale out. And, scale up is just making a bigger lot size, and scale out is replicating the process. Instead of making, one lot at a time, you'll make 10 lots at a time. So there's different considerations for each. So for scale up considerations, let's say you want to increase the yield. Well, there's lots of ways that could be done with, more flasks, bigger bioreactors, longer culture times, more passages, higher concentrations of growth factors, and so forth. But keep in mind that just because you get more cells doesn't mean it's the same quality cells, the same potency cells. The properties of those cells can change, and you need to monitor that. And also, not all processes scale well, like enzymatic steps or time-sensitive steps, or filling a biosafety cabinet full of a whole bunch of flasks is generally not a good idea. In terms of scale-out manufacturing considerations, it may be tempting to just replicate everything exactly. What you're doing with one lot, you'll do to all 10 lots exactly the same. But for individual product lots, again, you might want to consider in-process adjustments to help drive a more uniform final product. So a cookie-cutter approach looks good on paper, but it might not end up with products that are as consistent as they could be. You also have to be very careful about material and qualification and process monitoring. Because if you're making 10 things at once and something goes wrong, you're not going to know it, and you could impact 10 lots at once because you won't have the data from the first lot to inform about the second. And ultimately, you want to also think about certain bottlenecks that can happen at a GMP facility. And so we sometimes recommend for complex logistics or big increases in scale up to do a capacity study. And ultimately, we recommend, I know we talked about a lot of things, but we recommend not putting off evaluating, these kind of CMC concerns. You might be tempted to say, well, let's see if the clinical trial data looks good, and if so, then we'll put the investment into, dealing with all the CMC items. The problem with that is that could leave you in a precarious situation where you're sitting on really good clinical data, but you have a couple of years of manufacturing to deal with. And so that's why we recommend that you keep your clinical development and your product development functioning at the same pace. And if these are misaligned, it can lead to problems with, starting late stage studies or submitting a BLA. So that brings us to challenge question number 2. Which of the following best describes comparability studies for cell therapy products? Is it A, cell therapies are well-characterized biologics and therefore easy to demonstrate comparability? Is it B, comparability study design is the same regardless of cell therapy product type? Is it C, comparability can be challenging when there's no correlation between product attributes and clinical outcome? Or is it D, product variability for cell therapies is typically similar to recombinant proteins and other types of biologics? So which one is the best answer? Okay, in this case, the correct answer is comparability can be challenging when there's no correlation between product attributes and clinical outcome. So C. It doesn't mean you can't do a comparability study. It just means, it's hard to justify your approach. So to summarize, cell therapies have tremendous potential, but also present significant challenges to manufacturing and testing. And these challenges vary by product type. Quality of materials is always important, and there can be confusion surrounding that. Cell therapies are highly variable by nature. That differs by product type, but it can be extreme for some products. And so that definitely presents challenges. And patient-specific products can also have a lot of careful considerations about logistics and how things are handled at clinical sites and how they're administered and so forth. So don't overlook that. When scaling out or scaling up, you need to do a comparability study typically to ensure quality is maintained. And product development needs to keep pace with clinical development. Don't get caught off guard. And with that, I will end and be prepared to take questions in the question and answer session. Thank you.
Good afternoon, everyone. I'm going to spend a few minutes to give you a brief orientation to what is meant by a voluntary consensus standard and how it can streamline the regulatory process. What I hope to achieve by the end of the talk is the following. That you will know what a consensus standard is, understand the relation of standards to regulations, to appreciate how external consensus standards can help streamline regulatory compliance. Now, to help with this, let's focus your attention a little bit with a challenge question. How many amino acids are there in the enkephalin pentapeptide? 60, 9, 5, or 7? Believe it or not, this question was used routinely by a professor in my graduate department in prelim oral examinations. Well, if you weren't asleep, I think you probably figured out that the answer is 5. First, let's define a standard. What does the term mean? In this context, it is a common and repeated use of rules, conditions, guidelines, or characteristics for products or related processes and production methods and related management systems practice. And there are important differences between regulations and standards. First, regulations are developed by U.S. regulatory agencies like us to implement and enforce legislation enacted by Congress. In contrast, standards are developed by standards development organizations, or SDOs, and I'll say a little bit more about the characteristics of those organizations in a second. Second, while regulations are developed solely by government agencies, the standards have input not just from us, but also from academia and the private sector. In fact, participation, a characteristic of these organizations is that participation is open. Regulations are mandatory. They have the force and effect of law. But the use of standards is voluntary. Regulations are specific requirements that regulated entities must meet. And finally, in contrast, standards describe how manufacturers might meet regulatory requirements. Now, there are two very important standards development organizations that between them account for a lot of the standards which are of use for the products that our office regulates. The first of these is the International Organization for Standardization, or ISO. The other is the American Society for Testing and Materials, or ASTM International. These organizations satisfy a number of essential requirements in the United States. That conformance is accredited by the American National Standards Institute. And some of the salient requirements are the following. There is a well-defined due process development of a standard requires consensus. There is always an appeals process, and participation is open to any constituency. And finally, no group is allowed to dominate the process. There needs to be a balanced representation between various constituencies that have an interest in the standard in question. And more detail about this is available at the ANSI website. The URL is below, and we will be providing you with these slides so that you can copy that down at your leisure. Now, before I go on, there is a term in our regulations that is described in the a section of our regulations, 21 CFR 610, that's called General Biologics Product Standards. These are not what we're talking about. These are a few specific examples of requirements that biologics must meet. If it were up to me, I would retitle this section General Biological Products Requirements to avoid confusion with external standards. But this is the way the regulation is written at present, and deals, as you see, with what sort of things that you have to test, what are the basically requirements of how you sample, what are the general characteristics of testing for potency for mycoplasma, and what are the labeling standards. Next, I'll say a word or two about why to use these standards. To begin with, the Office of Management and Budget has mandated that we do in a document known as Circular A119, last updated in January 2016. The idea is that the use of standards can streamline some of our internal processes, and that's largely true. Second, the recently enacted 21st Century Cures Act explicitly directs us to cooperate with NIST in developing standards of various types in the area of regenerative medicine. I'll say a little bit more about this in a second. Third, they're kept up to date. These standards must be voted on at least every 5 years if developments in a particular field merit a revision at an earlier time. That can also be done. This is a big advantage over a typical average review article that may become dated in a rapidly emerging field. And finally, and most importantly, they can save you work. The main idea is that if somebody has come up with a specification for a material or an assay method, you don't have to reinvent the wheel. You can incorporate the standard by reference in your submission, and you will not have to justify every element of it in a one-off way in each submission. Now, I mentioned the 21st Century Cures Act, Section 3036, as it happens, and it tells us to work with the National Institute of Standards and Technology and external participants to, as the Act says, facilitate an effort to coordinate and prioritize the development of standards and consensus definition of terms through a public process to support through regulatory predictability the development, evaluation, and review of regenerative medicine therapies with respect to manufacturing processes and controls of such products. Well, the idea of cooperating with NIST is not new to us. We've been collaborating with NIST for many years in several areas. First, we have laboratory collaborations in areas such as cell counting, cell viability, and related attributes, flow cytometry, and others. Second, we co-host workshops in similar areas, including cell counting and characterization, flow cytometry, genome editing, and most recently, systems biology. And we also both participate in standards development committees within SDOs. Prominent examples are the ISO TC276 Biotechnology Committee and the ASTM FO4 committee, several of them related to tissue engineered medical products. So we're already ahead of the game as far as that goes. And just a comment about our joint participation. What that means is both NIST and we will have provided input into these standards. So if a standard makes it through the process and has been published, you can be pretty sure that we're going to be okay with it. Now, there are a couple of main types of standards: physical standards and written or documentary standards. A physical standard is defined as a material sufficiently homogeneous and stable with respect to one or more specified properties, which has been established to be fit for its intended use in a measurement process. Now that's very carefully parsed language, I think you'll appreciate, and the question of what is sufficiently homogeneous will depend in great measure on the material in question. So there is an ASTM standard for type 1 water that talks about a pure material. But there are other types of physical standards, for example, the genome in a bottle and standards which I know to be under development that won't be normal genomes in a bottle, but abnormal genomes in the bottle. It will be heterogeneous, but still sufficiently homogeneous to be fit for its intended use. And I may be belaboring that subtle parsing of language, but I want to point out to you that standards that have heterogeneity in them may still be useful. Now, there are a couple of levels of standardization. An in-house reference material is something that a particular party might develop to use simply to assure that an assay is performing consistently. But there are more formally qualified materials, such as a NIST standard reference material. That's actually a NIST trademark, or a certified reference material, which includes a statement of the value or values that are measured by a validated metrologically sound process, and limits of uncertainty about those values. But there are other types of standards as well. Now, and I'll say more about those in a second. Some basics of using these in regulatory submissions is, first, they're voluntary. Just because a standard exists doesn't mean you have to use them, number one. Number two, you can participate in developing them. As I said, the process is open to participation from any quarter, and I'll say more about that in a bit also. As I alluded to a little bit ago, they're quite useful in helping you avoid reinventing the wheel. And so an incorporation by reference can allow you to skip describing, qualifying, and justifying a particular assay method if there's already a standard describing it. Third, it's important to remind you that they're developed by a consensus process. That means that people agree from various constituencies on the content of the standard. And the people who show up at these meetings are really sharp. There is a characteristic of these standards that they're eminence-driven. And so the work products that result in published standards generally are of extremely high quality. Now, here are some examples of the second type of standard, the documentary standard. In the area of tissue engineering, there are many more of these than there are of physical standards. There are terminology or definition standards, so people call the same things by the same terms. There are two types of general overviews of the subject area. A standard guide, which is much like a literature review, although curated and revised by the 5-year limit that I mentioned earlier, and standard practices, which focus on methods of doing things. Then there are explicit, detailed procedures. How might you exactly measure the bone-forming activity of a test article in vivo, for example? That would be called a test method. And finally, there are explicit characteristics or performance standards. These are an enumeration of the characteristics that a particular material might have to make. I mentioned ASTM pipe water a second ago. It has to have a resistivity of 18 megohms per centimeter, a total organic content of, I think, not more than 5 or 10 parts per billion, and so forth. There's a whole list. And they can be used for exploratory preclinical work. That is to say, they're not necessarily regulatory documents. We say a lot about their use in regulatory submissions because a lot of people want to hear about that, but they are typically written in such a way that they can be used by a field in general, including for exploratory work, as well as in regulatory submissions for methods that may be germane to in-process or release testing, or, as I mentioned in the last slide, to specify explicit physicochemical or performance characteristics. So, to summarize, any type of standard can be used that's appropriate to the context. You must justify its use as written, or if you want to modify it, you can say this procedure was carried out according to ASTM or ISO or whatever standard number, whatever, modified as follows, and enumerate the modifications. How well you justify its use will be a review issue for us. There is a guidance document on standards. It's called Standards Development and the Use of Standards in Regulatory Submissions, reviewed in the Center for Biologics, Evaluation and Research, that describes the policy for staff participation in standards development, use of standards, documentation, how to use standards for CBER regulatory devices. So, to conclude, consensus standards are developed by SDOs. They can apply to exploratory or preclinical research, as well as used in regulatory submissions. When they are used in regulatory submissions, they can also streamline regulatory compliance. They are developed with an open, well-defined due process. In other terms, an open, voluntary, consensus-driven process means anyone can take a seat at the table or end up on the menu. To conclude, I'll give you my other challenge question. Which of the following statements is not true? The indicated standard must always be used exactly as written. Use of external consensus standards is voluntary. Standards must be reapproved every 5 years, but can be revised sooner if needed, or external consensus standards may be useful during research, preclinical development, not just regulatory filings. Well, the correct answer is A, you don't have to use the standard exactly as written. So to conclude, I will thank you for your attention and invite questions.
Thank you, Dr. Finn and Dr. Moose, for wonderful and really informative presentations. We will now move on to our Q&A session. Dr. Finn, for an investigation allogeneic cell bank-based product, would the change in the cell line always necessitate generating new non-clinical data, or could CMC comparability suffice, assuming extensive data package and data looks good?
Hi, so that's a great question, and it actually comes up fairly often.
Excuse us for interrupting Dr. Finn. There we go. It seems to be having some congestion. If you could try muting your mic and unmuting again, we may be able to get that audio issue taken care of. Please go ahead and try answering that audio again.
Hi, can you hear me okay now? Hi, can you hear me okay now? Okay.
Sorry, I was back on mute. Yes, we can hear you just fine. Go ahead.
Okay. So that's a great question, and it comes up fairly often because we will have
And it does seem we're still having some issues with Dr. Finn's microphone. Irina, if you could, please go ahead and let's move on to a question for Malcolm. And I'll work with Dr. Finn to try to figure out what's going on with the microphone.
Sounds good. We look forward to hearing Dr. Finn's response. In the meantime, let's move on to a couple of questions from Dr. Moose. Dr. Moose, could you say a bit more about participating in standards development?
Sure. I hope, can I be heard?
Yes, we can hear you loud and clear.
Okay, great. The process really has a very low bar for activation in ASTM. You basically just show up. There's a nominal yearly fee if you want to be a voting member of, I think, $50. But that's it. One of the things about this process that makes it work so well is its openness. That any interested party can participate. And this draws in, as I mentioned, a lot of very eminent people, number one, and number two, lowers the barriers to doing this. The other result is that the nature of the group involved tends to be extremely collegial, and it is possible to work together to develop very high quality work products in a way that is very open, transparent, and fair.
Excellent, thank you. And how exactly do standards get revised?
So that can happen in a number of ways. The default is that after 5 years, a standard is flagged to be reballoted, and people in the relevant subcommittee have to look over it in order to cast their ballot accurately. and determine if a revision is needed. It's not uncommon, especially in a field that's moving quickly, for something to happen as simple as simply the publication of one consequential paper that somebody notices may require a couple of sentences to be revised, and then there can be a quick series of emails, a revision suggested, and then a reballoting at the next opportunity. These ASTM ballots occur at relatively flexible intervals, not less for a given subcommittee than twice a year, but it can be more frequent depending on the need.
Thank you, Dr. Moose. And while Dr. Finn is getting back online, why don't we try to squeeze in another question for you? What happens when there's a disagreement during the development of a standard?
Well, there are a couple of points at which a disagreement can occur. The standard development process usually begins at one of the semi-annual meetings when the need for it is discussed, and either the committee votes to create what's called a work item, or decides it's premature and votes the other way. Frequently, discussions about the scope of the document are very, I would say, spirited, and after a great deal of discussion in many cases, there is agreement and draft language is proposed. Then a draft is circulated, and this sometimes happens informally before a formal vote, and points of discussion can be raised within the task group that's working on actually drafting the standard, and revisions can be done. But during a vote, somebody may have an objection to something technically or a language issue, and has the option to vote either, well, there are four vote types: affirmative with comment, which is not binding but suggests a revision, negative, and abstain. A negative vote has to be addressed. One negative vote stops the process cold. What usually happens first is that the technical lead for the standard contacts the negative voter, and it's an open process. You don't get to vote negative anonymously. And discusses the issue, discusses what would be a reasonable fix, and I would say a large majority of the time, the negative is addressed by a simple text revision. And this may require a little bit of back and forth if an issue is subtle. In relatively rare cases, the negative voter is adamant in his or her position, and the committee has to vote as to whether the negative is, quote, persuasive, unquote, or, quote, non-persuasive, unquote. And if the committee votes the negative as non-persuasive, it is overruled and the process resumes.
Thank you, Dr. Moose. Why don't we go back and see if Dr. Finn has his audio back online? So, Dr. Finn, when we're talking about allogeneic cell bank-based products, would a change in the cell line always necessitate generating new non-clinical data, or could CMC comparability suffice, of course assuming that there is extensive data package and the data looks good?
Hi, Irina. Can you hear me okay this time?
Yes, I can hear you. Thank you.
Okay, so let's try again. Okay, so we get that question every so often because we have situations where somebody develops a product with one cell line, but when they submit their IND, they've used So the answer to the question depends on what's the degree of change. If the change is, say, somebody just used a new working cell bank from the same master cell bank, manufactured the same way, tested the same way, and the data looks good as the question posed, then the answer is the comparability data would support the use of that new cell line, and new studies wouldn't be needed. Well, what if, say, a new master cell bank manufactured in a different, say, optimized way was used, and now the final product isn't quite the same? Then, that needs more careful review. Possibly bridging studies might be needed. It might not require all preclinical studies to be repeated, and maybe some bench testing could be used to support. If it's an entirely different cell line, let's say somebody's using MSCs for the animal studies and neural stem cells for the actual product, and the properties are quite different, then yes, it might require new preclinical studies.
Excellent. Thank you, Dr. Finn. And since we got a chance to hear from Dr. Moose on a couple of questions, we will keep the same pattern and do a couple of questions for you in a row. So my next question for you is, can a BLA be submitted prior to process validation of a cell therapy product?
So the short answer is no. And the reason is process validation is really fundamental for a BLA submission. So if one thinks of a rolling submission where one might submit, say, the clinical data first and CMC sections last, you might think, well, could I roll, within the CMC section something like process validation? It is possible, and it could be negotiated for some types of CMC information to be submitted after the BLA and after the CMC section is submitted, for things like ongoing stability data, where you're looking for longer time points and that data has yet to come in. That could be negotiated at the time of the pre-BLA. But something as fundamental as process validation should be submitted with the main application. I should point out that when we review either an IND or a BLA, we don't review each section independently. Something like process validation is important to help support control of materials and assay validation, and vice versa. So all these modules and sections within Module 3 are interdependent in many instances. And so leaving out process validation would be leaving out a lot more than just that one section.
Thank you. And we have just a few seconds over a minute left, so why don't we try squeezing in the last question. What is the difference between scale out and technology transfer?
So these are related. So tech transfer usually refers to say you're opening a new QC facility or a new manufacturing facility, and you're either transferring, manufacturing to that facility, or you're going to duplicate it at another facility. And so there's a process of where you're training individuals, you're setting up your equipment, and you're transferring all that SOPs and so forth to the new facility, because it's just going to be done the same way. And then you would do verification studies at the new facility to show that you have product comparability and that the manufacturing is functioning as intended. Scale out is like that, but it could also be scale out at the same facility where you're just expanding within the same facility. So scale out just refers to any replication usually with replicate equipment modules or clean room modules within the same facility or new facilities.
Thank you, Dr. Finn, and thank you to our audience for participation. Next up, we will take a 5-minute break and we will resume at 4:15.
Hi, welcome back. My name is Shayna Hardy. I am a pharmacology toxicology reviewer in the pharmacology toxicology branch in OTAT, and I will be serving as the moderator for this session. I will briefly introduce our next speakers. Our first speaker is Dr. Danielle Brooks. Dr. Brooks received her PhD in biomedical sciences at the University of Tennessee Health Science Center. Following her graduate training, Dr. Brooks completed her postdoctoral training in the Women's Malignancies branch of the National Cancer Institute. In 2017, she joined the NCI FDA Interagency Oncology Task Force Fellowship Program as a Product Quality Research Review Fellow in the Cellular and Tissues Therapies branch of OTAT. At the completion of her fellowship, Dr. Brooks joined the Pharmacology Toxicology branch as a Pharm Tox reviewer, where she now focuses on the review of preclinical toxicology and pharmacology data to support the safety of cell and gene therapies, tissue engineered products, devices, and combination products. Our second speaker will be Dr. Elizabeth Hart. Dr. Hart completed her undergraduate medical training at the University of Pennsylvania. She also completed a residency in pediatrics at Rainbow Babies and Children's Hospital and a fellowship in a pediatric endocrinology at Boston Children's Hospital. Upon joining the FDA, she initially served as a medical officer in the Division of Gastroenterology and Inborn Error Products, where she developed expertise in innovative clinical trial design and analysis for products aimed to treat rare diseases. She joined OTAT in 2019 and currently serves as the branch chief of the General Medicine branch, where she supervises a team of medical officers who are responsible for regulating cellular and gene therapies for a variety of medical conditions. I will now hand it off to our speakers, who will be discussing preclinical and clinical considerations for the development of cellular therapies.
Hello and good afternoon. My name is Danielle Brooks, and I am a preclinical reviewer in the Pharmacology Toxicology branch of the Division of Clinical Evaluation and Pharmacology Toxicology within CBER OTAT. I would like to thank the organizers of this meeting for inviting me to talk with you all and give you some preclinical development considerations for cellular therapies. So the learning objectives for today's talk will be to describe the organizational structure of CBER OTAT DSEPT and the products that we regulate, to understand preclinical considerations for assessing the safety of cell-based products, to understand principles for selecting appropriate animal species and models for preclinical studies of cellular therapies, and to be familiar with opportunities for early interaction with OTAT during development of your product. So let's get started by talking about the organizational structure of our OTAT and where we are in CBER and the products that we regulate. So the Office of Tissues and Advanced Therapies, or OTAT, is one of the main review offices within the Center for Biologics Evaluation and Research, or CBER. There are multiple divisions within OTAT that each perform a specific function. The review of clinical data as well as pharmacology toxicology data lies within the Division of Clinical Evaluation and Pharmacology Toxicology. Within the Division of Clinical Pharmacology and Toxicology, or DSEPT, are the PT reviewers as well as the clinical reviewers. There are 4 clinical branches of review, which are split up based on specific clinical indications. And then there are 2 pharmacology toxicology branches. And unlike the clinical branches, the PT branches are not split based on clinical indication nor on product type. So any one PT reviewer could review a file for any clinical indication as well as any product type. And when I say any product type, I do mean any of the diverse products that we regulate within OTAT. I am talking specifically about cell therapies today. However, we also regulate gene therapies, which my colleague Dan will talk about preclinical considerations for gene therapies tomorrow. So we regulate gene therapies, stem cells and stem cell-derived products, functionally mature or differentiated cells, products for xenotransplantation, therapeutic vaccines, blood and plasma-derived products, tissues, devices, and combination products, which can include device-biologic combinations. Some specific examples of cell-based products that we regulate within OTAT are mesenchymal stromal or stem cells. These can be bone marrow derived, adipose tissue, umbilical cord derived, cell-based therapeutic vaccines, which include dendritic cells or irradiated tumor cells. As part of cell-based products, we also regulate exosomes or extracellular vesicles, induced pluripotent stem cells or embryonic stem cell-based products, and immune cells such as NK cells, tumor-infiltrating lymphocytes, viral-specific T cells, and regulatory T cells. I do want to note here that when we talk about cell-based products within OTAT, we are talking specifically of unmodified, genetically modified cells. So any cell product that has undergone any sort of genetic modification, such as a CAR T cell or an NK cell that has been knocked out for any specific gene locus, those are considered gene therapies in OTAT. And as I stated, my colleague Dan will talk more about that tomorrow. So just to give you an idea of what the number of INDs look like within OTAT for cell therapy development programs, you can see over the last few years that we have had quite an increase in interest in cell therapy within OTAT. Specifically from 2016 to 2020, there was an increase of 140% in IND submissions with cell therapy development programs. So we have quite a lot of interest in cell therapies. And hopefully for those of you who are interested in specifically developing cell therapy, what I am going to talk about will be of help to you. So let's get started with some considerations for preclinical programs for cell therapy products. First, let's talk about how the preclinical data can contribute to the proposed clinical plan for our cell therapy. So the data should provide justification for the clinical trial in subjects with the target disease. The data should support the starting clinical dose level, the dosing regimen, and the route of administration, and should establish feasibility and reasonable safety of the product administration procedure. And this can include whether or not there is an associated delivery device, be it a novel device or maybe a more complicated delivery device that requires additional safety and feasibility assessments. The data should support patient eligibility criteria. This includes if the patient population is for pediatrics, then the preclinical data should provide a prospect of direct benefit to support this, especially if there is no existing clinical data that can be leveraged. Finally, the preclinical data should identify potential toxicities and physiologic parameters that can help guide a clinical monitoring plan. So how do we, within CBER OTAT, evaluate preclinical safety and activity of cell therapy products? And what should you, as a sponsor, be thinking about when developing a new product? So we do have a guidance for industry that came out in 2013 that is specific for preclinical assessment of investigational cellular and gene therapy products. And a lot of what I'm discussing in this talk can actually be found in the guidance. And this just provides our general recommendations to consider when developing your product. Before I get into more specifics about preclinical testing programs for cell therapies, I just want to say that the diversity and biological properties of cell therapy products really necessitate a flexible testing strategy. There is no one-size-fits-all approach for these products, as they are so diverse and have so many different biological properties. Any testing program should be based on accumulated knowledge and experience of both the product as well as the clinical indication. It should be based on available technology, and it should definitely be science-based and data-driven. So when designing a preclinical testing program, a sponsor should consider a few things. First, the putative mechanism of action and the intrinsic properties of the product. A good understanding of these is really going to guide the overall preclinical testing design. What the proposed clinical indication is and whether there are appropriate animal species or models to be able to adequately evaluate the product for the clinical indication. And also whether or not there is existing data, preclinical or clinical, that can be leveraged to support the product and the proposed patient population, as well as the clinical route of administration. So all of this should be considered as the sponsor is designing their preclinical testing program. So some potential safety concerns that we look at when we are evaluating preclinical data for a cell-based product are changes in cell properties that are a result of ex vivo manipulation. And this can include not only culture expansion, it can also include encapsulation of the cells or seeding of cells onto a scaffold. Any sort of manipulation that may change the intrinsic properties of the cells that could result in a potential safety concern. Potential for an inflammatory or immune response, tumorigenicity dependent on the cell type, or inappropriate cell differentiation. What the cell distribution is and whether the distribution is to non-target sites or tissues, and whether there are risks of the delivery procedure or the delivery device that is being used. So the overall preclinical testing program for a cell-based product can include multiple studies that can address the safety concerns that I just discussed on the previous slide. These studies include proof of concept studies, cell fate assessment studies, immunogenicity and host response assessments, and general safety and tumorigenicity assessments. And the goal of proof of concept studies is really to demonstrate evidence of biological activity of your cellular product in the clinical indication. And this can be done either in vitro or in vivo or both. There are many in vitro systems that can be used to demonstrate evidence of biological activity. Also animal models, whatever animal model is used, should recapitulate aspects of the clinical disease or clinical indication as feasible. There are multiple different studies that can be performed to assess cell fate. Persistence can give an idea of how long cells are viable following administration of the product. This can also be used to inform timing of safety studies, how long studies should be, what the sacrifice time points should be. Cell distribution studies can give an overall idea of where the cells are distributing following administration. And of course, this is going to depend on the route of administration, whether it's intravenous administration, intratumoral administration, just getting an idea of where the cells are going and making sure they're not going to any tissues that they shouldn't be. For example, intratumoral administration, making sure the cells are not migrating out of that local environment. Cell fate can also include the phenotype of the cells, comparing both prior to administration as well as following administration, and then proliferation of the cells can also be included in these studies. And all of this is going to help with interpretation of the overall product activity and safety. When we think about immunogenicity and host response studies, often for animal models, sponsors have to use immunocompromised animals. Since these are human cell products, immunocompromised animals are required so that the cells are not rejected. However, sometimes sponsors do choose to use an animal analogous product. There are instances where some ex vivo manipulations may end up changing some cell properties or potentially introducing some other biomaterials that could, when administered or implanted into a host, result in a potential host response. And so the use of an analogous product, an analogous animal product, could provide a clearer picture of any potential host response when the final clinical product is administered to a subject with an intact immune system. And then general safety assessments I will talk about in the next couple of slides, as well as tumorigenicity assessments. When considering the design of the specific preclinical studies that will be included in a testing program, the sponsor should ensure that the studies are non-biased, meaning that animals are randomized to study groups and any personnel performing assessments be masked to the study groups. The study should mimic the planned clinical scenario as closely as possible, which would include using dose levels that are within the clinical dose level range, the same or very similar dose regimen and dose frequency, including the use of any other products that may be included in the clinical plan, such as the use of immunosuppressions, whether or not there is another product that will be administered, such as a CEDAR-regulated product, a monoclonal antibody, for example, or any other STIBA-regulated products. This can also include whether or not there's a specific delivery device that is going to be used in the clinical trial. This should also be included in any animal studies. Administration of the clinical product as prepared in the clinical formulation should be used as feasible. However, as I mentioned, there is the option for using an analogous animal product or a surrogate product, and this is acceptable as long as there's sufficient justification and discussion on what the similarities and differences are between the clinical product and the preclinical product and how those differences may impact the overall study interpretation. And then using multiple dose levels of the investigational product in order to get an idea of a dose response. And using adequate numbers of animals per group in order to enable for robust study interpretation. Additional considerations include making sure that the study is of a sufficient duration in order to assess both acute and long-term outcomes, as well as multiple time points for evaluation. And of course, this is going to depend on the biology of the product, what the cell persistence is. That can inform how long the study should be and at what intervals you should be sacrificing animals for assessments. Also, comprehensive bioactivity and safety assessments. This can include a large number of things, such as standard toxicology endpoints, which can include mortality, clinical observations, clinical pathology, terminal assessments such as macroscopic observations and histopathology on a full set of tissues. Other more specific assessments can include product-dependent endpoints, such as whether there is an immune response to the product. Disease-specific endpoints can include survival or reversal of the phenotype if you're using an animal model of disease for safety assessments, any functional endpoints, and also safety assessments can include further evaluation of the cell distribution profile and persistence, and depending on the cell type, whether or not there is any evidence of tumorigenicity of the product. But that, again, that's going to depend on the cell product itself. So now that we've gone over some preclinical design considerations, let's talk about selecting appropriate animal species and animal models for the preclinical studies. When considering selecting appropriate animal species or animal models for a cell therapy product, there's no default to the use of non-human primates. There's also no default to the use of both a rodent and a non-rodent species. As I stated earlier, the design of a preclinical program for cell therapy product is done on a case-by-case basis, and this also applies to selecting the best model to use for the studies. The safety and bioactivity of any cell therapy product should be assessed using an appropriate animal model of disease as feasible. However, it's important to understand the limitations of the model as well as the species that the model is in. And scientific justification should always be provided for whatever animal species is selected and whatever model is used. When selecting the animal model that will be used in the preclinical studies, the sponsor should consider what the comparability of the model is to the target patient population. And this can include the phenotype of the disease, how the disease presents in the animal model, how it progresses in the animal model, the age of the animals, the pathophysiology, and any clinical outcomes, what the permissiveness is to the cell product in the specific animal species that's used. I mentioned, again, using human-derived products in animals, whether or not this is an autologous or allogeneic cell product. If you're using an analogous product, all of these should be considered when selecting species. Another important point is the anatomic site of product delivery. As I mentioned, we always recommend that preclinical studies be performed as closely as possible to the clinical scenario. And so this also includes how the product is delivered, the route of delivery, as well as any associated delivery device. The feasibility of the intended device or procedure should be considered. A mouse, for example, is a very small animal. Sometimes certain devices may not be feasible to use in such a small animal. In that case, a larger animal may be needed. So all of these should be considered when selecting the animal species and model. And just some additional considerations when selecting the animal species or model that's going to be used. The availability of the animals and the cost of either purchasing or maintaining the animals, the size, the sex, the age, and any specific housing needs, any animal care concerns. Some disease models may be a little more tricky. Technical feasibility, whether or not there's historical baseline data that can be used to compare maybe findings in clinical pathology data. Historical data could help inform whether or not those findings or changes are significant. And then statistical limitations. I'd also like to note that we encourage sponsors to consider any alternatives for testing that either support animal studies or may also be used in lieu of animal studies. If sponsors have any alternative testing that they would like to use, we are open for discussion for this. And just one final note on selecting animal species. It's important to understand the limitations of species and models and animal data in general when evaluating cell and gene therapies. And it's just important for the sponsors to provide a discussion on this and really understand how this informs the overall study interpretation and how that informs design of the clinical study. Okay, now I would just like to take a minute to go over some opportunities we have to get some early feedback on your preclinical program prior to submitting an IND. So we have two different meeting types available to sponsors during preclinical development. The first is called the Interact, which is the earliest point at which a sponsor can come in to discuss their product development, and the pre-IND meeting, which more people are probably familiar with. So in CBER, we have what's called an Interact meeting, which is initial targeted engagement for regulatory advice on CBER products. And the goal of these meetings is to obtain early feedback on product development for novel investigational agents. And this provides a mechanism for early communication with OTAT and a non-binding informal scientific discussion between CBER reviewers and the sponsor on specific issues. And the point at which a sponsor can request an Interact meeting usually happens once the clinical product has been identified, although finalizing the manufacturing process can still be ongoing. And there should be some preliminary proof of concept data that's generated, maybe some safety studies, but no definitive preclinical safety studies have yet to be conducted. And the PT advice during these meetings is really to provide comments on the design of proof of concept studies or other pilot safety biodistribution studies, to comment on the adequacy of the selected animal species and models, to comment on the acceptability of any innovative preclinical testing strategies or products or the delivery modality, and to maybe provide advice on whether or not the preclinical program or study design needs some modifications, and also, again, to discuss judicious use of animals. We don't want to have sponsors perform studies that really aren't necessary. So we can discuss that at that time as well. And then after the Interact stage, once the sponsor has generated proof of concept studies, they performed some preliminary safety studies and are getting ready to conduct pivotal safety studies, then we have the pre-IND meeting, which goal is to achieve a successful IND submission. And this is a non-binding formal scientific discussion between all review disciplines and the sponsor. And it's an opportunity to really comprehensively communicate the overall product and clinical development plan. And as I said, the sponsor should be ready to conduct pivotal safety studies, but we really prefer that the studies have not been started. Sometimes we do have instances where sponsors already started the study, and that really limits our opportunity to provide constructive and useful feedback. And so in the pre-IND meeting package, the sponsor should provide a comprehensive summary of all the completed studies, as well as complete protocols for the proposed definitive preclinical safety and toxicology studies in order for us to really provide the best, most constructive feedback that we can in order for the sponsor to complete their preclinical program for a successful IND submission. Okay, so that concludes my presentation. So let's quickly just go back and make sure that everyone was paying attention. So the first knowledge check today is what types of products are regulated by CBER OTAT? Device biologic combination products, gene therapy products, tissues, cell therapy products, or all of the above. I'll give you just a second to think on that. And if you guessed E, all of the above, then you would be correct. As I stated early on, we do regulate a wide array of products. The second knowledge check, number 2, non-human primates must be used to evaluate the safety of cell therapy products. True or false? And if you said false, then you would be correct. Non-human primates are not a default to evaluate cell therapy products. It's really a case-by-case, product-dependent whether or not non-human primates need to be used. So just to summarize what we went over today, OTAT resides within CBER, regulates a wide array of products, including cell-based therapies. The preclinical program for any cell therapy product is determined on a case-by-case basis. And the preclinical data submitted in an IND should support the safety and biological activity of the cell-based product in the proposed clinical indication. And there are multiple opportunities to obtain FDA feedback on preclinical plans prior to your IND submission. And with that, I would like to thank everyone for listening to me today and hopefully give you a little bit more understanding of what we're looking at for cell therapy products preclinically. And if there are any questions, I will be happy to answer them during the Q&A. Thank you.
Good afternoon. My name is Elizabeth Hart, and I am the Clinical Branch Chief of General Medicine 1 within the Office of Tissues and Advanced Therapies, also known as OTAT, in CBER. Today I will be discussing the development of cellular therapies, a clinical perspective. The learning objectives for today's session are to identify safety considerations for clinical trials for cellular products, understand strategies to mitigate safety risks in clinical trials for cellular products, and to understand strategies for efficient clinical development for cellular therapy products. I will begin by providing some background information. As you have already heard, cellular products include autologous, allogeneic, or xenogenic living cells that are manipulated or processed ex vivo. These products include, but are not limited to, products derived from pluripotent stem cells, including embryonic stem cells and induced pluripotent stem cells, adult stem cells or progenitor cells, including hematopoietic, neural, cardiac, adipose, and mesenchymal cells. They can be made from perinatal or fetal cells. They can also be differentiated mature cells, such as chondrocytes, pancreatic islet cells, hepatocytes, keratinocytes, retinal pigment, etc. The list goes on and on. These products are being developed for therapeutic, diagnostic, and preventative purposes. As you have already heard, cellular products that are more than minimally manipulated and used for non-homologous use are regulated under Section 351 of the PHS Act and the Federal Food, Drug, and Cosmetic Act. As such, pre-market review and approval is required. They are reviewed and regulated by OTAT. Each investigational product requires at least one IND to begin clinical studies in the United States. As you can see, the research into cellular products has been rising dramatically over the past 5 years. In 2016, there were 63 new INDs for cellular products, and then most recently, that number tripled from just a decade ago. In 2020, there were 151 new research INDs for cellular products. This is an exciting time for the field of cellular therapy. Clinical development for cell therapy is definitely not a one-size-fits-all approach. We recognize that cellular therapy encompasses a range of products, routes of administration, and these products are being developed for a myriad of indications and target populations. Therefore, we recommend that sponsors who are developing cellular therapy products meet with OTAT early in development. Today, I will provide some general consideration and advice. Let us now turn our attention to safety, focusing on first-in-human and early-phase studies. Safety is always primary and paramount. FDA's primary objectives in reviewing an IND are, in all phases of the investigation, to ensure the safety and the rights of subject, and in phase 2 and 3, to help assure the quality of the scientific evaluations of drugs is adequate to permit an evaluation of the product's effectiveness and safety. Some examples of potential safety concern for cell therapy products include immune response to the administered cellular product, such as anaphylaxis, inappropriate cell proliferation and differentiation, such as tumor or ectopic tissue formation, cell migration to non-target areas or tissues, transmission of infectious disease, risks from the delivery procedure, and interactions or complications from concomitant therapies, such as immunosuppressants. Risk mitigation in early phase clinical trials for cellular therapy products. There are 3 things to think about with regard to risk mitigation. First, Who? Second, what? And third, when? Who involves the choice of study population. The eligibility criteria should appropriately select subjects such that the risks are mitigated and subjects have a favorable benefit-risk profile. In evaluating benefit-risk, you need to consider the anticipated effects of the product, the population in which the product is expected to act, and what is known about the class of products and the specific anticipated toxicity for your product based on the preclinical studies. Based on the previously mentioned risks from cellular therapy, otherwise healthy subjects are unlikely to be appropriate for cellular therapy, as they would be exposed to all of the risks of the products without any potential benefit. Depending upon the product and the phase of disease in which the product is anticipated to have its benefit, it may be most appropriate to initially study patients with more advanced disease who may not have other treatment options, as these patients would be more willing to accept risk in the setting of uncertain benefits. Alternatively, it may be more appropriate to choose patients whose disease is not as advanced, as they may have fewer complications and comorbidities, making them less vulnerable to complications from the cellular therapy. Irrespective of the stage of disease selected for your initial clinical study, please ensure that you exclude subjects who would be at unreasonable risk from your product. For instance, a cellular product that is administered in a large volume of fluid may be inappropriate for a patient with advanced congestive heart failure. Eligibility criteria should specify vital signs, laboratory values, and stages of comorbid conditions that would preclude patients from enrolling in the first-in-human study. When there's less information known about the product, it may be appropriate for a first-in-human study to enroll a healthier population with a condition to reduce confounding and to improve interpretability of safety data. What refers to the potential harms related to the cellular product that need to be monitored for and the risk mitigation employed. Protocols should include sufficient description of all safety monitoring to adequately assess safety and mitigate those risks as appropriate. For instance, there should be adequate details to ensure that the product is being appropriately administered and the risks associated with administration are minimized. There should be monitoring for immediate reactions to cellular therapy, such as anaphylaxis, with appropriate personnel and equipment. There should be monitoring for subacute reactions to cellular therapy. For instance, cytokine release syndrome has been reported with some cellular products, so there should be parameters for intervention and increased monitoring if that is a concern with your product. As many cellular therapy products persist and can have long-term effects, it is important that there be adequate long-term safety follow-up. The specific monitoring will always depend upon the specific product. When refers to when to proceed or stop. For first-in-human studies, a staggering interval between subjects and, if applicable, dose cohorts gives an opportunity to monitor for acute and subacute toxicities prior to exposing additional subjects. This provides an opportunity for modifying study procedures and updating the informed consent document and investigator brochure, if appropriate. The specific staggering interval should be informed based on toxicology studies and clinical data from related products. There need to be stopping rules, both subject level and study stopping rules. Subject level stopping rules may be independent of the product and related to change in the subject's condition that affects the ability to safely administer the product. In general, unless a subject withdraws consent, each subject should continue to be followed for safety. Study stopping rules should be based on the number of adverse events, as well as the nature and/or severity of those events, which would trigger the temporary suspension of enrollment and product administration, pending a safety investigation. It is important that the study is paused while investigation of relatedness occurs. An independent data safety monitoring, especially for high-risk products and vulnerable populations, can help monitor the safety of subjects and make decisions regarding when to proceed or stop. The composition, timing of review of the Data Safety Monitoring Board should be included as part of the charter. Again, the overall purpose is to limit the number of patients put at risk if early experience uncovers important safety problems. While safety is paramount, safety alone is not sufficient, so I'd like to now focus on efficient product development. As with other treatments, by law, approval of cellular therapies for marketing is based on substantial evidence of effectiveness and evidence of safety. Evidence of effectiveness should be obtained from adequate and well-controlled studies. We recognize that certain aspects of product development that are feasible for common disease may not be feasible for rare disease. Therefore, FDA regulations provide flexibility in applying these regulatory standards. Let's now spend a moment discussing rare disease and evidence of effectiveness. There are no specific minimum number of patients that must be studied to establish effectiveness and safety. The number of patients needed is determined on a case-by-case basis, taking into consideration the persuasiveness of the data, including comprehensiveness and quality, nature of the benefit provided, length of the treatment or exposure, the patient population that would be treated after marketing approval, and concern for potential of harm from this treatment. Given all of these factors, in order to have efficient product development, it is important to start with the end in mind. While we recognize that the path is not always straight, it is important to think about the clinical data package that will be expected to support a BLA. Sufficient evidence that the product demonstrates a clinically meaningful benefit in how patients feel, function, or survive, and adequate safety information. With this in mind, we recommend that you try and hit a home run with your first study. Design your first-in-human clinical trial to provide evidence of effectiveness. Consider a randomized control trial. Also, whenever feasible, conduct a concurrent control with blinding. Also, I cannot emphasize this enough, please resolve manufacturing issues as much as possible before initiating your clinical trial. If there are substantial product changes, it may not be possible to demonstrate analytical comparability, and this would limit the amount of data that could potentially be extrapolated from your early phase studies with your non-to-be-marketed product. This is why careful planning throughout development is important. While clinical studies happen after submission of the IND, planning for those clinical studies must begin early. While the product development, assay development, and non-clinical studies are occurring, begin to think about if companion diagnostic or devices are needed. Also consider conducting a natural history study, particularly if the disease progression is not well known. Also consider if a clinical outcome assessment may be needed as part of your endpoint development. It is important that these are fit for purpose. They may need to be validated in your new target population. Lastly, begin patient engagement. A little bit more on natural history studies. Conducting a well-designed natural history study may be beneficial to product development. It provides an opportunity to prospectively collect data to understand the spectrum of disease, including variability in the disease course, identifying important baseline prognostic factors, and assessing clinical endpoints. We recommend that if you are conducting a natural history study, the study is designed to rigorously collect data to serve all intended purposes. Patient engagement should also begin early, as patients are the ones who are most knowledgeable about their diseases. The patient voice is an essential part of product development. Patients can help us understand the impact of disease and inform endpoint selection based on what is clinically meaningful to them, and also inform us about risk tolerance in the context of the specific disease and available treatment. Ultimately, they will inform us on the overall benefit-risk assessment. It is important that we also discuss dosing considerations, as due to manufacturing challenges, this affects clinical study design. Please ensure that clinical development includes adequate and complete investigation of the effective and safe dose range to ensure accurate recommendations in your product label. Cell therapies are often a mixture of different cell types. Therefore, please consider the total number of specific cell types, and the total number of cells delivered, as well as cell viability, and ensure that you have appropriate ways to measure the dose. As dose response curves for cellular products may be nonlinear, please consider the scientific rationale for dose escalation and de-escalation, and when appropriate, considerations for redosing. We encourage adequate dose exploration early in development so that a good candidate dose range can be identified as early as possible in your development program to expedite your development program. Lastly, I want to discuss considerations for cellular product development in children, as many cellular products are being developed for children. There are special regulations that apply to children. Under Subpart D, any trial in which more than a minor increase over minimal risk exists, such as all trials for cellular therapy, children must have prospect of direct benefit. Data for prospect of direct benefit can come from animal models of disease or proof of concept clinical studies in adults. From a safety perspective, whenever feasible, safety data should be obtained in adults before initiating pediatric studies. In summary, I'd like to leave you with a few points. First, for subject safety, when conducting an early phase cellular therapy, consider who, what, and when. Second, plan ahead, because with thoughtful design, risk can be minimized and expeditious product development is possible. And lastly, communicate early with the FDA and collaborate with scientists, clinicians, patient, and advocacy groups. For additional information, I recommend that you refer to our Guidance for Industry, Considerations for the Design of Early Phase Clinical Trials of Cellular and Gene Therapy Products. Okay, now it's time for our challenge question. Challenge question number 1. First in human study for a cellular product should always be in otherwise healthy adults. Please raise your hand if you think the answer is A, true. Now raise your hand if you think that the answer is B, false. Yes, you are correct. The answer is false. Based on the benefit risk profile, typically first in human studies for cellular products should be performed in the target population. Now for challenge question number 2. Which of the following statements is not true? A, a clinical study should only be paused for an expected serious adverse event that is attributed to a cellular product. B, an appropriate staggering interval can mitigate safety risks to subsequent subjects from a cellular product. C, a safety signal identified during long-term monitoring following administration of a cellular product can inform benefit risk for potential future subject and continued product development. D, a blinded randomized controlled study should be considered for a first-in-human study to improve interpretability of safety and efficacy data and expedite product development. Raise your hand if you think the answer is A. Raise your hand if you think the answer is B. Raise your hand if you think the answer is C. Raise your hand if you think the answer is D. Yes, those of you who selected A are correct. A is not true, as study stopping pausing rules are specific to the individual study, but should not be limited to expected adverse events, as important serious adverse events can be unexpected, and it is important to pause the study to give time to investigate relatedness, as relatedness is not always immediately apparent. Thank you for your attention. Now we will take some questions.
Many thanks to our speakers for their informative talks. Now we will start our Q&A session. So we will take some questions from the audience. Our first question is for Dr. Brooks. So Dr. Brooks, do you require all definitive safety studies to be performed according to GLP? What if that is not feasible?
Thank you for that question. Although we definitely prefer or recommend all definitive safety studies be performed according to GLP, we recognize that that might not always be feasible. And so if that's the case, if it wasn't performed to GLP or if it cannot be performed to GLP, then we just recommend that you try to follow GLP guidelines. So conduct the study according to a prospectively designed protocol, perform the study in a non-biased manner, have appropriate record keeping and documentation of all data. We also recommend that non-GLP studies include oversight of the conduct of the study, and that each final study report is quality assured, or there's a person who's overseeing it that is independent from the conduct of the study. Again, just trying to follow some of those GLP guidelines. But again, we don't require it. We just strongly recommend it. Thank you.
Thank you, Dr. Brooks. We now have a question for Dr. Hart. Is data from a different disease that shares the same mechanism of action, e.g. anti-inflammatory, sufficient to support prospect of direct benefit in initiating my study in children?
Thank you. We actually get this question a lot as far as what amount of evidence and from what sources is sufficient to support prospect of direct benefit. Typically, prospect of direct benefit should come from the same disease. As we know, like the example, there are many inflammatory conditions, but each of these conditions can be very different and affect different organ systems, which would make it difficult to extrapolate the data from one disease or condition to another. However, if you believe that there's a particular reason that two diseases are similar enough that that extrapolation can happen, you are welcome to provide that justification. And irrespective of whether or not that data is going to support prospect of direct benefit, all information on the product should be provided within your IND to support safety and dosing.
Thank you, Dr. Hart. We have another question for Dr. Brooks. Does the product used in preclinical studies need to be produced according to GMP?
Good question. So the product used in preclinical studies should be as close to the clinical product as possible, especially for any pivotal pharmacology or toxicology studies. We want it to be as close to the intended clinical product as feasible in terms of manufacturing process, formulation buffer, specific product characteristics, etc. However, we do not require that it be manufactured per GMP. But for any products used in the preclinical studies, we do ask sponsors to provide a discussion and compare the differences and similarities between the products used in the preclinical studies to the intended clinical product. Thank you.
Okay, Dr. Hart, you have another question. You mentioned that staggering is required for early phase studies of cellular products, but I do not want to unnecessarily delay recruitment and treatment and prolong the study. How many subjects do I need to stagger?
Thank you for this question. Yes, we agree with you. There's a lot of areas where there's unmet medical need, and we want to help expedite the product development, especially in these areas. However, at the same time, we need to balance this with the safety of the subjects. The other thing you've heard throughout today is that, particularly when we're dealing with cellular therapy, and you'll hear a similar message tomorrow with gene therapy, is that we need to individualize for each of the products and populations. This is definitely an area where it's not a one-size-fits-all approach. So for a fatal and rapidly progressive condition, there may be a higher risk tolerance. And in that setting, fewer subjects would need to be staggered. However, for a population with a lower risk tolerance, especially if there were safety signals that were seen in the preclinical setting, it may be appropriate to stagger many more subjects. So this is a topic for discussion as it relates to your individual product and population.
Dr. Brooks, you have a question. What if there is no appropriate animal model?
Yeah, so this is a great question, and we recognize that for some diseases that require cell therapies, there might not be an available animal model. And so there are really two aspects to answer this question. One is related to efficacy or biological activity of the product, and the other to safety. So while there might not be an available model, we do still expect sponsors to provide scientific justification for conducting the clinical trial. And this can be derived from in vitro studies, a cell culture model that can demonstrate biological activity. It could also be derived from clinical data, if there is data from other similar indications that can be used to leverage or to provide a rationale. We do accept this at times. And we also recommend that sponsors come to us early on in their product development using the different meetings available, specifically the Interact meeting. This would allow a discussion between sponsors and ourselves to maybe go over some alternatives if there are no animal models available. What might be accepted and not accepted for your specific product and your specific clinical indication. And then if we're talking about safety, you don't necessarily need an animal model to evaluate safety. Typically, we expect safety studies to be conducted in normal, healthy animals. And so in this case, it's not as big of an issue. It's really to demonstrate the biological activity of the product. And as I stated, there are other mechanisms in which this can be accomplished. Again, great question. Thank you.
Thank you, Dr. Brooks. So Dr. Hart, you have a question here from the audience. Do you recommend double-blind, placebo-controlled trials in first-in-human clinical trials for rare diseases?
Yes. Thank you. The answer to that is yes. If it is possible, yes, we recommend that you conduct a double-blind controlled study beginning with your first-in-human study. It increases the chance that you can hit a home run. It will make data interpretability for both safety and efficacy much easier. Thank you.
Dr. Brooks, we have a question for you. If you use an analogous product in the animal model rather than the human product, what additional studies would FDA like to see to bridge between the human and animal product?
Great question. So we wouldn't necessarily need to see bridging studies. what we would want to see is comparability of the products. So comparability of the analogous cell product to the human cell product in terms of the cell phenotype, the cell function, things like that. And the, level of comparability should be sufficiently discussed in the IND submission. However, it should be noted that there are some studies that an analogous product is not appropriate for, such as a tumorigenicity study. For a tumorigenicity study, you really need to use the clinical cell product. Analogous product isn't sufficient for that. So, good question again. Thank you.
Dr. Hart, you have a question that came in that says, is it okay to not require a dose response? Are there certain exceptions? And I believe this is for tissue or bacteria.
Thank you. Before I answer this question, I wanted to just clarify my response to the last question related to double-blind, randomized controlled first-in-human studies. I think I started off that question by stating that this was when possible. That includes when things are ethical. So it may not apply in oncology. I was thinking more in terms of rare medical conditions, such as the ones that are regulated in my branch. I did not want to imply that this extends to every condition or every instance. So now let me answer your new question as far as dose response. And again, we realize that every product that we're dealing with when we deal with cellular therapy is different. And so for some conditions, it may be possible to do a dose ranging study, and that may be very informative. In other areas, it may not be feasible, but nonetheless, the doses to be tested should be those doses that you want to advance in your development program, such that there is adequate information about the efficacy and safety of the product you intend to move forward.
Thank you, Dr. Hart, for that clarification, as well as the answer to the second question that we had. So I have another question for Dr. Brooks. You have a question that came in. How can we implement ICH S6 for cell and biotech-based products to determine a regimen for preclinical safety testing?
Yeah, this is a good question. Unfortunately, IHC S6 was not written with cell therapies in mind specifically. So while it can be used as sort of a guide, we really recommend that you refer to our guidance, FDA's preclinical therapy. And then again, as I mentioned, if you're having trouble deciding on the best approach and you already do have some data and you have your product identified, request an interact meeting. It really is a great opportunity for sponsors to get answers to some of these questions and to make sure that they're on the right track when they're determining their preclinical program. Again, thank you.
Thank you, Dr. Brooks. And so we'll take one last question with Dr. Hart. The question is, what is required to support starting dose for a first-in-human cellular therapy IND? Animal studies may not be applicable.
Thank you. So this can be a challenging situation. And again, we recognize the need to individualize and that there is not a one-size-fits-all approach. Therefore, we do need to still be consistent with the regulations. So making sure that the dose to be tested is likely to be safe. If you have other evidence to support that your proposed dosing is likely to be safe and is appropriate for the proposed route of administration and the population, you should provide the justification that you have. We are very willing to review, consider, and look at that justification because we do recognize the challenges that are faced with these products.
Thank you so much, Dr. Hart, and thank you to Dr. Brooks as well for providing these informative talks and taking the time out to answer our questions. I'm going to now turn this session over to Dr. Lapteva, and she will provide us with closing remarks. Thank you.
Thank you, Shayna. Good afternoon again, or it's early evening here on the East Coast in the United States, but of course, different times in other parts of the world. We have concluded the first day of the CBER track and heard presentations and discussions about the regulations and other aspects of the development of human cells, tissues, and tissue-based products, as well as cellular therapies. This has been a long day in which we combined both device and biologics related presentations from the Center for Devices and Radiological Health and the Center for Biologics Evaluation and Research. Please do not forget to complete speaker evaluations. They can be downloaded from the link on the right hand side of your screen, and also the final evaluation, which can be found through the link on sbiaevents.com. I would like to thank all of our presenters, speakers, and moderators, as well as the audience for your active participation. We have one more conference day tomorrow, filled with presentations from the Center for Biologics Evaluation and Research to complete our conference. And we saved a number of interesting topics for this last day, and we hope you can join us tomorrow for more CBER coverage, including expedited product development, topics on gene therapies, CBER regulated devices, and manufacturing facility inspections. Have a great rest of the day, everyone, and we will reconvene tomorrow. Thank you.